Good morning. Welcome to the last third day of the annual meeting of ACC. Also today, a very busy program, starting with the highlight session number 3 that will be fully devoted to discuss access of patient to genomic screen and new drugs, a really important topic. Chairs of the session will be Gennaro Ciliberto, Ruggero De Maria, and Pier Giuseppe Pellicci. Thank you, and I leave the stage to you for this session. Good job.
Okay. Good morning, everybody. I'm very glad to be here and to be part of this highlight session 3 as co-chair together with Ruggero and Pier Giuseppe. This is the last highlight session on genomic screens. This is a a topic which is a critical and central topic to the strategy of Alliance Against Cancer, or to the strategy that Alliance has been following over the past 4 years or so. So we have, together with, say, internal speakers to the network, also external speakers, and we are going to hear what is the application of genomic screens and in general of genomics medicine in cancer and the opportunities that are being created by this new approach. So at this point, I will stop and I will ask Ruggero to introduce the first speaker.
Thank you, Gennaro. It's my pleasure to introduce Fabien Calvot. Fabien doesn't need an introduction because he's an old friend of Alliance Against Cancer. I remember the first time I met him was in Trascan, and he gave an incredible view of what research should be in Europe. And then he had a prominent position, was director of INCA, and great work at the Gustave Roussy, and he was the scientific director of the Cancer Core Europe. And so he has a great vision, and we thank him. We're very grateful for being here with us. Fabien is going to talk about genomics and drug development, the two revolutions in the last 20 years in cancer management and care. Thank you, Fabien. Thank you for joining us.
Good morning. It's a great pleasure and honor to open this morning session of the annual meeting of the ALLEANZA under this special format using video conference. My contribution this morning is about the two revolutions we have faced these last 2 decades in the management of cancer patients, which are both tightly linked, namely the revolution of genomics and of the drug development, which both contributed to cancer patient health. As an introduction, we are facing in Western Europe very similar observations. This is data from France, a very slow increase in incidence rates, which is less marked in women than men, and a marked decrease in mortality rates linked to earlier diagnostic, a better understanding by the patients of their diseases and its treatment, and progresses in the quality of care. The number of efficient drugs is increasing, as we will see, and we know better how to associate them, reduce their toxicities, and give the best treatment to the right individual. An example is shown here with children B acute lymphoblastic leukemia, which had a cure rate of 25% 50 years ago in the 70s. And this cure rate gradually increased up to 90% these days. We can predict that with the use of CAR T cells and other immunological treatments, we could be able to cure almost all patients in the coming years. When you look at the 69 drugs which were approved by the FDA for cancer care during the last 4 years, chemotherapy accounts for only 3 medicines, targeted drugs for 39, and immuno-oncology therapies for 17. In the previous period, from 2006 to 2015, hormones and chemotherapy represented more than 50% of market authorizations by the FDA. This evolution faceted the way we treat our patients and how we manage and perform clinical trials. The use of cancer genomics and biology has become of daily practice for the management of patients, as well as the understanding of immunological tolerance and activity, and their control will be key for the oncologist. We understand now better the genetics, inherited or acquired in tumors, the influence of epigenetics, stemness and differentiation, and a clinician has to be aware of this complex biology to treat the patients more smartly. Mutations and gene alterations have been described for almost 50 years in cancer, but the technology development have considerably accelerated the possibility to explore quickly and in depth the genome of cancers. Some early examples are listed here, leading for some of them to very efficient drugs. In brief, cancer is characterized by autonomous expansion and spread of a somatic clone. To achieve this behavior, the cancer clone must co-opt multiple cellular pathways to circumvent normal constraints of cell growth, adapt its microenvironment, invade tissues, and escape immune surveillance. These clones arise from mutation acquired during life with the contribution from germline and epigenetic variation. Briefly, our cells are exposed to multiple types of DNA damages from exogenous or endogenous sources, such as ultraviolet, carcinogens, spontaneous error, or DNA modifications. In response to these damages, specific mechanisms of DNA repair are activated, which in some cases are incorrect, and lead to errors, which are named mutations. These mutations are very different in numbers and types according to the initial agent responsible, its effects, and the germline background of each individual. This type of work led to a number of excellent papers signed by Alexandrov and colleagues about the mutational signatures in cancer, according to the variations which are observed in tumors. For instance, C2A, C2G, C2T, etc., that were observed. These signatures are characteristic of carcinogens, such as UV, tobacco, DNA repair defects, enzymatic defects, and many other responsible agents. These observations were also extended to the analysis of doublet possible variation, and grouping cancer this way may direct us to the detection of the main causes at the origin of cancers. There were two major international projects to address the genomics of cancer, one stemming from the USA, TCGA, the other with a more international flavor, to which I contributed for 10 years with other excellent colleagues, such as Thomas Hudson, who has been key for this, Harold Varmus, Peter Lichter, Mike Stratton, and which was named ICGC for International Cancer Genome Consortium. ICGC is a real international collaborative effort, including geneticists, ethicists, lawyers, pathologists, clinicians, bioinformaticians. And you can see on this map the variety of tumors studied, including the contribution of the lead from Italy in the Verona University, Aldo Scarpa and his colleagues. The idea was to cover extensively all the aspects of cancer genomics and cover all tumor types. Several papers were published in Nature in the issue of April 2020, which present a part of the enormous work which associated hundreds of researchers across the globe. In summary, more than 2,500 donors of tumors and blood samples have been studied, with complete DNA sequencing, RNA sequencing, mitochondrial DNA sequencing, predicted tumor protein modelization, search for infectious DNA or RNA, and so on. Among the 43 million somatic SNVs observed, the 2,400,000 somatic indels, it can be observed that there is heterogeneity between patients and across tumor types, and that 91% of the explored tumors have at least one driver mutation, a mean of 2.6 drivers per tumor, including 13% in non-coding regions. As can be seen here, the mutation rates are variable among different tumor types, the higher being for SNVs in melanoma, lung, esophageal, colorectal, and bladder cancers. The lowest being in central nervous system tumors, leukemia, thyroid cancer, breast cancer being in the middle. The variation can be huge within a cancer type, and as you can see for melanoma, from 100 to 1 million, for instance. The panorama of driver mutation observed is depicted in this complex slide. As I told you, in total, 91% of tumors have an identified driver. Rearrangement are observed in 26%, mutations in coding region in 76% of patients, in non-coding regions in 26%. And in this population, driving germline mutations occur in 17% of the patients. On the right panel, you can observe the genes involved by number of patients and the type of mutations in coding, promoter, or introns, and the type of copy number alterations in somatic variants. On the bottom, the graph shows the importance of B allelic inactivation present in more than 10 cases from patients. Of note, the importance of deletion of cyclin-dependent kinases, CDKN1, CDKN2A and B, P10, SMAD4, RB1, and many others. Almost 10% of tumors did not show an identified driver, possibly explained by technical or biological causes, including failure with the informatics algorithms responsible for half of these. There remain, at the end, 5% of tumor in which could not be identified a driver. A recent paper by colleagues from the Netherlands described using whole genome sequencing the mutational landscape in metastatic, heavily treated patients and could identify signature of the use of drugs, but importantly, an actionable driver in 62% of the patients, either in oncogenes, tumor suppressor genes, or germline predisposition. Actionability in this case means using a marketed drug or one drug under current development. This brings us to the second part of my talk today, how all these has impacted our patients' treatment. It had consequences through precision medicine. We identify targets and we treat an individual with a specific tumor with the most appropriate treatment. All this has changed the design of clinical trials and led us to adapt to this paradigm. we need biological platforms to sequence genes and immunology platform to follow and understand who is going to respond and who will not. To illustrate this change in clinical trial design, I represented here the classical phases in use not only for cancer drugs, but for all medicines. Phase 1 is dedicated to safety, determination of a maximal tolerated dose, exploration of different schedules of administration, and usually include 20 to 50 patients. Phase 2 aims at reaching a proof of efficacy and to determine an overall response rate. Usually 100 to 500 patients are included. And phase 3 has a goal to compare the new treatment with the standard of care and usually need more than 1,000 patients. The mean duration of all this step before registration and market authorization is long, about 10 years, with many failures, and is very costly. This scheme has considerably changed with the advent of targeted treatment. It is now a mixed phase 1, phase 2 trial, aiming at defining a so-called tolerable dosage along with drug activity demonstration. Toxicity and response rates are essential, as well as survival. This phase 1-2 mixed trial is followed by phase 3 to eventually be compared to standard of care and to evaluate in depth progression-free survival and overall survival. It is essential to address at the same time the clinical aspects and the biomarker development. These clinical trials have a high success rate and the registration can be very quick. For immuno-oncology treatments, the classical phase 1 is usually reduced and completed by an extension cohort to define active dosage, safety, response, progression-free survival, and currently needs 100 to 1,000 patients who are frequently pre-selected. This phase 1 may reach conditional registration. As an example, pembrolizumab gained approval independently of tumor type for microsatellite instability high tumors. As shown here as a summary, with chemo, molecularly driven drugs, and immunostimulating antibodies, the number of patients needed to be enrolled is variable. Maximal tolerated dose is not always a goal. Pharmacokinetics and pharmacodynamics, which is important for chemo, is replaced by biology, genomics, circulating tumor DNA for targeted agents, and biomarkers for immuno-oncology treatments. Therefore, you can see that MPDL3280A, which is atezolizumab, needed to include 334 patients in an extended phase 1 trial for metastatic bladder cancer. The other drugs, pembrolizumab, nivolumab, ceritinib, and crizotinib, as you can see here, needed very low numbers compared to previous market authorization with chemo. Coming back to targeted treatments, there is a need to co-develop drugs and biomarker to be flexible to incorporate adaptive frameworks as compared to the rigidity of classical clinical trials, and obviously to have in the same site or to have an easy access to performing platforms for genomics, immunobiology, circulating tumor DNA detection and sequencing, and excellent facilities for biopsies. And finally, to be able to include patients fastly with various tumor types and therefore have close ties with organ specialists. When speaking about adaptive design, it is important to adapt the number of patients to be included, the selection of patients according to the clinical setting, and biological data to explore different doses and schedules during expansion cohorts and to improve the follow-up of patients with excellent up-to-date technologies. Therefore, it took 4 years for the identification of ALK activation in lung cancer to the market authorization of crizotinib, and to go from the use of the third generation of epidermal growth factor receptor tyrosine kinase inhibitors in 2008 to the identification of mutant-resistant T790, to the generation of subsequent inhibitors, and the authorization of osimertinib in 2016 after only 3 years of clinical trials. And this was developed along with the development of a biomarker testing circulating tumor DNA. I would like now to end my presentation with some cases I had the opportunity to participate from the Gustave Roussy Institute and CANCRO Europe. First, the Moscato trial, which was led by Jean-Charles Soria and Christophe Massard, was one of the first to demonstrate that the use of genomic can benefit patients. It included more than 1,000 patients for whom a molecular profile could be established in 81% of them, and an actionable mutation identified in 41%. A drug was available locally in 20% of the initial cases. In this non-randomized trial, each patient was his own control, the endpoint being the duration of response, progression-free survival 2, which had to be superior, at least 30% superior to the previous progression-free survival PSF1. This endpoint for patients for which a drug was available was achieved and demonstrated a general benefit of using genomic to treat advanced cancer. These 20% of patients treated have to be compared now with the theoretical potential of 62% of metastatic patients, as shown in the Priestley study in Nature 2019. The second case is shown here with erdafitinib, fibroblast growth factor receptor inhibitor. This multicentric trial was headed by Josep Tabanero. During the extension cohort, it was shown that the inhibition of activated forms of FGFR is only efficient when there is a translocation of the receptor, no response being observed with mutation of amplification. Therefore, most druggable genomic alterations are identifiable in a growing number of patients. The main difficulty at this point remains the drug access, both to the patient and their caregivers. There will be here a real challenge which needs to be addressed by collaborative actions. Building this at the national and European levels is mandatory. And second, as seen, acquired resistance is a major clue and will need important and costly efforts from biologists and medicinal chemists to overcome and generate second and third generation of products. Acquired resistance is less common with immune therapies, either monoclonal antibodies or cell therapies. The clinical trial design is held by early signals of efficacy. As you can see here, a large variety of cancer is sensitive to immuno-oncology treatment. And this will probably increase with a better understanding and efficacy of drug association and the development of biomarker of sensitivity. The most recent approach is the response of triple-negative breast cancer associating cancer and anti-PD-L1. To end my presentation, I would like to comment on the collaborative work which will be mandatory in the future, as I said, nationally and internationally, and especially within Europe. At CANCRO Europe, which is one network which need to be reproduced and expanded, we launched a new adaptive clinical trial, a basket trial, where all patients' tumors are first submitted to a 400 gene panel sequencing, then, if included, to one of the active arms, whole exome sequencing and RNA-seq is performed. To analyze and help clinicians decide the allocation of treatment, the tumor board portal was set up with automated data analysis, virtual tumor board reporting data, and recommendations according to expert consensus criteria and trials databases. This is an example, but a very smart one, where collaboration, including various specialties, may change the way cancer research and cancer care are conducted. With this, I finished my presentation, and I thank you very much for your attention.
Thank you very much. Again, your talk was really informative and gave us a great view of what is going on. Before starting the question from the audience, I have something to ask you. It's something that you didn't cover. So it's very likely, or not very likely, it could be that one setting that is very promising, it could be very effective, is the adjuvant in the target therapy scenario. Yesterday, there was a paper in the New England Journal of Medicine in which patients with EGFR mutation, stage 2 and stage 3A, were treated with adjuvant osimertinib. And after 2 years, the 89% of patients taking osimertinib were disease-free versus 52% with the placebo. So it's probably that this setting could provide us maybe more efficacy in terms of overall survival. It's still early to understand this. I would like to have your opinion about this.
I absolutely agree that it's very different than what we used to do with chemo. And when you look at the example of chronic myeloid leukemias, the earlier you use a treatment, the best results you get. And it is exactly the same when you use monoclonal antibodies such as trastuzumab. The earlier you treat the patients, you almost arrive to a complete cure of the patients. So we will have also to address differently in the context of clinical trials to which patients we should treat. And they probably, with these targeted drugs, to avoid the development of resistance, the earlier you treat the patient, the best should be. The second aspect is obviously between the type of drugs you use. Of course, monoclonal antibodies are much more efficient than usual kinase inhibitors or enzymatic inhibitors. And this is also something you have to take into account, and especially in the context of early treatment of patients.
Thank you, Fabienne. There's a question for Bruno Amati. Are there any mutations that are characteristic of metastasis as opposed to primary tumors?
It's very contradictory, but it seems that when you look at the recent papers exploring metastatic disease and comparing the drivers between the initial phases and the late phases, it seems that there are additional, but the majority of the drivers are still, the initial drivers are still present during the second phase. And when you look at the metastatic sites, the drivers were also present at the initial phase. So it remains a complex issue to be determined. The main difficulty is obviously the influence of the treatment that is given to the patient between these two periods. And obviously there are some specific pressure due to drugs that affect the genomics, but on specific enzymes and or specific targets.
Thank you, Fabienne.
I have a question.
Please go ahead.
Actually, I have two questions for Fabienne. The first is, I'm sure that you are familiar with the Dutch drug trial, which is a sort of basket trial. And so the question is to you is, what do you think about the trial and the results that are being obtained, and how the trial how much the trial is different from what CANCRO Europe plans to do? The second question is related to, you mentioned the issue of resistance. This, at least in large part, might be due to presence of co-mutations and the existence of coexistence of different clones with different mutations, and the fact that in order to, in this case, to fight resistance, you probably would have to develop combinations, and the present basket trials do not exploit the possibility of co-treatments and combination treatments in order to fight resistance.
It's very important problem that you raise here, and especially I guess the problem of combination treatment. We have been so far using only monotherapies with targeted drugs, and we have been using them at a late stage, and probably this will be the change that we can expect in the next years now, that combinations have to be explored. Probably it should be a major point, especially for using immuno-oncology treatment, because probably the addition of chemo, for instance, of radiation, although it's not very clear at this point, that it may potentiate the efficacy of immuno-oncology treatments. About the basket trials, I mean, basket trials can explore this. I mean, the idea of basket trials is, and especially of baskets, is that you have to identify a sign first, and then you can add a new arm with combination if you have a signal of activity. And it's probably what should be developed in the future, is really to have the possibility of extending one arm to another arm with the addition of drugs to the previous one. The main difficulty for this remains, of course, the the freedom of the researchers to do that, especially in the context of collaboration with the pharma industry, which is not very, I mean, they are not very open to this type of progression. And for this, I believe that we need academic trials. We need international collaboration for that, because I mean the number of patients is quite low when you go into these developments, and probably it's also the role of charities to help charities and networks to help finance this type of clinical trials.
Thank you.
Thank you.
Excellent, Fabienne. Thank you very much for joining us.
The pleasure, Ruggero.
So I will ask Pier Giuseppe to introduce the next speakers. Thank you, Fabienne, again.
Thank you.
On behalf of the ACC Working Group Lung, we will present preliminary data progress of our study and satellite studies. To give a brief of the background, we would like to remind that lung cancer is the leading cause of cancer-related death worldwide. More than 85% of the cases are classified as non-small cell lung cancer, with predicted 5 years survival of 16%. Technological advances, including the introduction of next-generation sequencing, have allowed for the identification of several genetic mutations, opening new roads for the therapy of oncogenic-driven non-small cell lung cancer. These can be targeted with selective inhibitors. However, most patients with non-small cell lung cancer do not harbor these oncogenic drivers, and for these patients, treatment options rely on PD-L1 status to proceed or not with immunotherapy or immune chemotherapy. In Italy in 2017 and 2018, the testing rate for actionable and druggable targets was considered low, and the number of molecularly classified non-small cell lung cancer patients was considered still insufficient. Therefore, our mission, given the high number of actionable genes and targets in non-small cell lung cancer, is to improve molecular diagnostics in Italy using NGS-based technologies on small tissue biopsies and to generate big data. ACC Working Group Lung program involves today 23 centers all over Italy, and today we welcome 2 more centers, Parma and Pavia, that have joined us this year. Let's go a bit more in detail in the study and have a look at the inclusion criteria. In the presence of suspected lung tumor, patients enrolled in the study are all consecutive patients with histological or cytological diagnosis of advanced suspected non-small cell lung cancer, treatment naive or after one-line therapy with incomplete molecular profile. Moreover, our primary objective is to validate the ACC lung panel as a tool for molecular profiling in patients with non-small cell lung cancer, comparing the frequency of activating mutations and translocations observed with NGS and routinely used techniques. Our secondary objectives will involve the evaluation of the samples and the panel. In particular, we will determine the percentage of enrolled non-small cell lung cancer patients with an adequate amount of tissue to perform NGS, and we will investigate of those critical issues that have determined unsuccessful molecular diagnosis. Moreover, we will evaluate frequency of the oncogenic alterations identified by NGS in comparison with the data from the literature and routinary techniques. Secondary objectives will also comprehend the assessment of survival impact and analysis of the performance of each center participating in the study. In order to achieve our aims, we have created and distributed to all working centers a manual to guide the work at the clinical and pathological, genomics, and bioinformatics level. According to our timeline up to date, we have enrolled 502 patients on a target of 1,000 of total. We are also conducting the comparison between tissue and liquid biopsy NGS analysis, and we have activated the radiomic study. Thanks to our partnership and important teamwork, we will aim possibly to reach 1,000 patients enrolled by the end of 2022. If we look at the enrollment status of all centers participating in the ACC lung program, we see that San Raffaele, San Giovanni Rotondo, Hospital of Catania, and San Martino from Genova have already fulfilled the expected enrollment of the patients. All centers are reaching the target. Other centers are reaching the target. You can see here, these are Humanitas, IRE, and Mendola. More centers are actively recruiting. You can see here, IOV Padova, Bari, Villa Sofia from Palermo, CROB Potenza, Parma Hospital, and Naples, and Candiolo. Canizzaro, Reggio Emilia, Aviano, and Yeo have stopped enrolling since January 2020, and other centers have never enrolled, such as La Maddalena, ED, INT di Milano, and Gemelli. Preliminary data extrapolated from CRF show that the median age in years is 66. Male gender is more representative. 90% of enrolled patients were four-staged, and 25 of them had brain metastasis. Most of the patients are treatment naive, and a really minority have received a first-line or an adjuvant therapy. Successful histological diagnosis has been performed in 92% of the patients, surpassing the expectation of 70%. Here we present interim analysis data on mutation techniques, mutation frequencies, and mutation variants for KRAS, EGFR, and BRAF obtained with standard molecular analysis starting from the tissue biopsy. Molecular NGS profiling analysis is currently ongoing. Among patients' clinical data we are collecting, the smoking status represents an interesting point for future data analysis, since 21% of the patients never smoked. Our question is whether molecular profiling will change over the years, also given that in 2003 smoking in public places have been banned in Italy, and a significant number of patients quitted smoking after low actuation. While the ACC Working Group Lung program aims to validate the ACC OncoChip Lung panel as a tool for molecular profiling, it includes multiple satellite and other studies. Here you can see some of them and the most important, such as liquid biopsy study, radiomics study, and immunoscore study that we are developing in collaborations with immunotherapy and lung working group. Moreover, one more independent study has been activated in the context of molecular profiling for stage 3 non-small cell lung cancer patients, and another one to validate Mirna signature predictive for neoadjuvant chemotherapy response in stage 3 but N2 non-small cell lung cancer. We will now go through the presentation of satellite studies and other studies that have been activated. The satellite study liquid biopsy is coordinated by Dr. Chiara Lazzari from San Raffaele and Dr. Patrizio Giacomini from IRE. This study aims to apply NGS panel in liquid biopsy and will compare NGS analysis performed on liquid biopsies with NGS analysis performed on tissue biopsies. Sample collection is ongoing from the start of the protocol, and 102 samples are being processed at San Raffaele and IRE, of which 40 patients have been screened using the Oncomine lung cell-free DNA assay. 62 patient samples analysis have been conducted with Oncomine pan-cancer cell-free assay, and 125 more patients have been identified, and the analysis of those are depending on the fundings. Preliminary results shows 74% of concordance rate between tumor tissue DNA and cell-free DNA, with a sensitivity of 79% and specificity of 82%. This study, this satellite study, is currently conducted in 16 centers of the working group lung. Let's have a look for the second satellite study, which is the radiomics study. This study is coordinated by Luca Boldrini from Gemelli and Professor Daniele Regge from Candiolo. And this study will correlate medical imaging features with genomic and clinical data of 300 patients involved in ACC lung study, with the aim to improve the prognosis and to develop radiogenomic models that can predict tumors' mutational triggers. Up to date, the amendment has been already approved for 18 centers, and we have had an investigators meeting in September this year. In the next future, we will aim to validate IT infrastructure to create an instruction manual to harmonize the anonymization procedures and imaging segmentation system between centers. Importantly, we will identify candidate patients, and we intend to provide data analysis and the report of this satellite study by 2021. The third interesting study, which is a separate independent study, is ICT Mirna predictive signature study coordinated by Dr. Fabrizio Bianchi from San Giovanni Rotondo. It is a new protocol which has the objective to validate Mirna signature for 100 patients that can be predictive for neoadjuvant chemotherapy response in stage 3 and N2 non-small cell lung cancer. This study has reached already 48 patient samples analysis. Other 55 patients have been already identified and collected at San Raffaele. We suppose that we are going to overfill our expectation to reach 100 patients exactly next year, because other 6 centers have joined us in this study, and they give their approval to participate in the study. And there are no problems with the fundings because this study is covered by IRC funding. Another independent study has been created within the working group LUNG. It is already a new protocol that has been submitted for ethical approval in October 2020. This study has been redacted given that stage 3 non-small cell lung cancer patients have not been up to now identified for their tumor molecular profile worldwide, and information about molecular profiling for this category of patients are still missing. 24% of non-small cell lung cancer patients have a stage 3 at the time of the diagnosis, and more than 50% of resectable non-small cell lung cancer patients relapse within 5 years. Moreover, the prognosis of these patients remains really poor, and currently clinical trials are evaluating neoadjuvant immunocheckpoint inhibitors. Stage 3 non-small cell lung cancer patients are characterized by high heterogeneity related because of the disease, patient's characteristics, and interinstitutional variability, and also multitude of therapeutic options. And this is the reason why we have designed this study that is focusing on stage 3, with the aim is to molecularly classify 500 patients using genome exome sequencing, whole exome sequencing, VES, and to correlate molecular classification with patients' clinical outcome. We have had an investigators meeting in September, and soon we are going to start to define the database with potential patients to be enrolled and to be analyzed for this study. The secondary aims will include the development of radiogenomics models for the prediction of the treatment response, overall survival, disease-free survival. We will correlate the radiomic signature with the molecular characteristics, integrating molecular, clinical, and radiomic features. The protocol workflow expects the tissue section from the surgical tumor samples and normal tissue DNA extraction will be sent to San Raffaele for whole exome sequencing, while DICOM files will be sent to IRE. We expect to complete patients' enrollment by April 2021 and to complete the VES and bioinformatic analysis by October 2021. 16 centers within the working group LUNG are participating in this stage 3 study. In conclusion, ACC working group LUNG have generated a network of institutions capable of performing molecular profiling using NGS. A completed interim analysis is expected for 500 patients by March 2021 and is going to be submitted for the publication. Also, in 2021, we are planning to verify discrepancies between NGS and standard analysis at the end of the study enrollment. Our ambition is to generate big data for non-small cell lung cancer patients, including liquid biopsy, radiological, and clinical data. As you can see, we are very close to reach 1,300 patients by 2021, and we plan to conclude the ACC lung study in 2022, reaching 1,500 total of patients, of which 500 will be stage 3, while 1,000 stage 4. Radiomic data for 800 patients are going to be available, and for the moment, regarding the liquid biopsy, at least 225 patients will be screened for liquid biopsies by the end of 2021, and we hope even more based on funding by 2022. It is a great effort, but also great satisfaction of great and important team that I represent, which is Working Group Lung. And I want to thank all centers, all patients, all families, all institutions, Alliance Against Cancer, that are supporting us in this ambitious program. Thank you.
So the speaker was Dr. Vanessa Gregor from Hospital San Raffaele, and she's also the PI of the ACC lung cancer project. Vanessa, I apologize. I didn't catch the time to introduce you, and I did it now. Thank you very much for your presentation. I think that we are now ready to take questions. I don't see question yet on the screen, so I will start, Vanessa. And I guess, I mean, since the study was started in 2018, you start seeing also relapses, and those patients have been treated, and unfortunately, some of them might have relapsed or relapse in the next months. And I'm wondering whether did you plan starting a study for same omic analysis, same granularity of analysis for the relapsing tumors, which, you know, that's very important, I guess.
This is an excellent question. In fact, up to now, we didn't start this activity because, as you can see, there was a really huge activity to be done to start all these aspects from molecular screening to radiomics. But now, in fact, we have to focus on, first of all, to study better discrepancies between NGS and standard techniques, because we see more patients with positive mutations with NGS with respect to standard. And the second thing is, obviously, it's important based on these so many patients that we study the resistance mechanism. Because especially for ALK and EGFR, they are quite well known. But we have detected BRAF mutations, we have detected RET mutations, MET. So obviously those subgroups are less studied. So it is a great opportunity because these patients are treatment naive when they started, and they are not heavily pretreated patients, and that means that we don't have a lot of interactions with the resistant mechanism. So it must be our next objective. With Luca Mazzarella, we have already discussed about that, and probably we have to include this in the next year, 22 or 21 or 22.
Thank you very much, Vanessa. I don't have yet a question from the audience. Are there questions from my chair colleagues?
I can ask a question. So, Vanessa, it's been a very nice overview, and there is a lot of work going on that has been done and going on. So the question to you is, can you say, because this is going to be important also for all the other working groups, what are the major issues and lessons learned during these past years which can help us to better approach further studies?
So first of all, we are different. Our X are different at the different stage and different equipments. So first of all, we have to always create a core of those who are capable to reach the objective, and then try to and start the trials or protocols, and then work on including other centers. If we wait that all centers are able to start in the same moment, in the same way, we will never start. So this is probably the first thing. The second thing is to think that we, in fact, are a network and that we have to help each other. So in the last years, I can say that I even quite sometimes I do mistakes. I don't remember anymore who is from which IRCs, because we really work as a group. And this is another important thing. And the third one is to be clear about the publication policies. It is very important, because if we clear this at the beginning, everybody knows that if they do something, they will be recognized. And if they don't, they are not going to be recognized. I think this should be very fair. This must be fair. So this is some, I think, ethical also things.
Thank you very much. Gennaro, sorry, did I interrupt?
Oh, thank you very much.
Okay, sorry. Yeah, Vanessa, thanks a lot. And, go ahead.
Thank you, thank you.
Please. Okay, so we go ahead with the next speaker, which is Dr. Domenica Lorusso from Policlinico Gemelli, Rome. The title of her talk is the ACC Gerson trial. She's the PI of this trial. Please, Ketta.
On behalf of my co-investigator, I'd like to thank you, the scientific committee, for giving me the opportunity to present the ACC Gerson trial. At present, the diagnostic pathway for identify actionable mutation, I mean mutations that are informative for prognosis and treatment, and those for genetic risk mapping, I mean hereditary variants of cancer predisposing gene that we call in the future CPG for the rest of this talk, are distinct. However, there is a considerable overlapping between CPG and mutated genes in tumor, because 10% of mutated genes also confer cancer susceptibility, and 40% of CPG are also mutated in tumor. Moreover, there is an expanding fraction of CPG which is informative for prognosis and therapeutic stratification. These are the so-called actionable genes. All these data suggest that the two pathways are destined to merge. Actually, many of the available mutational pathways already contains CPG, and this will increase the number of identified CPG variants and likely will increase the number of patients and families to candidate to genomic counseling and also further diagnostic investigator, and this will have an increase in cost. The Gerson trial is a feasibility study aiming at joining in a common pathway the genomic diagnosis, declined either in terms of genetic risk, but also in terms to evaluation of sensitivity to new drugs in a population of breast, ovarian, and colorectal cancer chosen because particularly enriched for genomic mutation. The feasibility basically refers to two main elements: the technical validation of a panel for mutational analysis that include actionable genes and CPG within a national network of laboratories, and the sharing of a common clinical management of the resulting information, both as regarding the somatic information which aim to the choice of a treatment, but also regarding the germinal information in terms of risk reduction for patient and family members. In particular, what we will share within the network centers participating in the study is the clinical significance, the evaluation of clinical significance of variable of uncertain significance, the promotion of clinical studies, and also the sharing of rules for the management of data protection, informed consent, genetic counseling, and the management of incidental finding. This is a very busy slide, but I want only to underline that the study has a main objective, which is for sure the validation with standard methodology of the germline and somatic mutation identified within the panel, but have also a lot of secondary objectives that can be merged in 3 to 4 points in particular. The objective aim at the validation of the test in the 3 population under study in terms of prevalence of germline and somatic mutation, prevalence of copy number variation, and frequency of 196 drug-related metabolism SNPs included in the Gerson panel. Some endpoints are related to the reproducibility of the test in terms of reproducibility of the gene panel used among individual workers, determination of percentage of mutation that are validated with the standard method, definition and validation of a path of clinical interpretation of the booth, evaluation of clinical applicability of the panel in terms of timing oriented to the decision-making progress, but also percentage of sample suitable for analysis. And we also aim to establish a macro-regional network in laboratories joining the trial and the validation of sharing in terms of protocol for the management of ethical and legal problems connected to the oncogenetic pathway. And very recently, the trial has been amended with the idea to add another type of analysis, and in particular, the characterization of the transcriptome with the low-cost parallel massive sequencing strategies of patients enrolled in the trial. What is this panel? The Gerson panel was developed within the ACC Genomic Working Group. It is a tool, an instrument to allow comprehensive germline and somatic mutational profiling for patients with multiple solid tumors. The panel includes 172 cancer genes, 295 tumor-alterated genes, and 196 pharmacogenomic variants. In this congress, Dr. Mazzarella presented the data of the technical validation of the ACC Gerson panel with respect to standard procedure, but also across samples on clinical specimen collected from 9 institutions, and the sensitivity of the test is very high, either with respect to the standard validation, either to respect to the clinical specimen. What type of patient we decided to enroll in the trial? We tried to select a population potentially enriched by genomic alteration. And in general, our idea was to prospectively enroll patients with ovarian, breast, and colon cancer who will receive a biopsy and a blood sample at the time of enrollment. We had the possibility to include patients who have previously received only 1 chemotherapy line, providing that the tissue sample of the primary tumor is available and not older than 2 years because of a possible problem in acid nucleide extraction. And according to this framework, we decided that all newly diagnosed patients with ovarian cancer, with the exception of borderline tumor, can be enrolled. Patients with relapse of disease who have received no more than 1 previous chemotherapy line can be enrolled. In order to facilitate the recruitment, a small part of the patients enrolled in the trial may be collected retrospectively. So we opened the possibility to enroll patients without evidence of disease enrolled during the follow-up, providing that these patients have not received no more than 1 previous chemotherapy line, and the paraffin-embedded tissue from the no pretreated primary tumor is available and not older than 2 years. So all ovarian cancer will be enrolled, triple-negative breast cancer will be enrolled, or breast cancer arising in patients younger than 40 years, and colon cancer patients younger than 50 years can be enrolled. The most important exclusion criteria had the inability or unwillingness to undergo oncogenetic counseling, more than one previous line of chemotherapy, the non-availability of no pretreated tumor tissue within 2 years in case of patient enrolled in the absence of disease at the time of enrollment, or the impossibility, inaccessibility to biopsy for patient enrolled with active disease. This is the patient flow that we are discussing inside the board. If patient, what the PI requested, this was a general request for all the PI joining the trial, that the flow, the journey of the patient enrolled in the trial is as close as possible to the flow of the patient treated in each institution and receiving a genetic test. So according to this suggestion, we try to design a patient journey which is absolutely respectful of what the center usually did. So when the patient is potentially eligible, we decide that it's the oncologist that proposed the study and discussed the informed consent, but there is the possibility to organize a meeting with a dedicated clinical genetist if required by the patient. If patient accepts the trial, blood and tissue sample should be acquired and the Gerson panel analysis performed. The Gerson report will be not returned to the patient. This was an aspect that we discussed a lot with all the PI, but in general it was agreed, according also to the ethical committee of the trial, that the report of the Gerson will be not returned, and what will be returned to the patient is a final report that will be produced after the validation of Gerson results by standard procedures. The report of Gerson will be discussed at each institutional level into a board composed by oncologists, geneticists, biologists, and pathologists. The most tricky cases, the cases for which there are no defined guidelines or for which we have no enough evidence that the genomic variation can be translated in clinical recommendation, will be discussed in a MTB, which is a trans-institutional board. For sure, all this flow will be reassessed periodically. In terms of returning of the test, it was decided that C4 and C5 CPG with well-defined management strategies and C4, C5 GPG with management strategies not well-defined will be returned by the genetist in a standard genetic counseling. For the boosts in CPG for which clinical trials are available for the patient or for the families, either in this case, a genetic counseling will be offered to the patient. The only situation in which will be the oncologist to discuss with the patient the results of the test will be the situation in which we identify C4, C5 a modification only in tissue in actionable genes. There are well-fixed rules for the biological sample management, and blood sample and tumor sample collected during surgery will be in parallel used for germline somatic analysis, and the tissue sample must contain not less than 30% of tumor tissue. And what we decided to implement with the amendment is the possibility to preserve the tissue at 4 degrees in a so-called cold fixation, for which a manual has been provided to the center. This probably will guarantee more in terms of nucleic acid extraction. How many patients? It was very difficult to provide an appropriate sample definition in this exploratory trial, but we decided to enroll a huge number of patients, in particular 1,500 ovarian cancer patients, 105,000 breast cancer patients, and 1,000 colorectal patients within 3 years. The trial is very difficult, so we decided to provide the trial with the governance, and in particular a steering committee was created with the president of ACC and also the scientific coordinator of ACC, the trial PI, and all the coordinators of the working group of ACC involved in the trial. And the first action of the steering committee was to decide that this trial required the tumor mutational board, and we decided to have this board across institution. 14 members from 17 HECs were enrolled in the board, including oncologists, molecular biologists, clinical genetics, bioinformatics, biotechnologists, and pathologists. All these persons are recognized with the high-level expertise in the field. The board will have periodic meetings to discuss cases on clinical institutional board requests, but in this moment, the board are doing a lot of work in terms of preparatory meeting to find a consensus on several very tricky issues, because the study is tricky. Gerson trial is a challenging trial which poses a lot of issues and a lot of questions regarding several aspects. How to define actionable alteration in the germline somatic level, what type of variant should be reported in the test, in the report, if all the variant of only that associated with possible therapeutic or preventive strategy, what is the appropriate method for consent administration, and what is the granularity of the consent if the patient is able to express consent of all the aspects, or a limited consent should be required. On this issue, we are working with patient advocacy group to receive also their advices on this. What kind of genetic counseling should be performed, and what will be the sustainability for the healthcare system given the increased number of patients requiring genetic counseling, and at least not least the access to the drug for this patient and the technological infrastructure. So actually, the state of the art, we have contractualized a CRO. Actually, the protocol has been approved in 15 out of 21 joining institutions, and 6 of them also approved the amendment, and 12% have also the contract finalizes. The electronic CRF is in preparation, but we are discussing a possible joining of this data with the Health Big Data project, which has been already started in ACC, and on the right, the list of participating centers that I want to thank deeply for their effort in this very important trial. Thank you so much for your attention.
Thank you very much, Keta, and thanks for the extensive presentation, the work that you are doing. So while I don't see questions yet. I start with one question, Keta. I was wondering, as a general issue, clearly I guess that the time needed to provide a molecular profile to the doctors, it is important. I mean, here there are patients which might require decision made on molecular genetics pretty quickly in a matter of, I would say, weeks. How did you consider that? What do you think about it?
One of the secondary endpoints of the trial is dedicated to this point. So in what time the results of the test are able to impact on the clinical decision making for the patient. Particularly, the problem may arise for probably colorectal cancer, in which we need very, very soon the molecular classification, and for breast cancer. For ovarian cancer, we have a different situation because in the setting of choice for the trial, the possibility to have the biological agent in terms of treatment is related to the maintenance treatment. So more or less, we have 4 months of chemotherapy, and so enough time. According to this, what we decided with all the other PI is a sort of fast track inside the trial, a sort of fast track for patients for which the results of the test have an immediate impact on treatment, and more relaxed time for patients for which we can wait.
And Keta, quick means what? Less than 1 week?
No, you are very optimistic. Quick means less than 2 or 3 weeks.
2 or 3 weeks. Okay, Keta, thank you very much. Let me see if there are no questions yet. Other questions from the chair?
No, I just want to thank Keta, because this study is very complex, and we also added some layers of complexity by adding the analysis of whole genome polymorphisms and the transcriptome analysis. So, it's already very difficult to analyze all the genetic predisposition to deal with the relatives of patients who has the pathogenetic variant that increase the risk of cancer. So very complex project, and so I thank Keta and all the investigators for all the institute for working on this project.
Okay, then I guess we go ahead with the next speaker, being Dr. Dr. Concetta Quintarelli from Hospital Bambin Gesù in Rome. And the title of her presentation is CAR-T Program in Alleanza Contro il Cancro, Activity Framework and Advances. Concetta, please.
Good morning to everyone. I have the honor to present our CAR-T program in Alleanza Contro il Cancro, showing the activity framework and the advances of this year. In particular, the program for CAR-T in Alleanza is divided in 6 different VP with specific aims. The first one for the definition of production process able to contain the cost of CAR-T. The second one, identification of new targets. The third one, the preclinical development of CAR-T against novel targets, either for solid tumors or hematologic disease. The number 4 is for the identification and validation on novel strategy to increase the activation and expansion, but also survival and tissue penetration of the CAR-T. The VP5 is to increase the safety profile of the CAR-T, all in the framework of legal aspect and intellectual property that are supported by the VP6. This is a 2 years program that maybe will be extended to 3 years with the scientific coordination of Professor Franco Locatelli in Ospedale Pediatrico Bambino Gesù, that is also the leader for the VP1 with other 2 institutions, Smolmed and Fondazione Tettamanti, try to optimize the process, increasing the efficacy and reducing the cost for CAR-T production. The VP2, as before underlined, has focused on the identification of novel target. In this case is Gemelli, the leading institution with all these other institution working with Gemelli. For the VP3 is the development of in vitro and in vivo preclinical models. In this case, the VP leader is the Candiolo institution, and the VP4 for increasing the CAR-T function is the ERST of Meldola. For the VP5 is linked to the safety improvement of the approach is Ospedale San Raffaele, the leading institution. For the technology transfer, IP and legal framework of the project is involved as leader the Istituto Oncologico Veneto. So going on the activity of each VP, for the VP1 are 2 major institutions involved. There are Ospedale Pediatrico Bambino Gesù and Fondazione Tettamanti with Professor Biondi. For the scientific coordination led by Ospedale Pediatrico Bambino Gesù, we have arranged the monthly meeting dates with the VP leader involved in this project, in which each VP leader updates about the activity of each of the units involved in the project, underlining if there is any issue to be solved to reach the milestone of this project. And also this is facilitating the scientific collaboration between the involved groups. The coordinator is also involved for managing of the communication between the project leader, the Italian Health Ministry and Patient Foundation, but also management of communication with company that have shown interest in collaboration with this project. For the scientific part of the VP1, one aim is the production and automatic manufacture product for CAR-T, trying to reduce the cost. And this has been done in OPBG using, as you can see here, an automatic closed system already performing 3 validation GNP run, in which we have shown very high purity of the product with a high level of transduction and high viability. Since these exciting results, OPBG, in particular Professor Franco Locatelli, has the intent to start at the end of 2020 a clinical trial for patients with acute lymphoblastic leukemia, DLBCL, and also PML. Also, another aim of this VP is the generation and allogenic CAR platform. OPBG is focused on natural killer cells genetically modified with the CAR, starting from peripheral blood of an healthy donor. In their process, since we have a full log of expansion of either NK or NK CAR, we have calculated to be able with 1 one leukapheresis to produce more than 500 doses for our patients, thanks to the use of bioreactors helping the manufacturing of this process. Also, Tettamanti Foundation is working on a similar project, trying to generate allogenic CAR, in this case CIK cells from cord blood. Also, Fondazione Tettamanti has very interesting data since they have already generated a process for the GMP expansion of CIK genetically modified with the CAR, reaching an high level of expansion and also of transduction. Going to the VP2, in this case, the VP leader is Ruggero De Maria in Gemelli Institution. This VP is characterized by very high density of activity. They are going from generation of new antibody targeting different kind of tumor model, from which they also generate single chain, and they perform a screening on tissue to verify specificity of a single chain or the antibody. Moreover, several other units in this VP are focused on the identification of the antigen targeted by a specific antibody, and also the generation and preliminary testing of new CARs. And this is especially linked to the high expertise of Gemelli in selecting new monoclonal antibody. As you can see here, this unit has generated more than 200 monoclonal antibody targeting glioblastoma and colorectal cancer. And this very high number of clones have been already started to be screened, as you can see here, also generating a single chain and try to identify the target antigen recognized by the specific antibody. IHEO has a very interesting tool to in vivo screen library of single chain in the model of PDX for acute myeloid leukemia, whereas Casa Sollievo Sofferenza has focused on a different disease and very high clinical needs of TLL with single cell analysis of PDX, trying to show novel target that can be used in this disease. L'Istituto Tumori of Milano is indeed focused on a specific antibody that they have already generated targeting LY antigen, generating a single chain that now in collaboration with the PBG they have used to generate a specific CAR. The activity are not yet done for VP2. Indeed, Fondazione Gemelli, they have already started to collaborate with Candiolo for the screening of the antibody specificity in tumor microarray with very interesting data on rectum cancer, colon rectal carcinoma, cervix carcinoma, and bladder carcinoma. Fondazione Gemelli has already generated CARs specific for, in particular, two different antibody with preclinical screening of the activity. Ospedale Pediatrico Bambino Gesù has generated a single chain for GD2, CD123, CD276, and in collaboration with the Takis company, also HER3 CAR to target sarcoma. For the VP3, the aim is the preclinical development of CAR killer cells against novel CAR targets, either for solid or hematologic disease. In this case, the leader is the Candiolo Institution with Dr. Enzo Medico. This is a relevant VP, which activity is impacting, I have to say, in all the other VP. And Candiolo has already launched a survey to understand the availability of patients-derived preclinical cancer model that can be cross-used in all the other VP. In particular, the surveys focus on 2D culture and 3D culture on colorectal cancer and glioblastoma developed by Gemelli. Patients derived a model for pediatric tumor in Ospedale Pediatrico Bambino Gesù, melanoma and pleural mesothelioma for the ears of Meldola, PDX, quindi animal model for breast, and AML generated respectively in Istituto Tumori Milano and Fondazione Tettamanti. EO is a very interesting tool to select ex vivo NK cells from haploidentical donor. All the CAR effector cells are covered by this VP, trying to characterize them. In particular, the majority of the unit involved in this VP are working with alpha beta T cells. 3 of them are also covering CIK, CAR cells, and NK92. Ospedale Pediatrico Bambino Gesù and EO are working for CAR NK primary cells, and OPBG also on gamma delta T cells. In vitro preclinical assays they are relevant to test the activity of novel CAR cells. In particular, the unit involved in VP3, they have developed the 2D and 3D cytotoxic assay, the relevant panel of cytokine for the detection of CAR T activity. They have been focused on proliferation assay for effector cells, but also assay to evaluate the migration and penetration of effector cells in tumor models. Ears of Meldola, they also have generated a panel to evaluate the effector cell signaling analysis using more than 500 probes for alpha beta T cells and CIK. The in vivo preclinical models are also relevant in this project, as you can see here, with a high focus on the unit they are involved, trying to cover the most relevant disease in the oncologic setting, in particular for brain cancer, for melanoma, all the colon cancer model. And in particular, I have to say that we thanks EAO for their work on the generation of MRI approach for the animal model using fluorine 19 reagent to track the effector cells in the animal model when we have to test their activity. The VP4 is to focus on the increase of activation, expansion, survival, tissue penetration, cytotoxicity, and then monitoring of CAR cells. In this case, the VP leader is Meldola, in particular Professor Giovanni Martinelli, and we have 7 different institutions working on this project. In particular, the ERST of Meldola as a leader activity here because they have developed a new vector to track CAR T cells, also able to optimize their proliferation. All of these tested in organotypic culture and 3D model system, also try to evaluate the tumor microenvironment in this setting. OPBG has focused the activity on a novel compound from FDA library drug, able to synergize with CAR, in particular testing the activity on novel CAR construct in 2D and 3D system. Ospedale San Raffaele has focused on optimize the effector cell used generated from T stem cell memory and NKT, also gene edited by CRISPR-Cas9 approach to reduce the immunosuppressive signals. Fondazione Tettamanti has already worked to generate CAR-CIK for AML and CLL, also in this case trying to target the tumor microenvironment represented by the leukemic niche. Regina Elena, thanks to the high expertise on Paola Nistico in the tumor microenvironment, they are also trying to develop a new approach for CAR T, reducing the exhaustion linked to the tumor microenvironment, trying to overcome the immunosuppressive status with the use of oncolytic virus. The CRO of Aviano is concentrating the activity on the tumor penetration, inducing the activation on metalloproteases in the setting of myeloma and ovarian cancer. Last but not least, Reto Amore is working on tumor microenvironment in the context of lymphoma and melanoma. The VP5, which leader is Ospedale San Raffaele, in particular Monica Gasucci, is focused on the safety of CAR T cells. In this case, we have 5 different institutions working with Ospedale San Raffaele, generating a novel animal model to test in a humanized mice the safety of novel CAR T. In particular, underlying the role of the glycosylating agents in helping CAR T to target the tumor. OPBG has developed a new suicide gene, now validated in vitro and in vivo, also drug library analysis to evaluate which drug is synergizing with the CAR T, reducing the monocyte activation linked to cytokine release syndrome. Also in the VP5, several units are involved in patients' immunomonitoring after the infusion of CAR T commercial product, but also the academic drug product in Ospedale Pediatrico Bambino Gesù, evaluating different soluble cytokine associated with inflammation, analyzing specific pathway and analytes, and also evaluating the role of the myeloid cells in this system. The last VP is the legal aspect, the intellectual property, in which leader is Istituto Oncologico Veneto with Dr. Lucchetta. The role is relevant indeed. They have led the IP advisory board, and they already have defined the enlarged IP advisory board, as well as the intellectual property committee, in which each unit have one delegate. One aspect that has been already carried out as activity by the WP6 is the support of the scientific coordinator in WP1 to collect the expression of interest about the company on this project. They also support all the unit in this project to exchange material and know-how, all in the regulation of the IP bodies. All of us have already signed, thanks to the activity of the WP6, the consortium agreement. So the take-home message for our project is to underline that this is the first attempt to create a national network for the development of innovative ATMP, and I hope that in my presentation I have shown how we are able to build a network on different ERCs with different expertise, and now all together are working to reach the milestone of this very difficult project. All of these with the ministerial endorsement involving regulatory bodies and the clinical transfer expected in the time frame of the project. We are currently defining the involvement of several companies that show high interest in the collaboration of this project that we may realize only because there is a lot of focus by people in Ministero della Salute, in particular I have to mention Dr. Leonardi and Dr. Scalera for their support on this project. Of course, the President Ruggero De Maria of Alleanza Contro il Cancro and the scientific coordinator Franco Locatelli that is hardly working to put all the other institutions working together. Thank you.
Thank you very much, Concetta. Here you gave us the impression of a massive amount of work, a very, very complex network and a lot of scientists involved. So we have time for a couple of questions. We start, Concetta. If I understand, Concetta, though, I mean, the projects are really across different stages of development with a lot of early projects. There are one or two projects which are pretty close to the clinical application. I mean, I would assume that you will run those clinical trials within the available and existing academic facilities. But my question is, are you discussing more in general what is the approach of our network? What should be the approach of our network with respect to the capacity in the near future to run this clinical trial in Italy? So academic, industrial, both. Are you discussing this issue, which is down the road pretty close, I would say?
Thank you very much, Giuseppe, for these questions. Actually, there is a lot of discussion about, although the aims of clinical translation is not one of the milestones of the project, so the clinical translation is behind the focus of the project. Of course, all of us are working to get the product in the clinic. As you so some of the projects are very close to the clinic and they're going to be academic trial. But we already have started with Ruggero and Dr. Leonardi to reason about collaboration with company. We have interest on several companies to start to see more in detail the activity of our project, and we really hope to engage some of them for the clinical translation.
That's very important. So you are going toward a mixed public and industrial system, which I guess is the right way.
It's the only way to go forward.
Yeah. Thank you very much, Concetta.
Thank you very much.
There are other very quick questions from the chair. I don't see questions from the outside for the time being.
No, I had the same question about involvement of companies, but you have anticipated me.
Fine. So, Concetta, thanks a lot, and congratulations for the work that you have presented. So I leave the duty of the coordination to Gennaro. Please, Gennaro.
Yes, thank you very much. So the next speaker is Luca Mazzarella from IEO. He is going to talk about an important project that has been financed through an application to a European Horizon 2020 program on public procurement. The project is called ONC-NGS and is on liquid biopsy. Please, Luca.
Hi, Gennaro, and thanks everyone. I'm just, can I ask you for a confirmation that you can hear me and you can see the presentation?
Yes.
Fantastic, thanks a lot. So I will very quickly introduce you this very interesting Horizon 2020 framework. So the project deals with the establishment of a tool for liquid biopsy, and we in the planning of the project, we realized that there definitely is a unmet need in current liquid biopsy tools, because basically there are two models that have emerged over the last couple of years. One model, which is typical of large companies that sell this as a service, that provide very large scale, high throughput panels with a centralized system. So basically you have to send them your sample and they will provide you with a report. And then there is another system that is typical instead of companies that provide reagents or tools for everyday diagnostics, who have developed a number of dedicated tools for either dedicated specific mutations or small panels. And the pros and cons balance between these two models is exactly one the mirror image of the other, if you want. So the pros of the large scale and centralized model is that it's very informative. It allows also the definition of complex signatures, not only of specific mutations, for instance, the tumor molecular burden, and you can use the same panel for multiple tumors. The cons, however, is that it's significantly more expensive than smaller panels. The turnaround time is often relatively long, and the control over data and samples is minimal, because you don't really have access to the raw data, for instance, and the sample is shipped and then can be returned, but for sure you don't have a control over the workflow at all. Instead, for the small scale panels, those that you can run in your pathology lab, the pros are exactly the cons of the large scale. So it's cheaper, the turnaround time is often very quick, and you have maximal control over data and over samples. And the cons are exactly the mirror image of the pros of the large scale. So it's fully informative, because usually these are small panels. You have problems in identifying complex signatures such as TMB, and they are often dedicated for individual tumors, so you have to run multiple panels for different tumors. And one common issue, just like in every field when you have a proliferation of technologies, is that there is a lack of standardization. So you may have the same patient that gets analyzed through one platform or the other. The limits of detection sensitivity are very different, and the results are not homogeneous. So we wondered if we could have it all, if we could join the pros of the large-scale systems, therefore have highly informative panels which would allow complex signature determinations and across multiple tumors, together with the pros of the localized systems, where you have a sustainable cost, the turnaround time is limited, and you have maximal control over data and samples. And of course, during the process, achieve some form of standardization that would allow the same standard to be delivered across different centers, and possibly this being a European project across countries. So this project was launched, the acronym is OncoNGS, and the important thing to stress here is that this is a pre-commercial procurement grant. It's not your standard scientific grant, and I will get back to that in a moment. So this is what we obtained. The scope of the call was to develop integrated solution for testing, analyzing, reporting, and storage of next-generation sequencing medical data within routine healthcare diagnostics. This was the general call. The budget is substantial, so it's around $12 million euros over a period of 5 years. And we have, and can I highlight this, a website that was recently launched that I invite everyone to have a look at. The challenge that we put forward is to provide efficient molecular DNA and RNA profiling of tumor-derived material in liquid biopsies by means of a pan-cancer tumor, a series of analysis on pan-cancer tumor markers, and integrated with an ICT decision support system. Multiple institutions from multiple countries have adhered, and I will show this in a moment. But first, I wanted to try to explain to you, although this is a difficult task indeed, what the grant is about. So this is, as I said, not your usual scientific project. This is a pre-commercial procurement. This is an idea that was launched in the European Union a few years ago. And how does it work? It's aimed at fostering competitive development. So basically, to create a framework where, in a controlled way, multiple private companies compete for developing prototypes at the pre-commercial phase. So they enter in competition before they actually enter onto the market. So the way it works is that you create consortia of public procurers that buy R&D from several competing suppliers in parallel to compare alternative solution approaches and identify the best value for money solutions that the market can deliver to address their needs. And the important thing is that this is a structured process. So it's split into phases, a first phase of solution design, prototyping, then original development, and a validation testing in a specific setting of samples in this case. This is the way the project is structured. We are now in the so-called phase 0, where we are basically defining the criteria for the prototypes of the liquid biopsy tools to be delivered. And the aim of this will be the issue of a tender due for the second half of 2021. Then we will have a triple phase of validation that will first undergo the solution design. So the prototype will be evaluated purely on the basis of its design. Then a analytical testing phase where a number of standardized samples will be provided in order to evaluate performance on the road, if you want. And then a final phase of pre-commercial testing on clinical samples. And this is the consortium composition, and the lead proponent of the grant and also the lead procurer in the pre-commercial procurement jargon is Scienzano, which is a national institution in Belgium, and other institutions, including us, as you can see, participate to the consortium, which includes not only medical institutions, but also, this being a legal procurement grant, a number of administrative issues which provide substantial support in terms of legal, financial, and ethical issues. This is the state of things as of October 2020. We are in the middle of the phase 0, as I said, where we are basically issuing a number of technical specifications. We have been discussing key markers. I will tell you about it in a few slides. And we are basically going through all the, if you want, boring part of issuing GDPR compliance documents and administrative documents for the tender issue. So one important issue is, how do you evaluate a liquid biopsy tool? It's not a trivial process. And one idea that emerged in discussions within the consortium is to use a so-called modular architecture coupled with a tiering system. How does this work? So when you have to score liquid performance of a liquid biopsy tool, you have to ask yourself a question: what samples will you use for this evaluation, and how exactly are you going to score it? This is even more important, since what we're dealing with is not our own research labs where we can play with the scoring criteria until we are satisfied, but we have to issue a tender here. So these criteria need to be objective and need to be transparent, need to be open, and need to be sufficiently well structured that the company is able to tell whether they are going to be able to meet this criteria or not. And so what we have proposed is to use modules. This is based on the idea that the realization that not all tumors are equal in terms of shedding cfDNA, circulating free DNA. So different tumors shed different amounts of circulating free DNA, different stages. So for instance, late metastatic sheds different amounts of circulating free DNA compared to, for instance, an early cancer that has just been operated. And the clinical significance also is different according to tumor type, to the stage, and to the clinical setting. So a module for the evaluation is a combination of a specific genetic alteration to be tested, specific disease, and a specific clinical setting. Just to provide you an example, if, let's say, we want to test the sensitivity of our liquid biopsy tool to identify mutations in PI3 kinase, which have recently become popular as companion diagnostics for specific PI3 kinase inhibitors, it would probably make little sense to test it onto random samples, for instance, samples from a metastatic lung, not because PI3 kinase is never going to be of any use in the metastatic lung setting, but simply because at the moment there is no drug that has any immediate sign of being approved for a PI3 kinase mutated lung, whereas the state of advancement of things in the breast cancer is much more advanced. So it makes sense to test it in the metastatic breast setting. It makes little sense to test it in the metastatic lung setting or in the early breast cancer setting, for instance. So this would be a good module. So samples from metastatic breast cancer would be good samples to test these mutations, whereas these other samples would not necessarily be that. And also, not all mutations are equally important to be identified in a test. And that's why we propose to introduce a tiering system, which is a good way to create a composite score that can be objectively issued to the companies and can undergo different phases of evaluation. How does this work? So the first tier is composed of core modules. So these are genetic alterations of established clinical utility, which are already standard of care, and for which sensitivity below a defined threshold is considered unacceptable. So these will be used to negatively select with a binary decision. So the tool may provide a fantastic sensitivity on 500 other genes, but if it fails to detect with a decent limit of detection, for instance, the T790M mutation in EGFR, then is obviously no use. And this will be used in the in the second phase of the evaluation procedure to negatively select inadequate tools and to define the limit of detection. Then there are the other three modules which provide also these types of parameters, but have importance only in the last phase. This is, of course, to make the whole testing procedure sufficiently rapid, sufficiently efficient for a commercial procurement process. So tier 2 are genetic alterations of established clinical utility, for instance, taken from established sources such as the SCAT or OncoKB, and will be tested using sensitivity and specificity across clinical samples. For instance, PI3 kinase in metastatic breast or the TMB in lung cancer. Non-essential modules, so they are maybe the non-essential tag is a bit of a misnomer, but these are still genetic alterations of likely clinical utility for which a minimal sensitivity threshold is not mandatory, but will contribute to creating the score. For instance, ESR1 in metastatic breast, which has an established role, but is not at the same level of clinical usefulness as, for instance, PI3 kinase. And lastly, a number of additional genes. I didn't provide examples because here you can basically name your favorite driver gene that you think would be useful that are of potential interest that will be included in the panel, but you will not collect samples that are specifically dedicated for testing those mutations. You will only test the coverage across that region in the tool. And finally, a number of additional parameters which are not specific for specific genes will be used to also score the financial and logistical issues. So these are the upcoming NGS steps. We are, as I said, undergoing through these administrative process at the moment. And I can basically skip this because there is only one specific slide that I think is relevant for us today. We, as I said, we have to ask ourselves how ACC as a consortium can benefit from and contribute to OncoNGS. As I said at the beginning, ACC is part of the BIAS consortium. So we are in the position where we can determine the conditions of the tender, and we will have early access to the prototypes. But then we can have two levels of engagement with this project, I think. One, I would call passive engagement. So we limit our involvement in the participation in the administrative process. We define the tender, and we take care of all the regulatory aspects. We will test the somehow, but in a way that we are unable to predict now because the project lasts for several years, the prototype. But then we can also have an active engagement. And I think this is actually a better way to contribute to the project and probably to benefit from it. Because what we could do is to actually systematically collect samples in the upcoming years, and we could definitely use and take advantage of the already ongoing trials such as the ACC Lung and the GERSOM trials to attach to these trials an observational part where that we will have to do is to add the collection of a few extra tubes for plasma storage that we will use for our own scientific purposes, of course, but that will also contribute to the standardization effort of the prototypes in the years to come. And the important thing is that most likely we will also be able to have some funding, although it's not going to be a major sum, that can contribute to the collection of these samples. Because of course, as in any other effort like this, the metadata and the possibility to attach and pre-validate these samples is going to be key for the outcome of the project. With this, I have finished. I would like to especially thank Patrizio Adeire and Roberta Yeo, who have provided a major help in this project. Thanks a lot.
Thank you very much, Luca. This is a very important project for our network. I don't see questions coming from the audience. I can start asking you a question. So the final purpose of can you clarify if it's going to be the generation of a universal liquid biopsy assay, or if you plan to develop disease-specific liquid biopsy panels?
So the idea is definitely to provide a tool that is as flexible as possible and as comprehensive as possible. Now, of course, the important thing to consider is that part of the decision making is by design left to the companies. So if the we will set the criteria for the evaluation of the prototypes, but then if the company thinks that these criteria are best met by multiple panels and not by one single comprehensive panel, then it's up to them. It will be part of the evaluation process.
Okay, a second question that I have is obviously thinking at the future and the future applications of liquid biopsy. One may imagine that liquid biopsy will be very important in identifying, in early diagnosis of cancer. So are you also thinking in that direction?
Yes, this has been at the center of some of the discussions, of course. However, that we have to very clearly define the scope of the tender. And at the moment, the scope of the tender does not include early diagnosis. Of course, this does not rule out that the same tool can be used for early diagnosis. But since, again, this is not a canonical scientific project, what we need to do is simply to provide the criteria for the tender. So, of course, if you have a comprehensive panel that has a very high limit of detection, is not expensive, can be used in multiple sites, then of course it will be absolutely of great interest to also test it in the early diagnostic setting.
Okay, thanks. Are there any questions from the co-chairs? Ruggero, Pier Giuseppe, comments?
Gennaro, thank you very much. I think we should proceed. We are a little bit late.
Okay, so thank you, Luca. And we can go to the next speaker, who is Giovanni Tonon from San Raffaele Hospital. He's going to talk about this very ambitious European project called 1 Million Genomes. Giovanni, are you there?
So good afternoon, everyone, and thank you very much for inviting me to present one initiative. It's the 1 Million Genome Project. Let me introduce you briefly to what it is. It has been declared, a declaration of cooperation has been established in April 2018. In the beginning, there were fewer countries. Now, at this point, we have 22 countries that have now signed, with the 6 that are still observers. In terms of working groups within the initiative, there's been identified 6 working groups, and then 5 use cases. The working groups are divided as follows. We have a working group 1, that is basically the management one, the stakeholders engagement. The second is ethical, legal, and social issues. And since this is a very hot topic, we're going to go back to this one later on. As well as in the working group 3, that is standards and quality guidelines. Also, this one is a very important aspect of the initiative. Technical infrastructure, I'm going to touch base on this aspect as well with a few slides. The working group 5 is personalized medicine and NIPTA. The working group 6 is coordination office. And then we do have use cases on which we have involvement of private sector, rare diseases. And the work group 9 is cancer, on which Ruggero De Maria and Thanos, together with myself, are the leading persons in the initiative. Also, common and complex diseases are included. And finally, recently included in the list is the working group 11, that is dealing with infectious diseases and COVID-19. As for the goal and the objective of this, the goal is establishing a European learning framework. So kind of a framework will include all the genomic data and also phenotypic data for a large population. As you can see in this initiative, many different ongoing activities has been interacting and included. Here is a kind of a quick list. I'm not going to deal into this one. I would prefer to go more into other details. But basically, the objectives of this initiative is to engage at really several levels in the EU basically both local, regional, national, and also European, to define cross-border access to genomics and personal medicine data. And as you will see, this is really kind of a daunting effort, given the heterogeneity between different countries, but also within countries. And so we get to the second aspect, that is the translation requirements for data quality, standards, infrastructure, and also legal and social issues, in a way to really be able to provide a technical specification and definitions and guidelines at the end, but we'll be able to really capture European best practice. And finally, the final goal and objective of this initiative is to really drive adoption and support long-term operation of this framework, in a way to really provide to European countries a way to generate, but also in the proper way, but also integrate and share personalized and genomic information across countries. There are three key aspects that I would like to raise. The first one is that we are focusing on use cases, so basically diseases and conditions that seems to be really the most relevant and important of this regard. And here we have rare diseases, cancer, and common and complex diseases. One of the aspects that is happening in this context is that the highly difference in approaches that are present, the impact that is promised to be really very important towards the national healthcare systems, and the integration, this is also a kind of a sore point in many ways, with the ongoing major European initiatives to build on. The second aspect has been chosen early on, is the idea of building a federated infrastructure. This is very important, but the federated approach has been embraced as the only way to really be able to define and to bring to success this initiative. And one of the aspects is the focus on high-quality whole genome sequencing, and with a less emphasis on whole exome sequencing. Talking about cancer, as up to now, most of the effort has been directed towards panels. This initiative of 1 million plasma genomes in Europe is really focusing more on broadly, as it will happen and is happening already in some countries more and more, also for cancer exome, but most of all whole genome sequencing. And so for that reason, as you will see in the last slides, the number of patient cases has been accrued in the cancer setting is somehow less prominent than in other conditions, like, for example, rare diseases, where whole genome sequencing has been, or exome sequencing as well, has been more widespread used in the past when compared to cancer. A second aspect of this key topic is the minimal set of associated phenotypic data. This is another area of active implementation. And finally, the distributed analysis that requires harmonization of data. Finally, provided that in the different countries do exist various ethical and legal frameworks, the initiative and the effort to really provide some flexibility and some ways of allowing the sharing of this very privacy-sensitive genomics and health data, it's really imperative and mandatory. The roadmap in terms of timeline, in a way, we just started, despite the fact that the agreement has been signed in 2018. We can say that really the kickoff has been happening in 2020 with the third major phases. One is the engagement in the governance, cooperation, and collaboration. The second one in the translation of this establishment of infrastructure, guidance, and pilots. And finally, in 2022, the idea will be to really implement the sharing, the scaling, and the sustainability of the whole project. And this time here, this is the somehow a satellite initiative that is beyond 1 million project genome. This is an EU grant with the 4 million granted for the 3 years, that really has the goal and the objective to put in place to implement this initiative. It started in June 1st and will end in May 2023. And these are some of the partners that have been engaged in the consortium. Again, I don't have time to go into detail in each of them, but as you can see, it's kind of a widespread EU level initiative. So let me touch base now on some of the working groups that I think are most relevant and also more important in this regard. The first one is definitely the ethical, legal, and social issues. This is actually fairly important because there are very, very roadblocks and challenges to be overcome. And the major aspect here, what is legally and ethically required for secondary use of genomic and health data in a joint pan-European infrastructure. And so Regina Becker, that is leading this effort, is really looking into all the different aspects and phases of the project that goes from the starting, of course, but also inclusion of data subjects, very private and personal matters, data access, data use, results and their manipulation and their analysis, and finally data retention. Each of these phases, of course, poses different challenges and issues that need to be addressed. Some of these include certainly justify the trust of the citizens, find solutions to overcome national differences in legislation and ethical practice that are really quite profound and variable across different situations. Some of them are also has to do with the solving of GDPR challenges that are quite important in this phase. Create an infrastructure that is able to really appear homogeneous despite data contribution from all over Europe. And finally, agree on policy framework and the legal basis for the initiative. As another important aspect, since in this effort we are basically combining already available data in this phase and not generating new ones because the time frame is so short and so close, is to provide standards and quality guidelines that are really good for all the sequencing that is collected. As I mentioned before, and I stressed, this is really a federated effort. So the objective of this working group is going to be drive the development of a European infrastructure to secure cross-border access to all this data and to translate all these requirements of data quality and standards throughout Europe. And here, this is actually quite a daunting effort, as there are really no standards for sequencing NGS, not only from a technical and quality aspect, but also basically by the nomenclature of the quality metrics. So building this aspect is going to be really, really very challenging. There are some very technical details, like, for example, sequencing of germline tumor DNA are both experimentally but informatically very profoundly different. And so it's very difficult to put them together. And unlike a clinical test on which common standards are really set and established, and SOPs, or standard operating procedures, this is really not the same for next generation sequencing of human genomes or even exomes, on which a common procedures and time frame is really not available. And finally, also the collection and the description of phenotypic and clinical information is very, very heterogeneous, with really not a uniform way of naming them and also of standardizing them. So as I mentioned before, working group 3 is really one of the most challenging across the board. And this is actually some of the guidelines, this is the quarters of EvoGAT, of the publication and definition first, because of these guidelines for cancer. As you can see, it's really kind of a daunting effort. And some of the major challenges on this aspect that has to do with primary tissue acquisition, the definition of cancer somatic mutation with the ratio between tumor and normal, the choice and the definition of the tools, Illumina, Oxford Nanopore, for example, and also, of course, the inclusion of population, like in the case of rare diseases with the germline, with the trios. And let me actually show you one of the first efforts towards this initiative made by EvoGAT within this working group. This is a paper that just recently came out in Nature Communications, defining the framework for quality assessment of whole genome cancer sequences. And as you can see, 5 parameters has been defined, and just providing at the end a kind of a star-based definition of quality that a whole genome sequence needs to meet to be included in the dataset. This is really a very important aspect because, as I mentioned before, in this case, we're really dealing with data that are already available. So we really need to somehow define which are the minimal common quality assurance that we need to implement. And the 5 QC parameters include mean coverage, evenness of coverage, somatic mutation calling, pair width mapping to different chromosomes that needs to be, of course, quite homogeneous, and ratio of difference in edits between pair grids. As you can see, this is actually quite important and daunting, and from this regard, within the, for example, the BCA-WG genome initiative, as you can see that across different tumors, the quality has been quite heterogeneous. So it's not that all the samples and studies have achieved the 5 stars they would hope, but in some cases, only a minority of them. Let me briefly touch base on the technical infrastructure. This is work group 4, another one very important, in the sense that having access to all this data provides some key challenges as well. It's been decided to somehow implement and introduce all the data into the European Genome Phenome Archive with a user authentication access process, and of course, a series of ELC compliance patterns that need to be met to have the data implemented in this group. At this stage, while the data collection and metadata has been approved to be within the federated EGA, the data analysis standardization is still ongoing and it needs to be decided as yet, and also the data quality, data product query interfaces. So we are really still in the phase here on which some of the aspects that I'm mentioning here are really not as well defined. So if you look at the different interoperability, you have a legal, organizational, the semantic, and the technical. And for some of the, for example, the organizational interoperability, the European Genome Phenome Archive is going to be the final repository. For the technical interoperability would be Elixir. But for others, for example, the semantic, this is actually a fairly complicated one. This is still ongoing, still a work to be completed. Let me close now with just one slide actually for the cancer. This is really a work in progress, so we don't really have much data to show at this point in the sense that we're still collecting them. I'm just showing this in this cartoon. Some of the countries have so far participated. Of course, the intensity of the color relates to the number of samples provided. The major message here is that groups in Europe that do have whole genome sequencing or whole exome sequencing available data on cancer patients that will be willing to participate, please get in touch because it will be very cost advisable to include them into this pan-European initiative. I've excluded here the data from the UK in the sense that the UK is really not yet clear whether it will be included in this initiative or not at this stage. But the data are basically collected and in many ways are finalizing. So let me finally conclude. Thank you for your attention, and I'm open for any question.
Okay, thank you very much, Giovanni, for the very clear presentation. And I don't see questions coming from the ground, but I will start asking you one. Which is a critical question. So as you pointed out, whole genome sequencing is very rarely done for cancer patients. So there is a sort of, I see an issue here between the declaration of the entire project and program and the financial support to it and the organization behind, especially for cancer. So how do you think we may solve this issue in the future?
Yes, Gennaro, this is definitely a very, very challenging point that you are raising. Even because, as I was mentioning, cancer is different from other diseases that has long been used to utilize whole genome sequencing for other reasons, or at least whole exome sequencing. We're really pushing this topic with the EU organization. And actually, in the afternoon today, we're going to have a meeting just to really try to foster some resources and additional help to really try to bring the cancer field and in par with the other ones. I would also say in parallel that we are also engaging other organizations and other initiatives. I'm thinking now by the Argo initiative, for example, to try really to corral data and whole genome sequencing data, whole exome sequencing data, to really try to improve them. So even pretty much research-oriented initiatives are going to be involved in this effort. Otherwise, we will not make it by the 2022 threshold.
Okay, thank you. Are there other questions or comments? No questions from the ground?
No, Giovanni, so sorry. In your opinion, when it's going to be, when the whole genome sequence will become a standard procedure in cancer?
Yes, this is a very good point as well, Ruggero. So in some countries, I'm thinking about Germany, for example, in many centers, whole genome sequencing, it's already a routine practice. In other countries, instead, there's nothing whatsoever, or there are still very, very limited panels. So there's a huge heterogeneity throughout Europe on this approach. If you take the UK, for example, this is another country that has embraced whole genome sequencing in many cases. So it's really kind of a very heterogeneous pattern throughout Europe. The tendency is to go through or towards whole genome sequencing. It's already happening in some countries. It will take some time until the other country will be able to match that.
Thank you, Giovanni. Thank you.
Gennaro, may I ask a question, brief question? Giovanni, I mean, we are all looking forward, and the whole genome is going to be probably in the future relevant also for cancer diagnostics. However, I mean, I have the impression that if we consider the needs of coverage for having whole genome sequencing becoming relevant for cancer in terms also of sensitivity, I have the impression that whole exome sequencing is ready now. Whole genome sequence is still a little bit of a problem because the cost doesn't allow to go to the coverage that we might need to cover the sensitivity that is needed in diagnostics. What do you think?
Yes, I agree with you, and actually we struck a balance with them because throughout the discussion, the idea was also to remove whole exome sequencing altogether and just to focus on whole genome sequencing. We basically raised the same issue that you're raising here, saying that, okay, we may forget panels for the present, for the near future, but at least whole exome sequencing, to us, it looks like a nice balance between costs, but also sequencing depth, as you correctly pointed out.
Thank you very much, Giovanni.
You're welcome.
Okay, grazie, Giovanni. Thank you very much.
Thank you.
We can move on. Ruggero?
Yes, Gennaro, and it's my pleasure to introduce you as a next speaker. And so Gennaro is going to talk about a project which is the first Italian whole exome sequencing screening.
Ruggero, this has been, so the program has been changed, as you probably remember.
Yes.
And that project has been incorporated into Vanessa's presentation. So we will move on directly to the next.
To Sandro. I'm sorry, because I guess I have an old program. So Sandro Pegnata is the next speaker. Sandro Pegnata is the director of okay. So we can probably move to Nello Martini.
You made it, Ruggero. Nello.
Nello Martini is talking about the ACC ION project on mutation oncology. Nello has been pushing a lot to have the availability of drugs, repurposing drugs for advanced cancer patients.
Allora, ringrazio Alleanza contro il cancro, Ruggero De Maria e Pier Giuseppe Pellicci per questa opportunità di presentare al congresso ACC il documento di consenso che peraltro in questi giorni abbiamo presentato approfittando anche dell'intervento del vice ministro Sileri e del direttore di AIFA Magrini, e quindi cercherò di fare un po' il punto sul progetto dell'oncologia mutazionale in Italia, toccando 4 punti. La premessa attraverso cui vorrei precisare gli obiettivi strategici dello sviluppo e dell'organizzazione dell'oncologia mutazionale in Italia, così come abbiamo condiviso con Alleanza contro il Cancro e quindi con la rete degli IRCS oncologici. I punti chiave del documento di consenso a cui Alleanza contro il Cancro ha dato, diciamo così, un apporto sostanziale dal punto di vista del contenuto e anche della condivisione, e alcune osservazioni finali di prospettiva e anche dei prossimi step per lo sviluppo dell'oncologia mutazionale in Italia. Questa slide è semplicemente per ricordare, era l'ottobre del 2017, il 12 ottobre esattamente, quando veniva pubblicato su New England questo perspective di Lemery, numero 2 dell'FDA, che significava l'inizio attraverso l'FDA della oncologia mutazionale, con l'autorizzazione del pembrolizumab nella iperattività satellitare e quindi di fatto l'inizio dell'oncologia mutazionale. È noto, e certamente non compete poi a me che non sono un oncologo, ma il punto chiave del cambio di paradigma è che nel modello tradizionale si muove, diciamo, dalla localizzazione del tumore, dall'istologia, dallo staging, per arrivare poi ai farmaci e all'indicazione by cancer site, e nel modello mutazionale si parte dalla mutazione, il driver è la mutazione, per arrivare ai farmaci druggable e quindi alla indicazione cosiddetto regardless cancer site, age and sex. Sono 2 modelli che non sono alternativi, che devono essere tra di loro integrati nell'ambito del percorso assistenziale e della condivisione del modello organizzativo. Il documento di consenso a cui facevo riferimento, riconoscendo il contributo sostanziale di Alleanza contro il Cancro, in collaborazione con la Fondazione Ricerca e Salute, la vedete qui rappresentata con tutti gli autori e soprattutto con tutte le associazioni che lo hanno sottoscritto. Alleanza contro il Cancro, l'AIOM, Periplo, il CIPOMO, SIAPEC, Cittadinanza Attiva, FAVO, la Fondazione per la Medicina Personalizzata e appunto la Fondazione RES. Volevo dare alcuni elementi, e cioè che il documento non è un punto di arrivo, è, come dire, uno step di un processo di approfondimento e di implementazione della oncologia mutazionale in Italia, ma vorrei richiamare l'enorme rilevanza di avere un documento che tutto il mondo dell'oncologia e dell'anatomia patologica, rappresentanza dei cittadini e rappresentanza dei pazienti, hanno sottoscritto. Questo è di grande rilievo perché ovviamente riflettere sul modello organizzativo e sulla governance in advance, peraltro, significa, come dire, costruire le condizioni per l'accesso, per il percorso assistenziale, la sostenibilità economica, evitando di essere travolti, diciamo, da un sistema non controllato di profilazioni genomiche, di farmaci off-label, di rischi di un conflitto tra l'oncologia e i pazienti. Questi sono i 10 punti in cui è strutturato il documento di consenso. Per quanto riguarda il Molecular Tumor Board, ripeto, si tratta di un processo complesso perché deve essere previsto un modello organizzativo condiviso con la individuazione dei team multi-interdisciplinari, appunto i Molecular Tumor Board, nell'ambito delle reti oncologiche regionali, quindi non strutture separate, ma strutture ovviamente integrate nell'ambito delle reti oncologiche. Poi lo vedremo, la necessità della profilazione genomica e quindi dell'inserimento NGS nei LEA, tariffe regionali, l'implementazione della piattaforma genomica, cioè un sistema complesso, e l'innovatività del documento è proprio di ragionare sull'intero processo organizzativo del percorso assistenziale del PDTA e non insistere su una singola variabile. Le indicazioni, non entro nel merito, sono delle indicazioni, diciamo, di carattere generale e soprattutto flessibili, tenendo conto che le conoscenze stanno rapidamente mutando in questo ambito e quindi le indicazioni dovranno essere, come dire, aggiustate e aggiornate sulla base dell'evoluzione delle conoscenze. La piattaforma genomica, questo è stato forse il punto di maggiore convergenza che è stato immediatamente trovato, e cioè sulla necessità che il Molecular Tumor Board, le reti oncologiche, gli IRCS, pur mantenendo ovviamente, ci mancherebbe, la titolarietà dei propri dati, ma possano progressivamente alimentare in maniera interoperabile, attraverso un processo di interoperabilità, una piattaforma oncologica nazionale che abbia le caratteristiche che sono richiamate in questa slide, ripeto, rispetto anche alla sicurezza, alla privacy e al GDPR. Per quanto riguarda il Comprehensive Genomic Profile e l'NGS, abbiamo vari problemi che devono essere affrontati in modo strutturato, e cioè la individuazione da parte delle regioni dei centri presso cui eseguire con qualità, sensitività e sensibilità questi test e riproducibilità sulla base di criteri di carattere nazionale, rimanendo alle regioni la responsabilità organizzativa locale. Dobbiamo garantire un accesso uniforme su tutto il territorio nazionale e quindi definire la rimborsabilità nell'ambito dei livelli essenziali di assistenza. È stata istituita la commissione per l'aggiornamento dei LEA e come passaggio intermedio poi la definizione delle tariffe delle singole regioni. Sappiamo che oggi c'è una situazione frammentata, alcune regioni, tra cui la Lombardia, hanno già previsto per alcuni test la rimborsabilità. Bisogna evitare che le regioni vadano avanti in maniera disordinata, con un sistema di Molecular Tumor Board, di compiti, di attività, di centri regionali, di piattaforme completamente, come dire, diverse e frammentate. Questo sarebbe il fallimento dell'oncologia mutazionale e ci sarebbe un grande rischio, ma soprattutto verrebbe tradito il principio di interesse del paziente e l'equità di cura. Quindi questo è anche il fondamento del documento di consenso, di aver fatto, come dire, un consenso e quindi un accordo prima del processo di cambiamento, per governare il processo di cambiamento e per assicurare l'equità di sistema. Abbiamo anche cercato di definire, ma non tanto come diciamo un budget impact definito e completo. L'impatto al primo anno sia dei farmaci off-label sia della profilazione genomica. In questa slide sono riportati alcuni dati, ma quello che interessa qui non è tanto il valore in sé. Sicuramente si tratta di dati che hanno necessità di ulteriori conferme anche sul piano, diciamo, della metodologia, però la cosa importante è di avere l'idea che è rappresentata in questa slide, che non si tratta di un impatto economico che mette in crisi la sostenibilità dell'intero sistema. Teniamo conto che attraverso la legge, il decreto legge Cura Italia, il decreto legge rilancio e ancora la finanziaria dello scorso anno, complessivamente il Fondo Sanitario Nazionale è stato aumentato di 4,7 miliardi di euro, il che significa un rifinanziamento, ad esempio, della farmaceutica di oltre 770 milioni di euro. Quindi abbiamo anche un'occasione unica e irripetibile per ammodernare il Servizio Sanitario Nazionale e all'interno dell'ammodernamento del Servizio Sanitario Nazionale di poter, come dire, organizzare e rilanciare l'oncologia mutazionale in Italia. Vengo ad alcune valutazioni di carattere conclusivo, e cioè cercando di raffigurare il modello attuale, quello istologico, che dal punto di vista dell'accesso e dal punto di vista regolatorio vede una serie di centri che sono stati individuati dalle regioni per la prescrizione di nuovi farmaci oncologici. Oltre 300 centri sono in rete, sono collegati con AIFA attraverso i registri, con la scheda di arruolamento, la scheda di follow-up e l'applicazione dei cosiddetti Managed Entry Agreements. Purtroppo questo schema, che è nato nel marzo del 2006, lungo gli anni ha un po' tradito gli obiettivi originali, soprattutto in termini di restituzione delle informazioni ai dipartimenti di oncologia, alle reti oncologiche, alle regioni, e quello che era un reale progetto di innovazione e di ricerca ha finito per diventare in larga misura un burden burocratico amministrativo. Io penso che vanno invece rilanciati in senso moderno, strategico, i registri, e quindi sulla destra in azzurro, in verde, quindi in verde speranza, vedete il modello mutazionale con la previsione di 20, 30, spero non di più, molecular tumor board allocati all'interno delle reti oncologiche regionali, collegati con AIFA e aventi, diciamo, come base la costruzione della piattaforma genomica, imputando quindi all'interno della piattaforma i risultati della profilazione, ma anche i dati del paziente, ovviamente i dati di laboratorio, in modo da creare un database che dovrà essere rilevantissimo per la produzione di nuove conoscenze, di attività di ricerca, quelle che con una dizione che vorrei riprendere, Pellicci aveva definito durante le riunioni di stesura di questo documento di consenso Real World Clinical Trials, cioè un concetto di ricerca che nasca e si alimenti nella normale pratica clinica. E di conseguenza quindi sono due modelli integrati fra di loro, all'interno dei processi assistenziali e dei PDTA, e quindi al modello tradizionale che certamente rimane e però si integra all'interno del processo assistenziale di trattamento dei tumori con il mutazionale, quindi i centri attualmente esistenti e in rete con i managed entry agreements, con i 20, 30, vedremo quali saranno, molecular tumor board che vengono come dire, nominati e allocati all'interno delle reti oncologiche e che vengono sostenuti dall'alimentazione della piattaforma genomica nazionale. Queste, diciamo, sono le prospettive e volevo chiudere con una connotazione di come il modello dell'oncologia mutazionale in qualche misura induce anche una riflessione sul cambiamento delle modalità di ricerca. Nel modello istituzionale classico abbiamo l'autorizzazione EMA, l'attività di ricerca che viene largamente, come dire, sponsorizzata da parte delle big pharma, poi abbiamo l'autorizzazione dei farmaci collegati con l'istologia e quindi la pratica clinica e i modelli organizzativi. Se guardiamo al modello mutazionale, noi abbiamo in realtà una pratica clinica che attraverso la profilazione genetica determina un impiego dei farmaci, alcuni dei quali non sono autorizzati esattamente, diciamo, per quella mutazione o per quelle mutazioni. Di conseguenza ci troviamo di fronte ad una opportunità di cura rispetto alla quale però i dati sono ancora dati non consolidati e abbiamo livelli importanti di incertezza. Questo porta a considerare la fase post-marketing, quella che citavo prima come real world clinical trials, a sviluppare, e lo dico ovviamente all'Alleanza contro il cancro e quindi alla rete degli IRCS oncologici, a fare della pratica clinica un'occasione importante, irrinunciabile per produrre protocolli di ricerca e quindi per valorizzare ancora di più l'oncologia in termini di ricerca, in qualche modo invertendo anche il rapporto tra ricerca e pratica clinica e assumendo quindi la pratica clinica come luogo, in questo caso preferenziale, su cui sviluppare attività e progetti di ricerca e di nuove conoscenze. Questo, a mio modo di vedere, la situazione oggi legata a questo documento di consenso sulla oncologia mutazionale in Italia. Next steps, certamente la necessità di una implementazione strutturata di molecular tumor board, al di là di alcuni esempi, di alcune esperienze molto qualificate già esistenti, l'inserimento nelle tariffe e nei LEA della profilazione genomica, l'istituzione della piattaforma oncologica nazionale. E io mi auguro anche che nella prossima legge di bilancio vi sia una norma che, come dire, dia fondamento giuridico all'oncologia mutazionale, senza entrare ovviamente nel merito dei molecular tumor board, eccetera, ma semplicemente per richiamare i principi, lasciando poi le attività e anche, diciamo, i ruoli attuativi al Ministero della Salute, all'Agenas, all'AIFA e alle regioni. Quello che è importante, secondo me, è dire che oggi l'oncologia italiana, nelle sue diverse espressioni, e la pratica clinica hanno in qualche modo anticipato i processi istituzionali, anche i processi regolatori, creando una base di consenso di straordinario valore appunto per l'organizzazione e lo sviluppo dell'oncologia mutazionale in Italia. Grazie.
Grazie Nello, molto interessante, è uno sforzo comune che credo noi stiamo facendo per il Paese, quindi è davvero importante riuscire a raggiungere questo obiettivo. Volevo solo sottolineare che il Presidente, diciamo, nella giornata della ricerca al Quirinale, il Ministro Speranza ha sottolineato due cose. Uno, come tutti gli italiani, i pazienti oncologici avanzati debbano avere accesso agli screening genomici e di sequenziamento. La seconda cosa che non può essere un limite il non avere la disponibilità di un farmaco per il trattamento di questi pazienti, quindi siamo in linea su questo. Credo che a questo punto possiamo proseguire dovrebbe esserci Sandro Pignata. Sandro Pignata is going to talk about Forza, the ACC clinical research infrastructure. And thank you, Sandro, for joining us.
Good morning to everyone. My name is Sandro Pignata. I'm a medical oncologist. I work at the National Cancer Institute of Naples. And I have been appointed together with Paolo Pronzato as coordinator of this committee, Comitato Ricerca Clinica and Strategia del Farmaco. I have the topic today to illustrate our project that is called Forza. So this strategic committee has been designed in September 2018. Is a strategic Alleanza committee that has the main goal in trying to have actions devoted to promote academic research and in particular to integrate clinical science with translational objectives. And this committee is composed by members of all the institutes belonging to ALLEANZA CONTRO. But as a background for this action of Alleanza, we have to remember that the clinical research in Italy is regulated by a specific law for academic research since 2004. This law contains simplified rules for clinical trials approval and also allow to have academic trials with reduced costs, particularly in terms of regulatory action and control arm drugs and insurance. However, this law has a specific problem that is now very important for us as clinical researchers, that is that there is no possibility of registration of new drug or new indication for all drugs based on the clinical trials performed according to these laws. The role of the 2004 law was very, very effective, particularly in the last year. So if we look to the data of the European community in the years between 2000 and 2009 and look at what happened in terms of academic research compared to commercial trials, as you can see in Europe, taken overall, the academic trials were decreased significantly after the approval of the new directive for the good clinical practice in Europe, while the commercial trials increased. So it was clear that the new directive for GCP make the academic research more difficult than in the past because of the increasing cost of the clinical trials, the need for more operational support for the clinical trials. After this, the Italian government at that time designed the new law to counteract this phenomenon, so the increase in commercial trials and the decrease in academic trials. And this law has been effective because compared to other countries, particularly in those periods, Italy was able to maintain the number of academic trials over time. So the goal of that law at that time was good because while in Europe there was a 25% decrease in academic trials, in Italy there was a stable number of trials in the academy over time. According to the principles of the 2004 law, the research and the clinical trials approved as a non-profit trial need to have the goal of improving the national health system. However, this is the main problem of the law, that in case we get positive results from the clinical trials, it is impossible to adopt the new indication in the clinical practice. So at the end, we are unable to improve the national health system outcomes, even in case of positive trials. And though this was a big problem, particularly in the last years, when according to the precision medicine revolution and the availability of a number of target-based therapies in oncology, we were unable to compete with the other countries, with the researchers of the other countries, because no pharmaceutical company would like to invest in our country for an academic trial that has no possibility to modify the indication of the drug. So this was a big problem for us. In May 2019, a new law has been published, and this law opened a new scenario. I have not translated the articles of the law that are related to this, but in particular, the new law established for the first time that also trials made in the academic setting can translate in a registration of new indication of new drugs. However, this law, this principle requires that other directives related to this principle should be produced by the government to define how this process should be regulated and how this could happen in our country. And these directives have not been published yet. So at the end, in this moment, we still rely on the law of 2004. So according to this new scenario, when the directives of the new laws will be published, we will have a great opportunity to compete with our international colleagues to have clinical trials in academy supported by the pharma industries, but with now the opportunity to have a change in indication and approval based on potential positive results of our research. However, on the other hand, since the regulation in Europe is really complicated, and of course the good clinical practice require a lot of actions in the management of the clinical trials, to have an academic trial with registration intent, we need to have a better quality of this trial. So we need to improve our capability to operationally control the trials and make the quality of the trial itself higher and close to the quality that we usually have in the pharma studies. So I have to say that having academic trials with a possible registration intends open also the new possibility for have academic research that have both clinical and translational endpoints. And this is a big problem now because most of the translational projects are related to pharma studies. So we do not get the results in many situations, and the results of this research are only related to the main goal of the clinical trial and do not contribute to the increase of the knowledge in the specific field where the trial is performed. So I think, in my view, that this new scenario open a very important opportunity for the Italian clinical and biological research. And we need to get this opportunity, but need to prepare to this new scenario. So based on this background, in our strategic committee in Alleanza Contro il Cancro, we designed a project that is called Forza. Forza is the idea to build a new infrastructure in Alleanza Contro il Cancro that may work as support to the academic research and have the possibility to guarantee the good quality that is now required for these academic trials. Forza, the idea of Forza is based on the fact that many institutes belonging to Alleanza Contro il Cancro have operational skills. They have worked as promoter in previous trials, and if we put together all these skills, we can have a central infrastructure that may be useful for all the activities of all the institutes belonging to Alleanza Contro il Cancro. And of course, to do this, we need to harmonize. Many of us, many institutes have, for example, procedures to improve the quality. Many of our institutes are producing new procedures based on the new GCP requirements. But the idea of Forza is to harmonize, share, and make this procedure common for all the institutes of Alleanza Contro il Cancro. So Forza has been, the project Forza has been hypothesized in 1.5 years ago. And according to the discussion with the board of Alleanza Contro il Cancro, is one of the strategic projects of our consortium. Forza today has the approval of 21 institutes and one research group, that is the Italian sarcoma group. So there are 22 members that are component of Forza today and want to proceed in this project altogether. We did at the beginning a survey evaluating the operational skills of all the institutes in terms of ability to promote clinical trials. And so we realized that, of course, there is a great heterogeneity in terms of operational capability in the different institutes, but putting all together, there is the possibility to have a common infrastructure that may help academic research in our country. Our program has been designed in 2019. First, we have defined to have a coordinating group, and then we have planned to have two main activities. That is, the first is a course, an educational course for monitors and auditors that should be the people working in Forza for the next years. And last but not least, to have a common informatic operational platform. Let's go to the first point. The coordinating group has been appointed at the end of 2019. These are the persons belonging to several institutes from Alleanza Contro il Cancro. These people have been selected based on the fact that their institutions have coordinated as promoter a higher number of clinical trials. This group is led by Paolo Pronzato and me, and we have also an Alleanza project manager that is Davide De Persis. So this is the group that is now conducting most of the activities that Forza is planning. The second point is the educational course for monitors and auditors. We have organized this in collaboration with the Ministry of Health, Istituto Superiore di Sanità, and AIFA. I think this collaboration is very important because it means that this institution recognize the role that Alleanza Contro il Cancro may have in this action to improve and promote the academic research. The course was planned in April 2020 and of course was cancelled because of COVID. It was first delayed to September, but at that time, Istituto Superiore di Sanità was still unable to host a meeting in person. And so we are now discussing to have, by the end of this year, this meeting organized virtually. And we still prefer to have this in collaboration with the institutional with the Istituto Superiore di Sanità and AIFA, because we think it's very important to conduct this action together with them. This is just the first part of the program. As you can see, it was divided in 5 days with frontal lectures, and then there is an on-site part where the monitors and auditors will contribute to monitoring actions done in the several institutes of Alleanza Contro il Cancro. So at the end, the plan was to have 40 people trained as monitors according to the AIFA criteria that will be part of Forza and can contribute to the activity of Forza in the next clinical trials. The last point is the informatic operational platform. The institution where I work as a central unit that is coordinated by Francesco Perrone, that has a very long experience in the conduction and coordination of clinical trials. We have an informatic platform, and the idea in Forza is to build a new version of this informatic platform that may become the ACC Forza platform to facilitate the clinical research planned in our consortium. This platform has several functions: manage fully the regulatory process with AIFA and with the local ethical committee, as internally trial-specific electronic CRF that, of course, are needed to collect and capture the data, have the possibility to make accountability of biological samples in trials where a biobank is planned, and also it can collect patient-reported outcomes from patient devices, and also to upload imaging examination where a central revision of the RECIST criteria outcomes is needed for trials that require this for registration, and also drug accountability, pharmacovigilance, and so on. So all the operational parts of the trial can be managed electronically through this platform. So the idea now is to build this new version that will be the tool that Forza will use for the next Alleanza trial. So in conclusion, I think it was a long time since we thought for the first time to the idea to build Forza. Now, I have reflected over time if there is still the need for this action, and I'm convinced that there is still the need, because we need to make the clinical and translational research in Italy more effective. And of course, Alleanza Contro il Cancro is the best consortium to promote this action that will help in general all the research in our country. However, I have to say that the project needs now to enter quickly the operative phase. And so to do this, I think that the Alleanza board and the Alleanza institutes need to give us a full endorsement to proceed effectively. And otherwise, I think that an idea will still remain an idea if we do not have the support, because this will require a lot of job. And so we need to have a clear signal from Alleanza to proceed in this direction that I think is what is needed in this moment in this phase of transition from the old law to the new law for clinical research. Thank you for your attention.
Thank you, Sandro, and I really wish to thank you, Paolo Ponzato, all the colleagues that are involved in Forza. Forza is actually the driving force of Alliance Against Cancer. We really need to implement the clinical activities, and what you guys are doing, trying to improve the systems to get the regulatory value of the clinical trials and the education, the platform for clinical trials are really key strategic points for Alliance Against Cancer. So you have certainly the endorsement of the executive and I believe of the directive as well. Is there any question for Sandro? I don't know. So I have to say that there is a contemporary, the first day of the IOM meeting. So there's an effort for the speakers to connect and because they switch between the two meetings. So maybe we can go on and start some discussion. Pier Giuseppe?
Okay, so here I am. We can start the roundtable discussion, and so the topic is the same of the session, which are the challenges to allow our patient to access genomic screens and drugs. What should we do to maximize the clinical value of the genomic screens? So we'll have a contribution from a couple of discussants, and I invite all the speakers of this session to remain at this roundtable. The first contribution is from Dr. Saverio Cinieri. He's the elected president of IOM, the Italian Association of Medical Oncology, with a short video, if we can send it.
Buongiorno a tutti, mi dispiace non essere lì con voi, ma come sapete sono in contemporanea impegnato nello svolgimento del congresso nazionale IOM. Però IOM ha recepito questo documento di consenso sullo sviluppo e l'organizzazione dell'oncologia mutazionale in Italia, e l'ha recepito così tanto che ben due degli autori, me, il presidente eletto, e il dottor Beretta, il presidente, sono appunto nel panel degli autori. Perché è così importante questo documento? Perché la medicina di precisione è il futuro e forse anche già il presente dell'oncologia medica, e perché tutti insieme, tutti uniti, faremo in modo che la difficoltà a eseguire i test di medicina di precisione, la difficoltà a recepire i risultati degli stessi possano essere unificati. Noi come società scientifica chiediamo un'unica piattaforma nazionale in cui tutto possa essere collegato, in cui ci sia facilità di accesso e di valutazione dei test, e in cui ogni oncologo medico possa attingere informazioni e attingere sapienza per la miglior cura del proprio paziente. Grazie a tutti e buon lavoro.
Okay, unfortunately, as he mentioned, Saverio has to attend the national meeting of IOM, which just started, and he won't be able to participate to our roundtable. The second contribution is from Alberto Bardelli. Alberto is PI at Candiolo in Turin. He's past president of the European Association for Cancer Research. He gave remarkable contributions at various aspects of on precision and personalized medicine. Alberto, please.
Hi, can you hear me?
Yes, we do.
Yes. Thank you, Pier Giuseppe. Thank you, Ruggero, for this very kind introduction. It's a pleasure to present to you a few slides today. As you know, my group has been focusing on the challenges to deliver precision medicine based on clinically valuable tests in the CRC space. And recently, we have worked extensively in the space of liquid biopsy. So today, I'd like to present unpublished work on how we are translating liquid biopsies in clinical practice. And cancer and colon cancer is an evolutive process. Those that were attending the previous days have heard from Mariangela Russo from my group how cancer continuously mutates and changes during therapies. And so a number of years ago, nearly 10 years ago, we started to reason that we needed to intercept these complicated changes in the tumor shape and molecular profile and genetic profiles using liquid biopsies. And we have done a number of studies that you may be aware of that really pointed to the role of liquid biopsy in understanding drug resistance, etc. But the challenge for the future, and I think ACC is really focused on that, is on how we can use liquid biopsy to change clinical practice. And so I thought for this roundtable, I would put forward a few questions that we have been thinking along, and I'll address them in two examples with two clinical trials that are ongoing. One is finished, in fact. Among the questions that we decided to tackle using precision medicine and genetics in the space of colon cancer is which patients are cured by surgery alone. This remains a major issue and a major clinical challenge. As you know, many patients with stage 2 and 3 are cured by surgery, but you are not sure whether the entire set of patients that have underwent surgery have in fact been cured. And so in the adjuvant setting, they receive chemotherapeutic agents, some of which are toxic and can have severe side effects. And so the goal here is whether we can avoid post-surgical chemotherapy or de-escalate post-surgical chemotherapy using ctDNA as a marker of chemotherapeutic resistance. And so we designed Pegasus. Pegasus was very complicated to design. It's the first clinical trial in Italy that is interventional in this space and wishes to measure minimal residual disease in colorectal cancer and decide based on a ctDNA, so based on a blood test, whether chemotherapy can be or not be escalated. And I'm very happy to present Pegasus, which is this clinical trial that is described here, is also supported by an ARC 5 for 1,000 grant that I'm the PI of. And so here is Pegasus based on post-surgical and pre-surgical liquid biopsy. According to the positivity of the liquid biopsy, the patients, as you can see here, will receive 2 types of chemotherapy. Then there will be a second follow-up, and whether, depending on whether the patients remain liquid biopsy negative or become positive, they will further be assigned to different therapies. So this is an important concept. We are not talking to an observational trial. We are talking about a prospective interventional trial. And associated, as you know, possibly my work, we do a lot of work on preclinical models. So we have designed this clinical trial to intercept from the beginning to the end these patients in multiple ways, including blood, tissue, feces, and radiological imaging, including a new way of analyzing radio imaging. It is a complicated trial, but it is where liquid biopsies should be in the future. I'm very pleased to say that Pegasus has started in August, despite all the challenges of the pandemic, has already accrued 5 patients, and we keep going despite the very challenges. These are the centers that are involved. There is no time to describe the role of each of them, but it summarizes in this picture. And we are very pleased that we were able to do this in Italy and centralize this in Italy. The second question that we wanted to ask, also in the space of liquid biopsy, personalized medicine, and genomics, is can we guide therapy with liquid biopsies in another setting, which is a historical setting for me, and it's the treatment of patients that have colorectal cancer that is KRAS and wild type, and BRAF wild type with antibodies, which are mentioned here, cetuximab and panitumumab. And historically, we and other groups have contributed to understanding the resistance in this space. You can see that the emergence of RAS is a very prominent mechanism. And so we designed a clinical trial that started from our initial data. So can we re-challenge patients that have received an initial treatment with these type of therapies based on the status of RAS and other markers in their blood? And as you can see here from the very beginning, what we learned is that these patients can respond multiple times. Of course, this was done and is done largely empirically. And so we wanted to see whether we could rationalize the approach. And so we designed Kronos. Kronos is finished, actually has finished accruing last week. So we are very, very excited. It was initially started by Giulia Siravegna in my group. Conceptually, in the preclinical space, Giulia now leads the liquid biopsy facility of unit at Mass General Hospital and Harvard Medical School. She's an assistant professor at Harvard Medical School. So liquid biopsy can help the career of people as well. And this is Kronos. We measure the rise and drop of RAS in the blood of the patients. And again, it's interventional. So we decide what to do based on that. This is the panel of mutation that we measure with the new addition that we made during the amendment. And this is the accrual of Kronos that, as I said, has finished. I am not in the liberty of giving you the details of the results at this stage. I can just tell you that we are very excited, let me put it this way, very excited by the results of Kronos, and we will be reporting the data very soon because they are very important for the scientific community. One example, a patient that has received multiple cycles of panitumumab or another anti-EGFR therapy based on the data that we detected in the blood. So it's interventional. I want to state this again. And this is the outcome of the patients. All these require a complicated network of reporting. We had to define a new way of reporting the data between us and the pathologist. This is a lot of work that was done in order to define the reports. And in conclusion, my group is taking forward the concept of liquid biopsy to the new challenge, the real challenge, which is prospective intervention. And that's where we are. And with that, I'll close by thanking all the people that are involved, including the people of my group and many, many other collaborators that I didn't have the time to properly thank because Pier Giuseppe wanted me to stay in 10 minutes, which I did. Thank you
very much, Alberto, for the contribution. Very exciting. You're right. You must be excited, and for being in time. Are there questions from the roundtable discussions for Alberto? Ruggero, you have the microphone off.
Sorry. I have a question for Alberto. And so I suppose that the data from Kronos would be positive and that this is very exciting for me. And I have to say also a little bit unexpected because it was very rational, but was ambitious and not so easy to carry out. So really, congratulations. But how easy would be to transfer these results into the clinical practice? Because this is, I think, it could be a major hurdle.
Yes, so you're right. It was very challenging to design, and we learned a lot. So we had, for example, to prepare a report that in 15 days takes the blood, gets to our lab, gets processed, and then Anna Sapino and Serena Marchiò and Caterina Marchiò will report together with Benedetta Mussolini, the technician, the results. So this was very challenging. I didn't say the trial is positive. I said I'm very excited by the results. We are reviewing all the TAC, and as you can imagine, we are in that step. Actually, we think this will be clinically applicable because you can. The way we have designed it in the end is just one blood test after a certain number of period after the last challenge with an anti-EGFR. And we want to take this forward to a very large trial in which we we hopefully can change clinical practice. So I think it can be applicable. I mean, rechallenge is done in this setting already. It's just very empirical. It's not based on a molecular test, which is what we introduced with Kronos.
Thank you. There's a question from Luca Mazzarella.
Ciao Alberto, good morning. Great. I mean, I'm also very happy from the perspective of an oncologist. I was wondering, colon cancer in terms of tumor DNA shedding is probably more efficient than other tumors. So do you think that it is realistic to think that the degree to which liquid biopsy can shape the clinical decision-making in colorectal cancer, it can be, translated into different diseases, or is it something that is intrinsically more informative in colorectal cancer?
Yes, you're absolutely right. So colon cancer has the advantage, possibly over other tumor types, like, for example, lung cancer, of being extremely amenable to this type of analysis. But the advance in the technologies has been amazing, and so I think that this will be broadly applicable to other diseases as well, including breast, lung, and others where minimal residual disease, especially breast, for example, are a major challenge for the clinician. So I think that the technology has moved the field forward and will continue to do so. But obviously, we always work on colorectal, so it was where we started. Even if we can only change clinical practice in colorectal, will be a major achievement. But I agree that other settings will be more difficult. Yes.
Alberto and Luca, maybe perhaps I can take the opportunity, since Luca has been talking about the liquid biopsy as well. Since we are going to have to profile and to discover a number of relatives of patients with pathogenetic variants in the Johnson clinical trials. So in other words, we're going to have with the Johnson, we're going to find a lot of persons who will have this variant, so that increased risk to get cancer. Would it be, can we envision, can we think about a possible liquid biopsy study for monitoring this healthy person at high risk of getting cancer? I just remember it's colorectal cancer, younger than 50, so the target of the patients from which we can get the relatives, so colorectal cancer, breast cancer, and ovary cancer.
So I'm happy to start, and then Luca, I'm sure, can add to that. So I think there is a large opportunity there. Colorectal cancer in young adults has been increasing massively. So this obviously is unclear at this stage why it is happening. The possibility to have variants that are associated with the early onset of colorectal cancer, of increased incidence, allows us to select a population at high risk, similarly possibly to what you would do in breast cancer. And I would recommend that brings the technology to the minimal residual disease space, right, where you have to be incredibly sensitive. And so I think the same technology in principle that are applied to the minimal residual disease can be applied to the setting you propose, Roger. And I think we can certainly integrate some of these analyses into GERSOM, and I think that would be very exciting. In that case, I would recommend a multiparametric liquid biopsy, something we have started to do. So not only ctDNA, but also other markers that altogether will improve sensitivity and specificity of the test.
Yeah, and attaching my comment to what Alberto just said, of course, one major difficulty in this setting is to identify which mutations or which other blood parameters are specific for the tumor. And of course, when you are in the early diagnosis setting, you are in a different setting than in the disease follow-up, because the disease has not manifested yet, so you have no access to which mutations will develop in the tumor. So either you are in a situation where you have a few mutations covering the vast majority of the cases, for somatic mutations I'm talking about, or you are in the more common situation where tumor may arise following a number of different mutational processes and a number of different drivers. So I think within the GERSOM trial, we are in an ideal situation for doing that, because the first stage will be to better characterize which are recurrent mutations that, for instance, populate the genomes of these cancers or diagnosis, so BRCA-related cancers, mismatch repair-related cancers. And then when we have that information on a very large population that the GERSOM will allow, then we will also be in a position to test with the same multi-parametric panel that may be focused on specific high-risk settings, also the relatives of these patients. So I think we have a great opportunity in the GERSOM trial to carry out this type of research.
Okay.
Thank you. Is there any other comment or question from Giuseppe or Gennaro? So perhaps we can stop here, and we should start in 10 minutes. So what do you suggest, Pier Giuseppe?
No, we go ahead. I think that the best thing is to go ahead with the roundtable. The roundtable, we have 40 minutes of time, and we can continue the discussion because without interruption. Is that okay?
Okay.
Okay. So as I was mentioning, Paolo Marchetti, which was also scheduled, cannot participate because of the concomitant meeting. So I would like to go ahead with this discussion with the speakers, with Alberto, if you can remain. Maybe starting from the two big issues that are coming out, and they came out from the previous talks and from this discussion, which are the major hurdles to allow for genomic screens, both in terms of diagnosis, rebiopsy at the metastasis or liquid biopsy, and for accessing drugs, which I guess is right now a major problem. And Fabian, would you start with your opinions on that?
Fabian just has to go.
Sorry, because he told me that he was able to remain.
He was here, but just congratulate for the excellent talks the meeting just a couple of minutes ago.
Okay. Fine. So at this point, if there are there comments from the speakers on these general issues? Otherwise,
yes. Thank you, Pier Giuseppe, for to underline the needs to have a new tool to characterize better our patients. Indeed, also for the immunotherapy based on CAR-T, these are becoming a big issue, especially for solid tumors. Because patients are introduced to CAR-T program with very long time before their biopsy on primary tumors. So the majority of the time we are never know about the status of the tumor at the time of the enrollment. Just an example, so in our institution we are performing a CAR-T in sarcoma patients. So we are the screening of the target antigens on the primary tumor at diagnosis, but for us it's impossible to perform a biopsy at the time of the enrollment. So liquid biopsy needs to be also incremented in all the field for target therapy for patients.
Yeah, thank you very much, Concetta. That's a good point. We do need to know our tumor when we start treatment, and we don't know it's not sufficient to know how the tumor was 2 years before, and either liquid biopsy or more biopsies or metastatic biopsies are needed.
Giuseppe, sorry, may I ask you also a question following up on Ruggero's point?
Sure.
There is room. Thank you. Very quickly, I would like to go back to a comment by Alberto and also by Luca. And Alberto was mentioning the multimodality that may be needed in some cases to really fetch and capture some of the tumor cells early on. I was wondering whether both you and Luca can expand a little bit on this. So you think, for example, that circulating tumor cells may be an option, an opening on this, or you are thinking about other multimodality tools?
I can start if you want. I think in the early detection or minimal residual disease, which are two settings that are relatively similar for some aspects, but let's talk about early detection. I'm not sure that circulating tumor cells, especially in the tumor in colorectal cancer, for example, would be valuable. But what I'm referring to is the use of standard protein marker like CA that, of course, you can integrate. And to the recent data that are coming out from large efforts done at Hopkins or by Grail, in which they have identified additional markers, which many of which are protein driven, that also increase the sensitivity and the specificity of the test. So I guess where I'm going is ctDNA, so mutation, single nucleotide variants, but also a structural aberration and protein-related markers. So this multiparametric liquid biopsy has the advantage of allowing you to identify the site of tumor of origin. I forgot to say epigenetics, but of course marker in the epigenetic space. So if you are in the early detection, you don't know, well, in the gersom, you may restrict the type of tumor that you expect to arise, but otherwise you don't really know. And so in that setting, knowing information from the protein marker, from the marker of the tissue of origin, which are typically built in during the development, will help a lot to identify the site of origin. And that's what I was referring to.
Okay, thank you.
So talking about, this is Gennaro, talking about additional circulating biomarkers, obviously there is the questionable point that microRNAs might be relatively highly variable from individual to individual. However, they might be very early biomarker or something wrong that is going on in the body. So what can you comment about that, Alberto? What is your thinking?
Yeah, I can try to comment, but I'm not an expert on microRNA. There is an extensive analysis being performed. I think they should certainly be incorporated as a test in the test. I typically am very critical, so I'm not aware of a specific study that has shown that microRNAs can help in the early detection, but I may be wrong. Okay, so I don't know enough, Gennaro, sorry. Maybe someone else in the panel knows more than I do.
So if I can add to Alberto's comment, I agree with him that multiparametric analyses are going to be important in the early diagnostic setting. They are probably going to add the sensitivity in a significant way. However, of course, one caveat there is going to be cost and feasibility, because the more parameters you analyze, the more sample you need, and the more expensive the whole test gets. So I think it's probably going to be very important to start this avenue of research in a very, I wouldn't say tumor-specific way, but identifying groups of individuals which have very well-defined risk that you can quantify with a dedicated panel. For instance, if you have BRCA, the spectrum of tumors is very variegated, but the vast majority is, of course, breast and ovarian cancer. So if you start, taking these very well-selected populations where you are in the position where you can collect also the biomarkers from the first diagnostic samples, I think it becomes very realistic to identify liquid biopsy tools for early diagnosis that can realistically be implemented in the clinic. There are other efforts, for instance, even Guardant Health is trying to provide a very, very large panel that can, in theory, identify every type of tumor in every setting. I think this is, of course, the holy grail of early diagnosis. I find it difficult to find one panel that fits all tumors for all patients, every age and every risk group. So probably, a stepwise approach in that direction is going to be the best way forward for me.
Yeah, I think I perfectly agree, because I think it's important that we distinguish clearly the clinical settings, because the needs and the clinical questioning, when you're talking about early diagnosis or minimal residual disease or treatment monitoring, are very different. And the compromise you have to make between space, genomic space, and depth becomes really important to achieve the goal. Also for the early diagnosis, the stricter, the sharpest is the population that you want to study, probably the best available tools are now, the best tools are now available.
I wanted to go back to the comment that Ruggero made earlier on, and I think Luca followed up. So where there is a clear opportunity, I was thinking more, is if Gerson identifies subpopulation of patients at high risk, right, in a very effective way, then we have what I sometimes call the Italian way to catch up with Grail and on the others, which would be to find a clever niche in which liquid biopsy for early diagnostic can be applied. Because we don't have the money, of course, that are available to these large consortia, but we definitely could interact and intercept these patients in a multiparametric liquid biopsy, something that my group has been working the last, so I didn't have time today to get into the details, but we have evolved in our sensitivity. We have now very advanced panel, I would say, that are very sensitive. And so I think that there is a clear opportunity there, and I'm not sure how far you are in designing this part of the trial, but I think intercepting individuals with high risk and looking at early diagnostic there would be very, very, very relevant clinically. I
agree. Alberto, perhaps what we can do, because I try to summarize the Gerson. The Gerson is, so we run this panel of almost 500 genes at germinal level with 172 genes whose variants are predisposed to cancer, and the somatic. Then we also amend to include the polymorphism analysis, genome-wide polymorphism analysis density. This is going to be done at IGM, and also we'll probably do the transcriptome analysis. It could be that we'll use a cheap assays like the Lexogen on the 3' untranslated, but perhaps we can find more money to have a more sophisticated assay. So we will. There's going to be a lot of research because no one has investigated with a good number of patients or people the importance of putting together the signature, the polymorphism that's predisposed to cancer, and the pathogenic variants. But we will certainly identify a lot of relatives, and we also are collaborating with a group of patients or people who are positive for these pathogenic variants, and they want also to collaborate. But my question is that perhaps we cannot run certainly we cannot run a liquid biopsy analysis with Garden Health or whatever on all these people. But perhaps we can start collecting blood samples in a prospective way, and then look at retrospectively to those who, because this patient will undergo screening, special screening for based on the risk, genetic risk. So perhaps we can try to design some sort of schedule for blood collection at different stages, and try to see in case we find some positivity with the conventional imaging or endoscopy or whatever, we can then go back and look at the samples and try to study from there. This could be an option. If this is the case, we need to design carefully and plan the collection of the blood.
Yes, so this is exactly how I also would envision it, right? So it would be extremely costly and possibly unfeasible to do it like the large consortia are doing. But the way you conceived it, so you run GERSOM and then you identify relatives and then you collect the samples, and then at follow-up, some of these will score positive in endoscopy or mammography, and then we have the samples and we can, I guess it's almost prospective, but obviously it's retrospectively done. But you can then nail in on zoom in on a few patients in which you can test the panel and see whether the sensitivity would have been enough to detect early the tumor. I think I like it personally.
Well, still staying on GERSOM trial, but talking about a different thing, not liquid biopsies. If we consider the new findings from Mario Negri and Maurizio Dincalci on the detection of p53 mutations in pap smears, that relatively to the ovarian cancer patient populations, this might also be something that we could set up, and actually we could do in parallel liquid biopsies and pap smears in order to see which is the most sensitive approach.
Yeah, of course, this is actually easier because the people with BRCA mutation, germinal variants or pathogenetic variants of BRCA are many. So this would be perhaps feasible regardless the GERSOM. If you do it on the GERSOM, it's more valuable because we have a lot of parameters. It's in the framework of a clinical trial, so the patients are more controlled and the results will be more reliable. So this is certainly something that I will try to do.
Okay, if there are no other comments from the panelists, Ruggero, Gennaro, I think we can close the session and no?
Yeah, reconvene.
Yes. So I thank all the speakers. I thank the chairman, including myself, and all the audience, people that is connected. So the meeting will restart at 1:15 with a session dedicated to ACC priorities.
Thank you. Thank you all.
Thank you very much.
Grazie.
Bye-bye.
Ciao, ciao.
Bye.
Bye-bye.
Bye, Concetta.
Okay, good afternoon again. Now we are moving to important session because we need to define the priority and the strategy of ACC. This session will be chaired by Ruggero De Maria, Paolo De Paoli, and Pier Giuseppe Pellicci. Please.
Okay. Good afternoon to everybody and welcome to this important session on the ACC strategies. As all of you have seen during this meeting, ACC has experienced a very high growth and a very increase in the activities in the last few years. This was because many ideas and many proposals came to the attention of the board, have been discussed. And furthermore, the Ministry of Health decided to invest in disease networks because they have provided networks with more resources, but also they have involved disease networks like ACC in planning the strategies and the actions to shape research and care in our country. More ideas and an increased commitment require to define how to proceed. And in other words, it's necessary to reflect and to prioritize specific actions. For this reason, the board and Pier Giuseppe and Ruggero underwent a discussion since a few months, and now there is on the way the preparation of a document that will result in a strategic plan for the next years. So after this short introduction, I leave the floor to Ruggero for a more detailed introduction. Thank you.
Thank you, Paolo. As you mentioned, Alleanza Against Cancer is continuously growing. Perhaps the COVID pandemic has reduced some activities and some projects had to slow down, and probably we hope that there won't be another lockdown, and it's going to be hard, but I'm sure that we'll be able to recover the delay that is a little bit accumulating with this pandemic. As Pier Giuseppe will show, the activity of Alleanza Against Cancer are increasing with increasing fundings, increasing interaction, working groups, and members of Alleanza Against Cancer. So we actually are growing as we plan, because we plan to become the players through the Ministry of Health to try to implement all the major oncological activities in Italy. You know that the Minister of Health at the House of the President of the Italian Republic during the Cancer Research Day said, and I quote, "The implementation of personalized oncology in our country requires, on the one hand, that all the patients have access to genomic screenings, and on the other hand, that our institutes are able to collect, analyze, and interpret patient data. Not least, the availability of new drugs cannot be and should not be a limiting factor." So we actually are working on the line that has been drawn by the Minister of Health and has been endorsed by the Minister of Health. And so what we are trying to do, we're trying to improve oncology in Italy, to bring the technology in the clinical practice and to foster all the activity to the final objective, which is the goal, which is improving the patient survival and the patient management. And this is a hard task, but we decided to take this responsibility. You may know that the Minister of Health mentioned 3 major projects of Alliance Against Cancer: the Gerson project, the CAR-T project, and the Health Big Data project. Alliance Against Cancer is doing a lot of projects, and you heard in the last session, you're going to hear more this afternoon for those who will attend the ACC workshop. And there are really a lot of activities from the lung cancer trial, which was the first Italian trials based on genome next generation sequencing analysis in lung cancer. I mean, something that is not a small panel, but more comprehensive analysis of the mutation in lung cancer. We won a project on a European project on liquid biopsy that could be very critical for both early diagnosis and patient management. And there's also we are players. We coordinate the activity of the 1 million genomes, as shown by Giovanni Tonon, and a special thank to the Forza, which really in the Forza, as I said today, is the driving force for the clinical activities in Italy. And this is something that we fully endorse as Alliance Against Cancer. And also the ACC ION project that Nello Martini pointed out, so this could have a really relevant implication, particularly strong implication for patients. As I said, the minister pointed out 3 projects, the Gerson that was presented today by Chetta Lorusso. Gerson will identify at early stage 1,000 family members of cancer patients at high risk of developing cancer. So that those with the genetic predisposition to get the cancer can be directed to dedicated prevention pathways. And so it's a double objective, better treatment for patient and prevention. Prevention is critical. We hope to work more on this topic, and perhaps this project with a dedicated prevention at the germinal level could open other innovative projects for preventing the disease of Italian patients. The CAR-T project we have from Concetta Quintarelli today, there are a lot of expectation, the therapeutic potential of CAR-T. We have been charged by the Italian Parliament to carry on this project, and we feel a considerable responsibility about this. So it's really important that despite some institutions were not able to fully address fulfill the program, the milestone, it's very important that we put a lot of efforts on this. And the third project is perhaps, not sure whether it's the most important, but it's really the long-term project. Was presented yesterday by Letizia Tanca. Here we try to foster the future, the research for Italy. Not just research, but also to increase. This project could have a really critical impact on the country. So the project, as you know, is to make interoperable all the clinical data collected by the IRCCS, Italian Research Hospital. This project has an incredible potential and will prepare us for the medicine of the future, which is based on the analysis of millions of patients and millions of data for each patient. And the use of artificial intelligence on this data will allow continuous improvement of the effectiveness and appropriateness of cancer care. We believe that with our project, then all the country will be aligned. We'll try to have the data of all the Italian patients with an incredible impact on the appropriateness we're going to know who is carrying what type of drugs works, in which settings, what kind of centers will have the best performance. So this is really a huge revolution. And the activity of Alliance Against Cancer will grow, and Pier Giuseppe will talk about the future, the current and future project of ACC. We are implementing the strategic plan with very ambitious goals. We hope we can do this. But before I pass the floor to Pier Giuseppe, I just want to outline that Alliance Against Cancer is still fighting to get rid or reduce the VAT on the biomedical research. We initiated this action last year and obtained the approval of the Parliament of 2 recommendations. The first with the help of Congressman Riccardo Maggi and Congresswoman Beatrice Lorenzin, who was the previously Minister of Health. The second obtained by Elena Cattaneo, the Senator of Cattaneo, who is now struggling to get the approval of a law in the next financial law. And then please to inform you that the introductory document of the financial law, what we call Documento di Economia e Finanza, now includes the reduction of the VAT for biomedical research. And I think that I think that we can say that Alliance Against Cancer, if we're going to make it as it was the driver of this revolution, because getting rid of the VAT, not just for the economic impact, but also for the meaning of the country, is a revolution. So we'll stop here. Giuseppe, please take the floor.
I'm sorry. Okay. Thank you very much, Ruggero. Thank you, Paolo. And good afternoon to everybody. So in the next 15 minutes, I will briefly mention some of our ongoing and future specific projects in the context of our strategies, our view of the role of Alliance Against Cancer. So we started the first with a pilot study on mutational oncology. So goal of sequencing 1,000 lung cancer patients using a proprietary gene panel with patients logistic point of view, sort of stressing the network to be ready to perform multicentric studies and to quickly assign patients to best treatment. We wanted to identify obstacles to approaches, typical approaches of precision oncology. So this project was funded, is funded with 700,000 euros from the Ministry of Health. It is very clear that the distance of our country with other mutational oncology national project is huge and embarrassing. I'm reporting here the 100,000 UK in Genome project. This project consists in the sequencing, whole genome sequencing of 100,000 genomes, aiming at creating a new genomic medicine service for the national health system. So the first sequence was in 2015. End of the 2008, almost 90,000 whole genome were sequenced. And the first installment in terms of funding was £300 million. So really it's embarrassing how distant with the size of the gap within Italy and other Western countries in terms of implementation of national oncology programs. Since 2008, as Paolo and Ruggero mentioned, Alliance Against Cancro grew significantly and many new projects have been started. Here in this slide, you see that the growth of our association can be measured by different parameters. The number of members of Alliance Against Cancer went from 25 to 31. in the last 5 years. The number of active working groups, the working groups are the tools where the activities of Alliance Against Cancro occur. And it went from 6 to 11, so it almost doubled. And the funds increased remarkably, up to 16 million a year in the current year. Clearly, we are very far from the 300 million pounds that I just mentioned, but is to say that the size of the funding, both from the Ministry of Health and from private agency, increased dramatically, especially in the last 3 years. And according to the number of projects, talking about a national-oriented project, which went up from 10 to 30, so 3 times more projects. In this slide, I summarized the ongoing projects. I don't go into details of any of these because we heard about them during the meeting and others will be discussed in the afternoon during the workshop of the ACC working groups. Just to mention the areas that are covered by these 30 more projects: mutational oncology, so medical genomics, basically. Other omic approaches, liquid biopsy, radiomics, West, RNA-seq, search for new predictive markers, especially for immunotherapy, for the stratification of patients undergoing checkpoint inhibitor treatments, new treatments, the CAR-T project, genetic prevention, the GERSOM, early diagnosis, couple of projects on outcome research, nutrition, and large efforts in establishing infrastructures like clinical trial infrastructure or information technology infrastructure. In the last few months, as mentioned by Paolo, a needed debate has started within the ACC, among the ACC leadership, on which strategy for the next 5 years, which I think is an important moment. I mean, every time an association, a network like ours grows such significantly, we must ask ourselves, what are we doing? And we have to reshape our strategies. So a number of questions, even contradictory questions. The first one is the most obvious. Are we spreading too thin? Shouldn't we concentrate on what we know the best in order to be competitive? On the other hand, are we focusing too much on genomics? More in general, are we focusing too much on systemic treatments? Radiotherapy, surgery, these are critical approaches to cancer treatment. Shouldn't we also focus on prevention and early diagnosis? Or shouldn't we also focus on health research? It comes the question of whether we are not running the risk to spread even more thin. I think that this debate, which is an healthy debate, is going across ACC, brought up a number of considerations that are critical to set our strategy. The first one is that there is finally the appreciation of the relevance of Alliance Against Cancer with respect to cancer research and the Italian oncology community. And in fact, it is clear that ACC brings together the excellence of the Italian oncology and is fully integrated within the entire Italian oncology community. We are 27 research hospitals dedicated to oncology, connected with 13 universities and 36 hospitals of the national health system. Simply focusing on the internal resources, the 27 hospitals, 5,000 beds, 160,000 admissions, 115,000 new patients, with more than 9 million outpatient visits in 2018, with remarkable research performance, more than 4,000 active clinical trials, around 8,000 scientists, more than 5,000 publications of good quality. The second thought, which I guess is very important, is the appreciation which is increasing that the potentiation of network activities is indispensable to front the current revolution in the oncology field. Oncology is changing dramatically with the so-called precision personalized medicine, changing the way we're doing oncology and the chances of our patients to be cured. However, precision oncology is dramatically fragmenting all the expertise which is required for diagnosis and treatment. And it's becoming clear that network activities can guarantee cutting-edge shared expertise, think to shared technology platforms, the possibility of executing multicentric clinical studies on rare tumors, which are basically all of them, or the molecular tumor boards to share the responsibility of making proper decisions in front of the enormous genetic heterogeneity of the tumors. Patients of each hospital in this logic become patients of the network, and the network itself becomes the subject of knowledge and research. And it is clear that the network creates value in each of our institute. Third, there is an appreciation that the absence of a national plan for precision and personalized oncology comes with an high risk for our patients in terms of access to the benefits of precision oncology, personalized oncology. Either because we are not good in providing that, or because it becomes not sustainable, or even worse, because the access to precision oncology becomes uneven. Not all patients will access it. And finally, as Ruggero was mentioning, it is important to realize that the expectation of the Italian government on ACC for its ability to lead the tremendous change of oncology in Italy is high. And Ruggero mentioned the words of our Ministry of Health a few days ago, when he clearly mentioned the support of the governments to Alliance Against Cancer in its goal, in its project of drafting a national personalized oncology plan, which might then become relevant for all the health structure of our country. So in conclusion, this debate is taking into serious consideration the possibility that we may start thinking of spreading thicker and formalize the commitment to draft a national plan for personalized precision medicine, which implies a redefinition and extension of our goals and approaches respectively, in accordance with the responsibility of thinking to the entire population of cancer patients, like thinking on how we can reduce cancer mortality in Italy effectively, or extending our approach to include prevention, cure, and follow-up, being ready and able to offer medical plans to every patient, including personalized precision approaches if and when they are required. And this must be based, I guess, on our vision, which remains the DNA of our association, the fact that we are entirely devoted to cancer research and to generation of innovation as a tool of offering better chances of prevention and cure to our patients. And the second pillar of our vision, which is considering the patients as partners in treatment management, and maybe more and more in the future, partner in research governance. The important thing is also to discuss which priorities, because this is all nice on paper, but what do we do starting tomorrow in the next few years? I took the liberty to classify our priorities in 4 boxes: technology dissemination, network infrastructures, specific scientific project, and national and international positioning of ACC as a reference for oncology. Technology dissemination, I guess there are 2 lines which have been already started in our network, which should be solidified and potentiated. The first one, in terms of technology, to accelerate the transition from genomics to multi-omics. We need to potentiate other omic technologies, transcriptomics, proteomics, metabolomics, radiomics, and so on, and to set up innovative analytical approaches like single-cell analysis, liquid biopsy, and clearly apply all these multi-omic approaches to patient samples directly in the clinical routine setting, and extend our analysis to precancerous lesions. The other pillar of our technology efforts must be that of accelerating the transition towards big data and real-world data. We have to favor the collection of different types of data, including administrative documents, disease registries, or all data coming from digital technologies, from the social media platforms to the online patient communities, or wearable and environmental sensor when they will become available, and favor the generation of real-world evidence using AI or machine learning treatment of real-world data. And we should start using this in clinical research. They are already used in drug discovery or by regulatory agency. But it's important for us that real-world evidence is used both in clinical research, in terms of outcome analysis and efficacy, comparative studies, and also in clinical practice. Network infrastructures, 2 are the major projects that are ongoing. The first one is to create an informatic infrastructure for data sharing among the 50 hospitals of the research hospitals, which are coordinated by the Ministry of Health. And we heard about it, the so-called Health Big Data Project. Difficult to move toward real-world data in general to big data if we don't create an infrastructure which allows to treat those data and to share them, and if we don't learn how to analyze them. And this is the health big data, as I said, together with the other 2 networks, neuroscience and cardiology, and also the DigiCore, which is an European consortium where Alliance Against Cancro will play a leading role. And the second infrastructure that we're working on is that of aimed at generating the feasibility for multicentric clinical study, the Progetto Forza that we already discussed a few minutes ago in the previous session. Specific scientific project, I guess 2 are the thing we have to look at. On one side, we have to potentiate the projects which are already ongoing, which are funded already, or in advanced phases of the funding process. And the other one to start new ones, which are the ongoing project. The first one is the ACC project on mutational oncology. I mean, our goal is clear. We have to guarantee access of patients to both screenings and new drugs. And the approach that is already signed is the consolidation of the ability of our hospital to perform genomic screens with the project that we heard about it, the lung cancer, the gersom, and other projects for other tumor types, which are ongoing in our working groups. Creation of molecular tumor boards for each institute, and create a level of coordination among the molecular tumor boards in order to share information and to share way of decision-making tools. And most importantly, we have to tackle our government, our parliament, in order to obtain a mutational oncology program to become a national goal. And this is the sense of the ACC IOM project, which will be soon presented to the politician, which should guarantee resources to allow our patients not only to enter genomic screens, but also the molecular tumor boards to be organized at national level, and more importantly, to access drugs for repurposing scope in the context of clinical trials. Other projects are the liquid biopsy, a number of translational research feasibility projects which have been started in July, mainly focusing on stratification biomarkers, early diagnosis, and the GERSON project for genetic prevention. I don't go over this project because Ruggero has already mentioned to it. This is a large project, 4,000 patients being sequenced in a completely new approach, which is that of assessing simultaneously at the time of diagnostic both somatic mutation for therapeutic stratification and germline actionability for identification of risk, and then mapping back to the family of the patients. Project to be started, some of them potentiated. Prevention, screening, and early diagnosis. For example, revision and evaluation of very selected ongoing screenings, or project aiming at defining in particular setting the issues of diagnosis and sustainability. Health research, it has been proposed to apply health research approaches to mutational oncology. So tackling the issue of organization model, of sanitary government, cost efficacy, involvement, genetic counseling, or quality outcome control studies of other approaches, specifically surgery and radiotherapy, and research on quality of life. These projects have to be funded. We have to find the money, and I'll come back to that. And finally, the fourth box of potential activities and strategic activities for Alliance Against Cancro is that of repositioning ACC at the national international level as a reference for oncology, which means increasing the synergy and collaboration with the Ministry of Health in different sectors. They have prevention department, informatization department, and so on. Increase the interaction with national, international societies and European networks, and also the big pharmas. Start project for the generation of policies, guidelines, national campaigns of communication, especially in prevention and early diagnosis, and potentiate the scientific communication activities to the public. Where do we find the money for all of that? As I mentioned, all the ongoing projects are already financed, and they have enough to go ahead for the next couple of years. However, we need a lot of more money if we want to take the responsibility of acting as a reference in our country for oncology. And I guess occasions are there from the European community, which is privileging network activities in cancer on teams which are very close and parallel to the strategic lines that we have envisioned, like the mission board, the Horizon itself, and the IMEI and others. If we start discussing, and as we are doing, for example, with the ACC IOM project, if we start discussing about access to drugs, organization of multicentric interventional clinical trials, we have to start collaborating with big pharmas. And I guess our association is in the position to take the lead with respect to the thinking, ideation, creation of new clinical trials. Finally, our leadership has to tackle in exchange with respect to our role, the public funding, which in the near future, in the next months, will be dedicated especially to the health system. I'm referring to the recovery fund and eventually the MES. Do we have the people to do all of that? I guess yes. I mean, we have 8,000 scientists within our networks, and we need all of them. So far, we have involved a lot, basic scientists, pathologists, technologists, bioinformaticians, mathematicians, and IT scientists. I guess now it's time to extend the participation of this type of professionals and also others, which should be feeling ACC being at the center of their own activities. Epidemiologists, biostatisticians, medical oncologists, radiotherapists, surgeons, and radiologists. And I guess I stop here. So what basically my entire point is, we already started to think thick. We have to appreciate what we actually already started, transforming it into coherent national program and extend dramatically the basis and the involvement of our people in our network activity. And I thank you for your attention.
Thank you, Pier Giuseppe, for this nice talk. And sorry, I need to. Yeah, sure. Okay. Thank you for this nice talk. And now the presentation is open for discussion. I hope that the platform for questions will work, because I have old questions from this morning. While we wait, I have a couple of comments or questions. The first one is, I don't have any comment on projects, because as you said, there are ACC is rich of projects. My question regards the network infrastructures. We have sustained and developing the Health Big Data project, but there are other infrastructures, as you mentioned, but also others that can be developed. I referred, for example, to the UniCancer network in France that has a very active clinical trial office and biorepository. So, which is your idea on how we can sustain networks with adequate funding?
Yeah, thank you for the question. I mean, as I mentioned in my talk, I took the liberty to categorize projects in those 4 boxes, but I mean, the projects are pretty overlapping. I mean, for a network to create infrastructures is vital. And not only the 2 that I mentioned, so the health, the informatization, and the Forza project are needed. For example, I think that the best tool that we will have to create a real infrastructure within the ACC is within Alliance Against Cancro is the mutational oncology project. If that project goes to fruition, which means if we will be funded as we think we should and will be, then I mean that project means organizing all the genomic screens in the network, organizing the molecular tumor boards and organizing a national clinical trial. I call it like this, because the idea is not to provide money to access to off-label drugs, but to organize that access within the context of a clinical study. So I think that is instrumental, Paolo, to shock the network in terms of its capability to work as such, as Unicancer has done with the first clinical trials, I guess.
Okay, I don't see questions in the platform. I don't know if Ruggero has access to new questions.
No, there are actually no questions, and so perhaps we can probably have another meeting with the discussion of the strategic planning that has been prepared by Pier Giuseppe, and we have been extensively discussed by email all the implications. So it's clear that it's not going to be easy to fulfill all the requests, all the ideas to try to cover almost every single item relevant for cancer. So we started from what was what we believe, what I and Pier Giuseppe believed had the highest impact with a short amount of money that was available at the time, and we've been growing a lot. It's true that we have money. We don't know what is going to happen because the COVID pandemic has taken some resources from our budgets, and not just ours, but the budget of the other networks. So as you know, that this year the networks should have 50 millions and 20 millions next year, and but half of the money has been moved to research on the COVID. So it's and plus there are certainly difficulties in our lab because many institutions gave the priority of the study to the COVID, and we need to reduce the number of people in the lab, and even many hospitals, many activities has been replaced by activity to COVID. And we hope that this is going to last in the next spring, and so that we can keep on with our planning. It's true that it's not easy to plan, but certainly we need to prioritize, and we need to probably identify people who can be in charge of different sectors, can lead different projects and be responsible for the implementation, because it's very important that each of the scientific director and all the preeminent investigators that we have in Alliance Against Cancer could provide his own contribution on this. So I think that there will be discussion. We will certainly increase, and also our political activities will probably intensify our relationship with the IOM, with the Italian Society for Oncology, of Medical Oncology, because IOM is the connection with all the oncology, and so I think that there's a mutual benefit, the interaction with the Alliance Against Cancer, that has more basic research, more translational research as compared to IO that is mostly clinical. And so I think that, and the same will be with the Society of the pathology, the pathology society, and also we will probably we should have an increased interaction with the regional networks for oncology. This is key because if we want to push and implement prevention planning or other studies related to the epidemiology or to other topics that are usually done at the level of a region, I think that we should foster this interaction. Actually, there should be one member of our board that should be representing the regions, and this is not actually happening. So probably we should start from there. And with Periplo and other networks, we have eminent members of Alliance Against Cancer who are responsible for the networks in the regions. So this could be probably the way to increase the what we call ricerca sanitaria. It's probably not in the DNA of all the scientists, but it's certainly something important, and so that we can have a more holistic approach in Alliance Against Cancer. So I think that perhaps we can stop here. I don't know whether Roberto is around, and
I think that the next session will start at 2:15, so in 15 minutes.
And so the idea is that Roberto is here.
Yeah, I am already here and follow this session. So, say me if you have to wait 10 minutes or to start now.
I will go ahead. I think that I will start with the conclusion, and then I will start the workshop at 15 to 3 as planned.
Okay. Hi. The annual meeting of ACC is arrived at the final act. But before leaving the stage to our president, Ruggero De Maria, let me to thank all the people involved in organizing this meeting, all the executive, scientific, organizing committees, and press office did a really extraordinary job. The same by the chairs and speaker with their very high quality contributions. The enormous number of colleagues, 1,100, registered and actively participating the scientific session indicate the success of the event. I confess that at first we were worried about the quantity and complexity of the connection and links, but everything seems to have gone very well thanks to the technical support of our agency. Again, during this meeting, many and significant new ideas and proposals for the future of oncology have been presented and developed. But among the different topics discussed during these 3 days, I just would like to highlight 2 of them because are important not only for cancer, but for the general population. First is the importance of a correct scientific communication. In recent months, we have witnessed a fragmented, discordant communication. This has created confusion among the population and distrust of science. It is essential that the results of researches and therapies of cancer are communicated on the basis of robust data and real evidence. Second, we are in the age of molecular medicine. Access of the population to personalized prevention, genomic and biomarker screens, and new target drug, when necessary, must be guaranteed by the health system with clear and homogeneous rules. And now I would like to introduce Professor Ruggero De Maria, President of ACC. Professor De Maria is also full professor of pathology at the Catholic University in Rome and president of the Italian Institute for Genomic Medicine in Turin. I personally, and on behalf of IAO, thanks Ruggero for letting us organize this meeting, but especially for his continued support to the initiative and activity of our cancer network, ACC, that in few years has become one of the most important research associations in Europe and will grow much more in the next future. Thank you again for all. Please, Ruggero.
Thank you, Roberto. And there's not much to say because I think that we all agree that this meeting has been really exciting. Thank you very much for the organization. The European Institute of Oncology has been a driver in Alliance Against Cancer for a number of activities. And so I'm very happy that we had this meeting organized in Milan. It's too bad that that it was a meeting just online, but probably we may have other chances in the future. And plus, this opened the floor for new technologies. The IT is probably entering the communication in science in all the way, and so this is probably what we're gonna have. We should take the positive of this pandemic because we now are able to interact with more people, and we probably learn from this meeting that the technology can spread and help to reach all the different areas and also the international level. We'll learn and we'll probably do better, even better next time. The meeting was really exciting. I heard the preclinical breakthroughs were really outstanding, and I think that this is pretty unique because the other society usually concentrate on the basic science or clinical research, but we were able to span from the preclinical activities to the epidemiology to clinical translational, and this year was particularly intense and informative because we had a lot of talks on the different areas like proton therapies and also real-world data, high definition oncology, the clinical trials, a lot of topic about the genomic screens that we have today. And the high definition oncology was really organized. I have to thank Roberto, Pier Giuseppe, Gennaro, and Fabrizio Tagliavini for bringing really such a high qualified people of institutional people. We are the president of the Italian National Institute of Nuclear Physics, the president of Telecom, the president of the Francesco Profumo, the important people from IQVIA, and we have really the best scientists, the president of the Italian NIH, the president of the Italian Council for Health. So really top scientists, opinion leader, stakeholders. And so I think that this year, we thought that last year was really an amazing meeting, but I see that every year we are improving and improving. So that's really a big thank. And if I may, I would like to thank not just all the people that organized this, and of course, Roberto, Pier Giuseppe, but also Cristina Bonvicini was always helping us all the hours of the day, and try to help in every way. And thanks also to the executive and the board, that they were very helpful. Before closing, I would like to show you, I hope I can share the screen, and show you a slide of the next meeting, which is going to be in Bari, or June 24, 26, 2021. Of course, we still have some concern about the COVID, because perhaps we are confident that in springtime it should be certainly reduced. Hopefully we're going to have some vaccine, but still the location, the modalities need to be defined. And the organizers, not just the Istituto Giovanni Paolo II in Bari, but it's also the Amore Network, so with the Fondazione Istituto Tumori Pascale of Naples, and the CROB of Rio Nero in Vulture. So, and this year we have a little overlap with IOM, but I'm confident that next year the dates should be, we shouldn't have overlap with other institutions. And so I stop here, and thank you everybody. And so we're gonna meet, for those who will attend the ACC workshop at 2:45, so in about half an hour. Thank you again. Good morning, everybody. So we start this afternoon session, the ACC workshop after the ACC meeting. And we're going to have the secretary of the working groups presenting the data, at least those that... Good morning, everybody. So we start this afternoon session, the ACC workshop after the ACC meeting. And we're going to have the secretary of the working groups presenting the data, at least those that... So we start this afternoon session, the ACC workshop after the ACC meeting. Okay, now it should be fine, hopefully. And we have Miriam Alcalay, which is a deputy director of the deputy coordinator, scientific coordinator, Alliance Against Cancer, and Miriam follows closely the working group activities and the working group through the working group secretariat. And Pier Giuseppe Pedicci and Gennaro Ciliberto. Miriam, do you want to introduce the first speaker? And so it's more or less 10 minutes for a total of 15. Then we have some minutes of Pier Giuseppe Pedicci and Gennaro Ciliberto. Miriam, do you want to introduce the first speaker?
Yes, okay. So the first speaker this afternoon...
And, so it's more or less 10 minutes for a total of 15. Then we have some minutes of Giuseppe Pedicci and...
Okay. 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 So the first speaker is Caterina Marchiò, who will tell us about the progress of the Pathology and Biobanking Working Group.
Hello, everybody. My name is Caterina Marchiò, and I'm the secretary of the Working Group of Pathology Biobanking, who has a clinical coordinator in Massimo Barberis from IEO Milan, and the clinical coordinator, the figure Angelo Paradiso from Istituto Oncologico Bari. And today, in the next 15 minutes or so, I will give you an update of our project funded by Ricerca Corrente 2020, whose main aim is the standardization of the pre-analytical phase in order to achieve quality in our experimental pathology studies. The general objectives of this study were to obtain tissues of enough good quality for our downstream molecular pathology assays, and also to perform an accurate biobanking within the different institutions of the network of Alleanza. And the second objective was actually to value all of the data that we produce from the archival samples, even including all those data that are coming from digital slides. And so we also focus on digital pathology for the project. Within the frame of the 3-year program, we had specific objectives that were focused for a part of the project on the validation of DNA quality and RNA quality in tissue samples fixed at room temperature informally versus samples that have been fixed at 4 degrees, so the so-called cold fixation. And the second specific objective was to perform a survey of the imaging technologies that are present in the network of the Alleanza network, to digitalize then all the slides that are pertaining to the JAXO studies, and finally to activate the subgroup of experts that could have been useful and instrumental to coordinate the certification program within the biobanking field. So the first part of the project that is focused on the validation of the cold fixation method stems from some results that we obtained last year in Candiolo, where we demonstrated that actually the DNA fragmentation in tissue samples that have been fixed at 4 degrees rather than at room temperature have actually a better quality. So the fragmentation is actually lower compared to the DNA samples that are coming from fixation at room temperature. And this advantage is actually valued actually over time. So samples that have been extracted, whose DNA have been extracted months after the archival, are actually better if they have been actually fixed at 4 degrees rather at that room temperature. So the part of the validation of the cold fixation was a little bit ambitious and also high risk because it was strictly connected to the Gerson project, because the aim was to validate this cold fixation within the frame of the Gerson study. And for all the projects that have actually a high risk because they are strictly connected to another, we need a backup plan, because as you probably all know, the Gerson study is not actually has not kicked off right now. We are in the process of performing all the site initiation visit, and the general direction of ALLEANZA is actually curating these aspects. So we are hoping to be starting very soon. However, given that the Gerson study has not kicked off as yet, we had to find a kind of backup plan to start working on the quality of our samples. So we decided to take advantage of some samples that had already been analyzed by massive parallel sequencing by a targeted panel that is the True Site Oncology 500 panel by Illumina. And we had some samples that had been sequenced in the same run. 5 samples that have been fixed at standard room temperature versus 3 samples that had been fixed at 4 degrees. And for these samples, we did a very detailed analysis on the input DNA quality from the samples in terms of also library qualification. And then we analyzed the sequencing QC data. First of all, we realized that the input DNA was actually good for all of the samples that were submitted to the sequencing. As you can see here from the histograms, all the samples were actually usable for this type of assay, and we did not observe any significant differences in terms of DNA quality run per QC standard analysis suggested by the Illumina protocol. However, when we analyze a little bit more close up the quality of the libraries that have been generated by this assay, we realized by analyzing the bioanalyzer data, and in particular the smear analysis, that the area of the smear that was actually beyond 300 base pair was actually more represented in the samples that had been subjected to cold fixation. And this was actually statistically significant. Most importantly, if we focus on the details of the sequencing QC data, even though we actually managed to obtain a total read count that was comparable between samples that were fixed at room temperature versus samples that have been fixed at with cold fixation, all of the other mean values for median insert site, median exon coverage, so on and so forth, were actually favoring the cold fixation, suggesting that even though we may be able to obtain and to perform this type of sequencing even from standard fixed tissue samples, if we use and if we have the chance to work on samples that have been fixed at 4 degrees, we may have our life easier in terms of analysis of sequencing data. The second bit of the project focused on the quality was actually dedicated to RNA quality that had not been that detailed in our previous work. So we decided to use the core that was used for the first study, and we analyzed quality and quantity of RNA by different methods using the nanodrop and the qubit and also the bioanalyzer. All of the samples that had been sampled in parallel and fixed with the 2 methods were subjected to this type of analysis. Again, in terms of RNA yield, we did not detect any substantial differences between the two set of samples, whereas when we focus on the 260-80 ratio, we actually highlighted a statistical significant difference between the two set of samples, with higher ratios coming from the RNA extracting from the cold fixed samples. And again, when we focus on the DV200, and so on the percentage of the RNA fragments that are actually larger than 200 base pair, we observed that actually this DV200 is actually higher in the cold fixed samples, and this is true for the cohort that was collected prospectively, and also for the cohort that was actually a kind of retrospective cohort coming from a collection performed some years ago. Now, this is important because you probably know that the Gerson protocol has been amended, and RNA sequencing has been introduced. So we have the high throughput sequencing that is foreseen for this type of study, and we will have the 3' UTR sequencing that will be performed thanks to the collaboration of Dr. Cereda that is working at the Institute of Genomic Medicine here in Candiolo. And we are actually performing some tests, preliminary tests on this type of sequencing in these weeks. The second bit of the project was dedicated to digital pathology, and the digital pathology activity in Alleanza was for the first year to gain a survey of the different imaging technologies that are already available in the network. So the main aim was to create a kind of survey of the different scanners that are present in the different institutions. As you can see, half of the institutions are already well equipped with a scanner, and other institutions are actually in the process of the acquisition of this type of technology. The type of digital analysis that is currently performed in the different institutions is mainly based on research studies. And so we do not have any labs that is actually totally digital at the moment and totally digitally performing digital diagnostic at present. And so the Gerson study will give the chance probably to have a kind of a try in terms of having a clinical study totally digitalized in terms of tissue samples that are actually is actually conveying in the same study. When we ask for details about the type of scanner present in the different institutions, we realize that it is a kind of a heterogeneity between the different institutions. And this was a kind of surprising at first. And we thought that this heterogeneity could have a kind of affected our organization within the group, because as you can see, if we also ask details about the capacity of the scanner, the heterogeneity is really marked. However, we decided to create a sort of small working group, subgroup of digital pathology with the help of Dr. Filippo Fraggetta, who is working at Ospedale Cannizzaro Catania, and together with the help of Luca Di Tommaso at Humanity Research Hospital and Paola Graziano at Casa Sollievo della Sofferenza San Giovanni Rotondo, we had some theses working with Filippo to understand actually the potential of this heterogeneity within the different scanners that are available at the different institutions. And Filippo has actually reassured on the fact that having heterogeneity among the different digital scanners is actually a positive start, because we will be able at the end of the study to validate the digital analysis among any type of scanner that is actually used within the single institutions. And we are actually taking advantage of this contact with Filippo Fraggetta, because Filippo is actually a leader in this field at present. And in Italy, we can say that the first digital pathology is actually at his institution. Right now, we need to take action within the group and to liaise strictly with the general direction of Alleanza to concoct an adequate plan to set up all the digital network within Alleanza. And last but not least, we had set up the working group for the biobanking with some experts that are present already in our working group: Barbara Parodi from San Martino Hospital, Maria Grazia Daidone from Istituto Nazionale Tumori in Milan, Vincenzo Canzonieri from CRO Aviano, and Professor Angelo Paradiso from Istituto Oncologico Bari. And as a first overview of this working group, we decided to draft a review article was going to be focused on basic principle in the field of biobanking, and Dr. Laro Naratone at our institution is actually leading this review article, who is going to be submitted soon for peer review. And we also took advantage of our biobanks because we realized within our network of biobanks that Istituto Regina Elena has already performed cold fixation systematically over the past years. So Simona Di Martino has agreed to work together to validate provide a second line of validation of the cold fixation by exploiting the pressure score that has been accrued at Istituto Regina Elena. So together with the statistician at Istituto Regina Elena, they designed this retrospective validation of cold fixation based on the blind Altman method, and we realized that we need 34 pairs of surgical specimen, and for the moment we are focusing on breast carcinomas because breast carcinoma were the samples that had the highest advantage overall in terms of cold fixation. And so Simona is leading this retrospective validation of the cold fixation. Now, coming to an end, it was a difficult year for everybody, and we had many stops and go in the working group, but I was really happy to see that there are actually new proposals emerging from different institutions. I've already mentioned Luca Di Tommaso and Simona Di Martino. I would like also to take the chance to say that I have been recently contacted by Dr. Vinciani from Istituto Nazionale Tumori, who is going to be happy to propose within our working group a specific study focused on lesion of uncertain malignant potential in breast cancer. And this study is going to be focusing on molecular profiling as well as on artificial intelligence analysis. And so this was for a young working group like ours that has just been recently set up, was actually to me the most positive effect of this year. And with this, these are just 3 examples of our brilliant colleagues who are going to be joining us in the next year with active contribution, but I would like to thank overall the different institutions and the different professionals that have been working in collaboration with all of you. Thank you.
Thank you very much, Caterina. That was a very informative talk. And so we have a question from Pier Giuseppe.
Yes, I mean, it's actually between a comment and a question. Thank you very much, Caterina. I have 2 recommendations. One is very minor. I mean, as soon as we have standardized protocols, we should circulate among all the labs in ACC. And as a matter of fact, Miriam, we should put our attention to the ACC site to create a section which must be much, much more user-friendly than it is now for sharing protocols, guidelines, pipelines, and so on. Instead, the other comment is maybe important for everybody, for all the working group. So I am very happy to hear that you started this mini working group, as you called, on digital pathology. That's fantastic. I mean, and as you briefly mentioned, at a certain point, you'll have the need to structure sharing of data and an analysis of data, I guess, a connection with the radiomics, with the genomics. And I mean, I guess this is, at the end, another omic approach of digitalization of our samples. Fine. So where you are going to do that? And then I would like to to clarify this point with everybody. So we have this health big data project. We will have the DigiCore project down the road. So we are creating an infrastructure for exactly this purpose, which is data sharing. So it's very, very difficult to put together two needs. One that of creating a coherent structure, and the other one is to tackle emergent issues. Because if you wait for the first to be finished, the second are over, in the sense that we'll never satisfy the needs. So the idea is to go in parallel, two directions, bottom up and top down. In this moment, as a top-down operation, the colleagues of the Polytechnic have started a number of working groups. They are cooking in these days, where they would like to discuss with the institutes, starting with the first ones that reply to the survey, with the scientific direction, with the IT people, with the security and privacy responsible, and so on. Start discussing everything they need to discuss to achieve the goal. Meanwhile, what we decided is to start working on the specific needs. For example, last week I processed the needs of the GERSOM to share radiomic data. That's and clinical data. So what we did is to put the GERSOM people in contact with the Polytechnico people in order to start working on the processing on their need within the ACC data center, which will then migrate within the larger health big data center. So my message is very simple. Whoever has problems of data sharing, data storage, and blah, blah, the best thing, instead of starting new depository, new collections, new everything, that will be a problem then when we have to put everything together. Just contact us. We'll put you in contact with the Polytechnico people, and within the ACC data center, we start processing the needs in a way that, as much as possible, they will be coherent with the parallel top-down larger activity of the group. This has for you two advantages. One, money and people, because there is money in the health big data project. Not enormous amount of money, but some money. Instead, in your working group, there is zero money. So that's easy to map. Money, I mean, for IT enterprises. And second, we hope in this way to make a coherent growth of the urgent and emerging needs, which we cannot wait. We cannot wait to have a definitive structure to deal with the digital pathology. We have to deal with it tomorrow. And that's for everybody in the other working groups. And that's it. That's my comment, Caterina. Don't you think it's good?
Yeah, well, I was just about to say that we don't have money for that. Of course, my comment on the liaison with you was actually to do something like that. The first step was, first of all, I don't like to do things that I'm not able to do. So when I realized that I was not good enough in digital pathology as a startup, I just asked for help. And the help right now was actually outside the network, because we didn't have specific experts in this field. So that's why we liaised together with your approval with the other people that are much more involved in this type of projects. And we also realized that we didn't have the informed consent for digitalizing slides within the GERSON. So we amended this. So, okay, these are small bits for sure. Yes, the next step is not to start anything from scratch and do copy and paste or do something like Me Too things. We don't like to do that, but definitely to be integrated. Digital pathology has a big issues of space. So we need money and we need also a lot of space. So definitely
storage.
Yeah,
I don't know,
storage.
what do you think? Of this parallel bottom-up, top-down approach? Shouldn't we allow the working group to start taking care of their needs?
Yes, absolutely. But as I mentioned the other times, we really need right now to know if if other working groups have specific projects and they want to digitalize slides, we need at least to be able to guide them in where to put these slides, how to manage them, how to get access to that. So there is really a kind of impasse that is technical right now.
Okay, I think perhaps we should move on. We have two more questions, one from Gennaro.
Okay, now I wanted to, so I have a quick comment to add to what Pier Giuseppe said at the beginning for Caterina, is that a strong encouragement to write SOPs or official guidelines. This is going to be very important, more and more important. We have also to find a way to validate this outside, so to have a sort of stamp for every document that goes out from ACC. So once you have defined the protocol and SOPs, don't forget to undergo this step.
I will.
One additional thing that just to come back to what Giuseppe said at the end. For example, we discussed a lot with Vanessa about the stage 3 lung cancer protocol that we are going to start, which is going to be a combination of whole exome sequence and radiomics. And so if we have the possibility to use health big data already, if there is a space, a room available already there to put all the data in a single box without reinventing separate wheels, this is something absolutely important, and we should be instructed how to do this.
Gennaro, sorry, Miriam, I understand that I shouldn't, but that's a good occasion. I mean, this is our workshop. So,
of course.
So it is not, Gennaro, automatic in the sense that last week or 10 days ago, I don't remember, we met with the president of ENFN to modify quickly the contract that we have as ACC to accept the gersom. Now we have to discuss for the lung because there are a little bit of technicality associated. We'll do it immediately, and then to transfer to the other project. This is to say, Gennaro, that is not so simple, but can be done. We did it for the gersom, and now has to be done one by one for the others. And we have some money for, you know very well, in the budget for the storage, and we have some money for the for the software. But more importantly, we can start now, Gennaro, the hiring of the people to be assigned to the IRCs, because we have one FTE per IRCs, fully dedicated to IT structural project. And I think we should move on with the activation of that part, because we need full-time people for these things. Otherwise, then they remain a little bit theoretical. Okay, sorry, that was just to...
Okay, so we have one more technical question, and I think it would be interesting. Actually, we have two. Caterina, do you think there may be potentially specific tissue or tumor tissues which would give different results compared to what you have seen on breast? This is Maria Vinci.
Yeah, well, when we ran the first study, we had a collection of different tumors that were coming from different organs. There was not a super good representative from all the tissue from the tissue sites. So we had lung, we had colon, we had breast, we had thyroid. And so, yeah, it might be site specific, as we observed in breast. We think that in breast, the problem is the fat tissue that is affecting in a way the proper fixation. So it might be, right now we observed it in breast. We didn't run every single site and every single possible tumor, but it would be good, for instance, to assess in sarcomas, for instance, that may have, some types of these type of components.
Of course. Okay. And then very briefly, there's a question from Angelo Paradiso. priority for new accreditation 2387 ISO among ACC biobanks?
Well, it's good that actually Angela is asking. I think that this should be totally dedicated to the mini working group of the biobanks. So definitely we need to have a chat and to, again, for the subgroup of the biobankings, we decided that we do not want to do documents that have already been done by others. So definitely we need to focus on something that is going to be helpful for the network. So, well, yes, let's have a chat.
Okay. Thank you very much, Caterina, for your very stimulating presentation. And now we move on to Luca Mazzarella, Secretary of the Genomics and Bioinformatics Working Group. He already talked this morning about a specific project, and he will now update us on the progress of the working group. Thank you, Luca.
Thanks, Miriam. Can you confirm that you see the screen?
Yes.
Great, thanks. So I'd like to start by thanking also my co-coordinators, Giovanni and Simonetta, who helped me with the activities in coordinating the group. So I will try to be quick. So, there are big news, I guess, in 2020, because we managed to collect some reasonable amounts of data, which allowed us to progress in all our works. But unfortunately, as you all know, we also had a minimal issue in pandemics that didn't really help in finalizing everything that we thought. But I must say that I think that we managed to achieve quite a few goals, and I will very briefly go through these. So there's going to be 4 topics. The most important one is going to be the validation of the Gerson panel, which, as you all know, is instrumental for a number of things. I will skip this. You all know that the Gerson panel is a very large gene panel that includes a large number of genes that we deemed actionable through a review work that dates back to a few years now. A large number of cancer drivers that we took mostly from the pan-cancer analysis that was issued 2 years ago. And the same thing applies to cancer risk-associated genes. And it also adds some drug toxicity variants. And a significant share of the genes have been also optimized for CMV, meaning that the number of probes that map onto these genes has been increased because we deem these genes to be important from the copy number point of view. And importantly, this is a test that is that we are aiming to provide a relatively low cost. You probably all know that the technology that was chosen is the ion torrent-based technology, mostly on the basis of good experiences that we already accumulated on the ACC lung cancer, and also based on the fact that this is definitely a very good protocol in terms of feasibility, ease of reproducibility, and speed. Of course, other technologies have also improved over the years, but when the panel was conceived 2 years ago, this was definitely the easiest to reproduce technology available. And this is a very important parameter for a project that aims to, if you want to democratize access to NGS in the clinics. It also has the advantage of being optimized for FFP samples, and we just heard from Caterina how important the preparation of the FFP samples may be on the quality of the samples, and it allows to work from low input material. So we had to validate technically the performance of the panel, and we followed as well as possible the recommendations from the American College of Pathologists that were issued a couple of years ago. And very briefly, what we did were 2 series of validation assays, which I would group into across-base validations and across-sample validation. I will tell you about it in detail in a second. This is just to mention the specifics of the samples that we use for the validation. For the across-base validation, we use the standard DNAs, for which there is a large number of variants that have been reproducibly identified through orthogonal technologies. And also for this particular sample, there is a large genomic space, this is the overlap with our panel, that has been sequenced with multiple technologies, and therefore the variant calling, the base calling in those areas is considered to be high confidence. So when you find an A or a T or a C or a G within these 888 kilobases, you are sure, or as sure as you can be, that the call is correct. And then we started collecting samples from multiple institutes within the ACC network. So we issued a call at the beginning of the year for voluntary collection of the samples, and we so far obtained 103 samples from 9 different institutes, and the analysis that I will show you today has been finalized on 55 samples, of which 9 were FFPE biopsies and 46 were blood, and with 57 total variants. Of course, a couple of samples had dual variants. 26 of these were SNVs, 21 were indels. Now, what's the difference between a cross-base and a cross-sample validation? This is very relevant for our purposes. So a cross-sample is when you have multiple samples with different variants. This is what you expect. Let's say you have 10 samples, each with 1 variant. You expect to find 10 variants overall. You instead find only 8 of these, and then you say that your sensitivity is 80% across samples. Here, that means that each sample only counts for 1 variant. It doesn't matter whether in this sample you observe another variant, because you don't have any validation of that variant. So you don't know if that's a false positive or a true positive. Instead, a cross-base validation is when you have 1 sample, for instance, where you have a lot of variants, all of them are validated, and then let's say you have 1 sample with 10 variants, and you only see 8 of these, and then you call your sensitivity as 80%. Now, there's a number of challenges when you want to use this reasoning for validating our panel. So first of all, you don't have a ground truth, a basis of samples that cover all possible variants that you may be interested in. So where you may have a series of high confidence expected variants, but then the potential variants that you have and that you may be interested in is way bigger than what you can ever collect, even with the largest sample collection effort. So, okay, you validate these 10 variants, but then there's dozens of others that you may be interested in validating. So how do you validate that? You cannot use sensitivity as a parameter. So what you do in this case, you use coverage as a proxy for sensitivity, because you know that if you do not achieve significant coverage in certain regions, then you know that that variant cannot be called with sufficient confidence. A second challenge that at the beginning we thought was a minor problem, but instead the more we worked on this we realized how important this problem was, is that it looks trivial, but in fact different sequencers, different variant callers, different softwares may call the same variant in completely different ways. So for instance, if you have an indel in a homopolymer and you have a stretch, let's say, of 5 A's, and the sequencer sees C followed by 4 A's instead of 6, then you don't really know which of these 6 A's have been deleted. And so you may have one caller that calls it like this and another one that calls it like that. So if you don't have the correct way of calling, of reporting the variants, then you may mistakenly interpret something as a false negative, whereas instead you have the same variant and you are underestimating your sensitivity. And then a final challenge that is of utmost importance for the specific needs of the GERSOM is that not all variants are equally relevant. Some variants are clinically relevant, but some variants are not clinically relevant, and therefore the sensitivity on these variants is less important for the scope of the panel. So you may, for instance, if you have 10 variants and only 5 of these are clinically relevant, if the ones that you do not detect are clinically relevant, then your actual sensitivity in clinical terms is less than your overall sensitivity. Otherwise, if you focus on your clinically relevant variants, but you do not detect non-clinically relevant variants, then your actual clinical sensitivity is much better. And these are only some of the challenges that we are faced, and I'm pointing this out because this is also an interesting effort. It required the development of dedicated tools and which has actually improved our understanding of the system. So we started this validation effort by separately calculating total and clinical sensitivity. Not all of these data are fully mature, and we calculated, as I said, a cross-based sensitivity on the two standard samples and cross-sample sensitivity, as I said before. Now, this is probably the most relevant slide here. These are the final results on the 55 samples. So a cross-based sensitivity was 89% for the NA sample and 93% for the OncoSpan sample. Now, a very important point here is that the sensitivity is not the same depending on which type of variant we are considering. On SNV, the sensitivity is very high, but on the INDAS, as expected, given that this is a known flaw of the technology, the sensitivity was way lower. And specificity, as expected, has never been thought to be a problem. So the specificity is very high. If we call a variant, we are pretty sure that that's a true variant. We then looked at the false negatives in this sample. We had 168 out of a little bit more than 1,500 false negatives. We reevaluated in time for this conference 30 of those, and we actually realized that the vast majority of these are things that can be corrected bioinformatically. There's a margin of correction for homopolymers and for other bioinformatic issues. So, and the other important thing to highlight is that if you look at how many of the total expected or the false negative variants are actually clinically relevant by using a definition that uses a number of clinical relevance databases, you see that the false negatives are actually depleted for clinically relevant variants, significantly depleted. So that means that of the variants that we are not calling here, even if they are in theirs, only few of them are actually clinically relevant. This is a table that collects all the clinically relevant variants and validated variants that we got from the 9 participating institutions. So you see that there's an enrichment for BRCA variants, and these are the results. We got a overall sensitivity of 88%, but again here, if you look very carefully at the variants that have not been called, those variants were sequenced. So if you eliminate the variants that, if you recorrect, if you want, the variants that were sequenced but were not either appropriately called or appropriately interpreted by the variant caller, then we achieve a very high sensitivity at 98%. Basically, only one variant was not correctly called. So that means that we can definitely work on the bioinformatics side of things to improve significantly the performance of the panel. So the conclusion on this is that the sensitivity of the panel was pretty good, but it definitely can and should be improved at the bioinformatic level. Specificity is not really a problem, and that questions the need for performing systematically Sanger validation. Good aspects of it were that the performance on FFP samples was good, but the sample size on FFP was still too small. There was good reproducibility and ease of workflow, and this is definitely a good tool for large-scale sequencing. So if you want to sequence 4,000 patients, of course, you need to have a compromise with sensitivity, because otherwise the cost would be much higher. It's probably not yet ready for the complete diagnostic follow-up, although we may improve this. Very briefly on the other activities of the working group, as I said, we had to develop novel software. I will just give you an example also, because I think this is a virtuous example in terms of collaboration in different institutes. We have almost finalized a software for calling TMB. This also seems like a very trivial issue, but as you can see in this slide, if you include in the TMB calculation different types of variants, you decide that you want to remove deletions, you want to remove driver associated variants or something like that, the predictive power of TMB calculated on in silico dataset changes dramatically. So when here you have red, it means that it allows you to predict responders to immune oncology. If it turns to blue or violet, the predictive power is almost as much as flipping a coin. And these are different types of variant filtering that are used to calculate the TMB. You see that in some diseases, this change is dramatic. And I can tell you that most of the commercial panels do perform some of these variant filtering choices. Therefore, this may significantly affect the performance. And this is going to be a very precious tool for us when we want to use the Gerson for calling the TMB. We have a number of other softwares in development, and the important thing is that now we have significant amounts of data, both from the ACC lung project and the Gerson project, so we can actually have data sets to benchmark and optimize the different variants. Very quickly, and in a brief response also to the comments and the previous discussion, you all know that the platform that we initially set up for the ACC lung project is now growing into a platform for both integrating different types of data also from other working groups, both within the ACC lung project, but also within the Gerson. And just to briefly comment on the previous issue, we already have some parts of the infrastructures that collect even the protocols for fixation or for the wet lab that are routinely used to inform the centers on the procedures to use in the projects. And finally, I mentioned to the unique project, which is a project that we launched this year. Of course, there have been a number of issues associated with the COVID that have significantly slowed down the activities here. The central idea of the project is to change the focus of the working group. We don't want to necessarily anymore provide instruments for democratizing NGS. We actually want to use high definition NGS, whole exome sequencing or whole genome sequencing, to tackle specific clinically relevant issues where, for instance, the genomics understanding of the disease has not been finalized, has not been carried to the levels of big killers like lung cancer or melanoma or colon cancer that have been very heavily sequenced throughout. So the idea for 2020 was to actually have capacity building. So we were relatively unambitious in our goals because we knew that we couldn't necessarily achieve a significant improvement, but we wanted to survey what samples, what cases could be collected among the different centers in ACC. And the criteria for identifying the study cases were novelty, the relevance of the genomic analysis, the quality of the cases, number, and how rich the clinical data is, whether this could trigger synergies among different centers, and the possibility then to apply for grants for further making the projects grow. And also an added value of the project is that, as you heard from Giovanni Tonon this morning, these whole exome sequencings or whole genome sequencing may participate to the 1 million genome. These very briefly are the projects that have been presented at the workshop and have been selected. And now we are in the process of setting up a further meeting for finalizing the details of how to progress in the project. So final slide, what I think we can improve. We are definitely, I guess, a bit too slow in delivering the things that we have put as aims for our group. Probably it's also because the goals of the group are quite ambitious, I think, and they are not easy to achieve in the current situations. It's definitely difficult to manage a large and heterogeneous group of people. We should probably improve on the organization. And we definitely have a blind spot on a number of logistical aspects that are usually, put under the carpet as relatively non-important, but in fact have already in the past significantly hampered our ability to deliver. Ongoing works I've basically already mentioned. Just one thing that I know a couple of people in this meeting now are going to ask me about. We haven't yet developed a good pipeline for detecting fusions, and I think that this needs to be a focus for our activities in 2021. these are the people that have been involved in the works, and thanks a lot.
Okay, thank you very much, Luca, for your excellent talk. I don't know if we have any questions here from the panel or from the public. Let me see. No, I see no questions. So any comments, any questions from the panel?
The only, if I may say something, ask, since I have very little opportunities to talk to Luca, is to try to understand the level of engagement of the other centers and involvement of the other centers in the network in planning and conducting these activities, and which look quite a lot, and really to try to understand how the network of bioinformaticians and also other folks are connected to each other and are involved into this.
Yeah, so I mean, I mentioned it in the presentation. It's definitely not an easy task. The number of different projects is high. So basically, as there is not a single project that is ongoing in the working group, but multiple projects, the level and type of involvement changes according to the project. So there are some bioinformatics projects where we try to engage all the bioinformaticians, and this has been, I think, successful. But I must say that very often what happens is that there is a burst of enthusiasm when each of these projects is launched. Then there's a couple of meetings where we define tasks and we say, you know, this, we assign specific subtasks. And then not all the components are able, I wouldn't say are able, but are always returning the expected results. And I'm not necessarily blaming anyone here. It's just that all the activities within ACC, which are very intense also from the organizational point of view, they always enter in conflicts with some local activities in each institute. And this has been a leading theme in my experience within the group. It has happened over and over that we have failed to be on time with a number of things because at some point someone had to also commit to other things. So that's why what I am trying to do, but I admit that it's not easy, is to divide further the activities of the group into smaller working groups. This is something that I mentioned in an earlier communication this year. I tried to, especially in the bioinformatics where this is easier, to divide the tasks into subtasks and assign a specific coordinator to each of these subtasks. I sent a communication like a week ago, and I will try to organize a new meeting with one specific for the bioinformaticians, where we will set up the organization of the working group with these smaller coordinators. For the TMB, it worked pretty well so far, for the TMB software.
If I may ask another question, going to a more practical question, the GERSOM program project has suffered some delays, which are reasonable because the protocol has been amended and now is going back to the ethical committees for approval. So we expect starting to enroll patients basically since month. So we might have the centers ready with patients to be sequenced already starting in December, hopefully. So are we ready to send the material to the various centers for sequencing, or so would you expect any further delay?
So this is something I try to make clear in the presentation. I do think that there is an issue with logistics. And I want to be very open on this. We are not, we as our institute we are not going to take care of the logistics for the entire network. And we have discussed, and I guess we should discuss a little bit more in details with people at the administration in ACC on how the delivery of the reagents is going to take place when the study actually kicks off. I do think that this is an issue, and I think this should be definitely discussed, because it seems like something like the elephant in the room, if you want. You know, we've been thinking about bioinformatic validation, reagent validation, and everything else, but then these organizational and logistical steps are not well taken care of, I think. So we should devote some meetings for doing that.
Okay, so Pier Giuseppe has a comment.
Very briefly, because that's a critical issue. So let's distinguish logistics from science. Do I understand correctly, Luca, that beyond the fact that the pipeline, the bioinformatic pipeline, has to refine further, we can start sequencing? Yes, right?
Absolutely.
Okay, so the validation is over. Now, the problem is the logistics. The logistics is a nightmare. We know, because for the lung, we served that function as an institute, and we went really crazy after that, especially with our administrations, which had problems in handling it. Now, the money, finally, is in ACC. So you should take the lead. I'm not talking about you, Luca, to tell to the PI and to Chetta to organize a meeting immediately with Enrico Patiti and with Paolo De Paoli, because nobody's taking care of this that I know. So it has to be done really urgently, because it is going to be a nightmare. And the last thing I would like to see is that we fixed everything but the stupid logistics. And I'm worrying that this is what's going to happen. So please take the lead, organize a meeting with De Paoli and with Patiti, together with Chetta, and we have to tackle immediately this issue.
Okay, thank you, Pier Giuseppe, and thank you very much, Luca, for your thorough update. And so we move on to the next presentation, which is from the immunotherapy working group, which has two secretaries. We heard Concetta Quintarelli this morning tell us about the progress with CAR T cells, and now we will hear from Paola Nistico. Paola?
Good evening to everybody. It's so strange not to be together, but this is our life daily, our daily life. It's up to me to present the report of the working group of immunotherapy. And here are the project milestones and the deliverables. As you could see from the RAG status, all the first part of our project is on green, since we have been able to conclude at least in a number of samples all the aims of the first year. And this regarding the previous project, differently regarding the project of the ACB-treated patients, non-small cell lung, which is shared with the trial of working group lung. Here you can see that the situation is different, and we probably need more time and more cooperation to go ahead. But I would like to start with all the positive results. And at the last meeting, we debated a lot because we have a lot of difficulties to include National Institute of Milan in the project of Immunoscore in lymph node negative patients affected by non-small cell lung cancer. Now they have been able to collaborate, and although we don't have yet the results of the score as defined by digital pathology, but the three institution participants, San Raffaele, Regina Elena, and Istituto Nazionale Tumori, we have important data that at least show that 10 consecutive sections from the same tissues may be considered to study immunoinfiltration of T, B, myeloid and macrophagic lineage. And you can see here a representative case, which is non-small cell lung adenocarcinoma, T1B lymph node negative. You could see that this tumor is strongly infiltrated by B cells as well as T cells in terms of CD4 and CD8, and there is expression of CTLA4 at least in a part of the lymphocytes as well as FOXP3. And this case express PD-L1 in the tumor as well as in some macrophages, and the PD-1 are present, is expressed in infiltrating T cells. And CD33 and C163 are, although at low level, expressed in this case. This is another case where it's very interesting to note that we have the organization of tertiary lymphoid structures with B cells at the germinative center, with the cortical of this structure represented by CD3 T cells, and this case has a low level of expression of FOXP3 and CTLA4, whereas express a level of CD163 and CD33, and the PD-L1 is negative. We hope that in the next month altogether we are able to define the score and conclude this project. And now we move to the other project, is more complex, is aimed at identifying biomarkers in immunocheckpoint blockade treated non-small cell lung cancer patients. And here is presented the multi-omics platform. The paraffin embedded tissues collected from different institutions, but I have to point that only Regina Elena, San Raffaele, and Humanitas have been sent to the coordination center, the Regina Elena, samples. Here at Regina Elena, we have the extraction of DNA, RNA after differently setting of the methodology, and now we are sharing these methodologies with the group of Candiolo to define which is the best method to go ahead. But now for this analysis, we are continuing with our methodology. And we have DNA and RNA. From DNA, we, thanks to San Raffaele, we have the HLA typing. Thanks to the EO, we have whole exome sequencing, and TCR-seq by Bambino Gesù. And starting from RNA, the majority of the work is ongoing at Regina Elena. And these are the 24 cases, tissue samples that have been analyzed to date by the multi-omics platform. The cohort of patients consists of 13 good responders identified as patients with no progressive disease after 10 months from the starting of the treatment, and 11 fast progressors who are the patients who progressed at the first CT scan. And here is presented the RNA-seq analysis workflow. And at Regina Elena, you see that we have an unsupervised machine learning clustering. We obtained from raw data supervised machine learning. This is the pipeline differential expression analysis, and by CyberSort we are able to deconvolute the tumor immunomicroenvironment. The data obtained by unsupervised analysis clustering were then analyzed by supervised machine learning, and we obtained a list of genes which separated, you can see here in yellow, the good responder from the fast progressors in violet. We were able to obtain a list of genes, and many of these genes, or at least some of these genes, overlap with the genes identified by differential expression analysis, where we found that 17 genes are down and 12 are up between the two different groups. And the deconvolution by CyberSort, although identified many immunocells which infiltrate the primary tumors of the patients treated with ICB, was unable to differentiate the two groups, fast progressor from good responders. And moving to whole exome sequencing analysis, to date we have done only the primary analysis on 20 samples, but we know that there is a high base calling quality with a red mapping ratio of 99.8%, and is in progress the variant calling, as well as the CNV calling. What is important that we are planning to integrate very soon this data with the transcriptomic and the immunological data. And thanks to Concetta Quintarelli, starting from DNA of tissue samples, we have analyzed the TCR repertoire of these tumors. And although these data are only in 10 patients, and all the patients have been analyzed, but the analysis is not still available. We found that we have some TCR beta usage in these tissues, as you can see here, where 10 out of 10 primary non-small cell lung tumor overexpress V-beta 03 and V-beta 29, as well as V-beta 12. These data are very interesting because there are not many data in literature with respect to the usage of TCR V-beta in non-small cell lung cancer patients, and we hope that as soon as all the data are available, we are able also to identify some V-beta expressed in good responder with respect to fast progressors. And moving to HLA typing, at San Raffaele, Silvia Bonfiglio and collaborators have set this methodology to analyze the HLA class 1 and class 2 haplotypes from paraffin embedded tissue. Only 10 patients have been to date analyzed, but the other 10 or 14 are ongoing and available very soon. But in the meantime, at Regina Elena, the group of bioinformatics, Martina Betti, Lorenzo D'Ambrosio, and Matteo Pallocca, were able to develop a pipeline, a framework to obtain the HLA class 1 haplotype from RNA, from bulk RNA-seq. And we hope that these data will be validated by the experimental analysis of San Raffaele group. But we found, because we found a very interesting data that out of 13 good responder patients expressed the allele HLA 2.1 with 61% of good responder versus only 10% of fast progressor. You can see here that the expression of HLA2 allele in the good responder with a p-value of 0.029. This is an interesting data, which we hope to be confirmed by experimental data. And although the study of viroma microbiome has been planned almost for the other trial, the trial with the working group Lung, we asked to Martin Schaeffer and Luigi Netti to try to understand if it is possible to identify or to analyze the viroma from RNA-seq of the data we have already obtained. And they define the computational pipeline to reconstruct microbiome and viroma by extracting bacterial reads, aligning them to microbial reference genome using the tool PATSEC, removing contamination signals, and so on. I hope if you have a question, they will clarify everything during the meeting. And at the moment, probably the quality of our previous RNA-seq and the lack of enough materials doesn't give us very comfortable, very good results, but we are trying to have more RNA from the same samples, convinced that is really important to know the viroma of these tumors. And if this is part of the project is more or less going ahead with very interesting results. The other part of the project of the working group of immunotherapy is relative to a very challenging project, which is the ACC immunoportal. This immunoportal would be an Italian repository for a more effective immunocheckpoint treatment, and our aim is to create a multicentric repository, web portal comprehensive of clinical, biological, and lifestyle data of more or less 2,000 ICB-treated patients in accordance with the current guidelines. the idea is that with this repository in our hands, integrated in the second step with the biological data, ACC network could represent an Italian task force and become an excellent reference in international immunotherapy landscape. As you can imagine, also this project that need the cooperation of all institutions is partially to date going ahead, but the coordinator center is the Regina Elena. We have submitted at the ethical committee the study, and the study was approved. Some institutions have already the study approved, and some institutions are ongoing to submit or are waiting for approval. But again, we have some institutions that are less cooperative, but I am sure that in the next year we are able to have the cooperation of all the institutions, since the last year was really full of difficulties for everybody. And at Regina Elena, Marco Canfora and Diana Giannarelli implemented a REDCap-based ECRF based on IFA mandatory information on easy treatment according to approved guidelines, and we plan to have additional fields about the diet thanks to a questionnaire that Paolo Pedrazzoli was able to share with all the institutions, and mutational profile, vaccination status, antibiotic treatment, and body mass index. In progress, there is an extensive stress test on multi-center data entry. This is an example of the how the REDCap may show the data from IFA. And after the presentation of the data that are very close to the working group of immunotherapy, I would like to present the data relative to a project which is in between of the working group of immunotherapy and the CAR-T project. And this project has been developed thanks to Massimiliano Mazza and his group at Meldola. Since at the last meeting in Rome, Massimiliano presented his data, and after his presentation, we have many meetings together, and we decided to go ahead. And Massimiliano was able to start from RNA-seq data and to extract and reconstruct the variable chains VH and VL of the antibodies expressed by the B lymphocytes infiltrating the tumors. And I would like to remind you that in many of the tissues we have analyzed, those data are from lymph node negative patients, but we have preliminary data that also in the tumors of the patients treated with ICB, and the literature have recently demonstrated that B cells are really important in the group of responders in different tumors. But no data are available with respect to non-small cell lung cancer. But Massimiliano was able so to choose the oligoclones of the good. His aim is to choose the oligoclones of the good responders patients and to produce immunoglobulins which may be directly tested on non-small cell lung cancer patients. And his aim, the aim of the group is to generate putative target antibodies by permutation of VH and VL sequences retrieved in situ, thanks to the combination with the most frequent variable kappa and lambda light chains from nivolumab best responders into human urine chimeric IgG that will be tested for binding to non-small cell lung cancer cells, and the consortium have a large panel of these primary cell lines. And so we consider this project really a new hope for both the working group of immunotherapy and the CAR T cells. For every question about this part of the project, sure Massimiliano will be answer at all the questions. So now I am finished my presentation, and I would like to thanks all the collaborative participants, and to remind that it's not so easy to work in the network, but I would like to tell to everybody that many results could be obtained by this cooperative work. So I hope that in soon many more institutions are collaborative, as some institutions have already done. Thank you.
Thank you very much, Paola, for your very thorough update on the immunotherapy working group. Are there comments or questions from the panel? Let me see.
I have a small question for Massimiliano. I mean, Paola, I was impressed by the extensive characterization of those of these for the immunoscore. I mean, just a question for Massimiliano. Massimiliano, what is the evidence that the tumor-infiltrated B cells do indeed produce or should produce blocking antibodies?
So there are some reports which show that for some tumor types, if you transplant those tumors into mice where you don't have B lymphocytes, I'm talking about syngeneic models. Basically, the effect of immunotherapy, which is usually eradicating the disease, is not working anymore. And moreover, there are a lot of, as Paola was already mentioning, there are a lot of reports regarding the importance and the prognostic value of tertiary lymphoid structures, and in particular of CD20 positive cells inside the tumor mass, which are prognostic and positively prognostic. Inside the tumor, tertiary lymphoid structures, actually you can distinguish the plasma cells to the memory B cells.
Yeah.
So we know that there are some that are actively producing antibodies. What we don't know if those antibodies are important to promote the activation of the T cells, or they act to target directly epitopes on the surface. What we are trying to do is to check if among those antibodies we can find targeting antibodies.
Thank you very much. And Paola, I have just a small question for you. I mean, impressive. The project of generating a central data set to collect data from patients that are treated with checkpoint inhibitor is gigantically important. And I was wondering, there was a period last year where we were discussing with the Parker Institute to take their version of the REDCap that was adapted to collect especially data regarding the toxicity of checkpoint inhibitor. There was a follow-up on that. I mean,
I have never been in contact with the Parker Institute, so the problem is that it's not so easy to transfer from IFA to REDCap, but we are trying to ask to IFA to give us the possibilities. Otherwise, we have to enter by manual everything. So we hope.
Wow.
So we hope that IFA will be more sensitive to this, and we wrote also with Ruggero to try to understand, and then it would be more easy. But I never have contact with the Parker Institute.
So just in case, I mean, the project is so important, so anything you may need on the side of the IT support, if possible, from the other project is great. For the Parker Institute, if you think it might be important, the people that the president and the scientific director that we discussed with, that was 1 year ago. I mean, then, the COVID blocked everything as usually, but we can transfer to you the contacts in case you are interested. Let me know.
No, I think for us is really a great chance. So I would like to be sure that we have everything set. And I think that when we have everything and we have also the ability to put some minimal information on diet, vaccination, that I think is really important also in this period, and probably also COVID situation now, we have to speak with them because.
Okay, thank you.
Thank you.
Okay, thank you very much again, Paola. And I think we move on to our next speaker, who is Luca Boldrini, who will update us on the progress of the radiomics working group.
Good afternoon, everyone. I'm Luca Boldrini, and I'm Scientific Secretary of the Radiomics Working Group of Alliance Against Cancer. And we will together see the activity report for 2020 and the scientific plans for 2021 of the Radiomics Working Group. The working group was created in November 2019 and then later ratified in December. Then we moved to the identification of the members and the different team leaders for the different specialties about imaging and the technical aspects of radiomics. Then we started the scientific activity itself, submitting for the triennial project of the Ricerca Corrente Reti 2019, the 3 years project record, to the Coordinating Center Ethical Committee. The working group was then involved in the COVID-19 proposal from the Italian Ministry of Health, and the spontaneous research activity then was moved to the team leaders meeting in May in order to take advantage of the very nice response we had from the participating institutions and all the different aspects related to the joint efforts with the other working groups. In early June, the working group was also active with the ACC 2020 call for proposals, participating with very active involvement. The last but not least very important date to have in mind in this report timeline is that on the 1st of October 2020, the research fellow that is linked to the record project has started her activity. We hired an engineer, a biomedical engineer with a postdoc degree for image analysis. The working group numbers are pretty remarkable. We have 27 involved institutions and 103 participants, which are divided in the different specialties in the image sciences. We have, of course, radiologists, nuclear physicians, radiation oncologists, medical physicists, engineers, image scientists, and also medical oncologists. We also have a very nice gender balance with 45 female and 55% male members. This is the structures that we gave to the working group. The working group was divided into working teams, reflecting on the clinical side the existing working groups of Alleanza Contro il Cancro, while on the physics and technical teams, the most used technologies for radiomics analysis. As for the clinical working teams, we do have, of course, breast, which is coordinated by Antonella Petrillo of Pascale, Napoli, glioblastoma, Stefano Bastianello, Mondino, colon, Marta Scorsetti, Humanitas, melanoma, Tommaso Tartaglione of IDI, lung, Daniele Reggia of Candiolo, and then we have sarcoma with Antonino De Paoli from CRO of Aviano, radiogenomics and genomics links, Nicola Di Napoli, Gemelli, immunotherapy, Antonello Vidiri of Regina Elena, pathology and biobanking, Carlo Tacchetti of San Raffaele, and then ovarian cancer, Massimo Bellomi of Yale. Then we go straight to the physics and technical working teams, having CT, Tiziana Rancati of Istituto Tumori Milano, MRI, Roberto Sghedoni of Reggio Emilia, PET-CT, Francesca Botta, Yale, and Tesomics with Michele Avanzo of Aviano, and software research and development, Jacopo Lenkovic of Gemelli Hospital of Rome. So here we try to tackle all the possible dimensions of clinical cooperation and scientific and technical development for radiomics. Of course, this is the structure was given in order to enhance the relationship with the other working groups, making translational studies with radiomics even more powerful thanks to the joint cooperation with the other working groups. The triennial project record, radiomics in rectal cancer real-world data, based on the development and validation in a multicentric setting of radiomics models, was firstly submitted to the Coordinating Center Ethical Committee, which is Gemelli Hospital of Rome, in February 2020, and was given the last approval, administrative approval, in August 2020. Then the participant centers started approving the local ethics committees, and at the time being, we do have 4 approvals: Istituto Tumori Milano, Pascale, Humanitas, and Yale. And we have other centers still pending with the decisions of the ethical committee for San Raffaele, CRO, Meldola, Reggio Emilia, and Candiolo. Just to summarize very quickly the primary and secondary aims of this project, we do have as primary aim the setup of radiomics-based predictive models for progression-free survival at 2 and 5 years, metastasis-free survival and local regional control, always at 2 and 5 years, for patients affected by locally advanced rectal cancer. The secondary aims are the setup of radiomics-based predictive models for pathological complete response, radiogenomics models of RAS mutational status and the microsatellite instability, histological grading, overall survival, and 2 other more technical aspects, which are field strength independent validation, 1.5 versus 3 Tesla images will be used to this end, and the setup of an auto-segmentation model for rectal cancer based on neural networks. The reporting of the done activities can be divided into 4 main activities, which are activity number 1, protocol finalization and ethical committee authorization for coordinating center. Activity 2, the anonymization and test image uploading with dedicated reporting. activity 3, radiomics mode-DICOM platform optimization for the specific MRI analysis. And activity 4, the upload and segmentation of 30% of the cases of dedicated storage systems. As you can see, the deliverable were expected between month 6 and the year. And despite the situation and the hard times related to the COVID outbreak, we do have all the activities in time. So the scientific plan is the foreseen one. We do not have to make significant changes to the Gantt charts of the RECT study. And for 2021, we will have the first activity, the collection of all the ethical committee authorization for the participating centers, which we expect to finalize in month 3. The upload and segmentation of the remaining cases, we expect nearly 70% of them on the dedicated storage systems, expected for month 9. The upload and QA verification for month 9. And then we have activity 4 with radiomics feature extraction and preliminary data analysis for the primary endpoint modeling for the end of the year. And throughout the year, we're going to also have an evaluation for international centers' requests of collaboration. We had 3 centers asking for participation in this study, which are the University of Chengdu, China, the Madrid La Princesa Hospital in Spain, and Sydney University in Australia. So we hope to have also occasion to further increase the collaboration and the number of patients, which is currently expected between 1,000 and 1,200. Then we moved also onwards with the cooperation with other working groups, more specifically the rational study, the radiomics validation of the ALLEANZA CONTRO il CANCRO lung panel in non-small cell lung cancer, with close cooperation with the working groups of lung and genomics, was finally approved as an amendment to the original protocol in July 2020, and it is currently being approved by the local ethics committees of the 18 centers participating. Then we have primary aims, which are the development of radiogenomics model for the prediction of mutations and treatment response and overall survival prediction. So we have also clinical outcomes. Secondary aims, which are delta radiomics applications for the available datasets, the development of a clinical decision support system that can support the clinician in taking such complex decisions in the workflow of non-small cell lung cancer patients, and development of automatic segmentation algorithms for lung volumes, which will be, of course, executed using convolutional neural networks or other machine learning solutions. The working group then tried to also govern at best the spontaneous research, and we set up the DECIDE study, modelli radiomici multiparametrici nella predizione del profilo molecolare mammario, coordinated by Pascale, having as primary aims the development of breast characterization radiomics model on both MRI and mammograms, which is quite innovative, having the two different techniques together, histological grading-based radiomics classification of breast cancer, and as secondary aims, the breast parenchyma radiomics characterization, having a special focus on the glandular density and background signal, radiomics histotype characterization, as for luminal A, B, HER2, and triple negative kinds, radiomics characterization between MRI and mammograms of tumoral and peritumoral tissues. These studies that I listed are the currently active one, which required the ethical committee clearance, being studies that involve human beings. We do have also, of course, active protocols which do not directly involve humans, so were defined as technical protocols without the need to have specific ethical committee clearance, which are the CT FAN, MRI FAN, DOMSI, and image quality assessment. The first one is related to understand and govern uncertainties of CT radiomics features and is coordinated by Rancati and Inter Milano. The second one, MRI, coordinated by Schedoni and Reggio Emilia. And then we have the Zomics for Michele Avanza of CRO and the image quality assessment, the SDN, with Salvatore and Soricelli. We do also have two different studies, authorization pending from the ethical committees, for ovarian cancer, Massimo Bellomi, and glioblastoma, Bastianello. The two studies, of course, will be, of course, opened to all the other institutions as soon as we get the authorization for the coordinating centers ethical committees. Then we have some work in progress for the research and development validation of radiomics models in the setting of analysis and feature extraction platforms, coordinated by Lankovic of Gemelli, the PET-CT image analysis to predict the primary refractory and relapsing Hodgkin lymphoma at baseline, coordinated by Botta of Yale, and firstly started by Arturo Chiti, who was a former team leader of the PET-CT working team, and we thank you for having led the first moments of this study. Melanoma and immunotherapy have a joint project with radiomics markers for response prediction to immunotherapy in advanced melanoma, with Antonello Vidiri and Tommaso Tattaglione co-chairing this initiative. To be started with more specific protocols are pathological biobanking, radiomics prediction of metastasis occurrence and preclinical studies, Carlo Tacchetti, and the preoperative radiomics characterization of sarcomas for Antonino De Paoli. So doing an appraisal of the first year activity, we can underline that the working group has several strength points. The main ones are represented by the enthusiastic response from the institutions, with, I repeat, 103 members at the time being. The complementary competencies that have infused in such a high number of participants, both from a technical, clinical, and translational dimensions, and the inter-working group activities. I want to thank also the other secretariats for the working group of lung and genomics, for the very nice job we have done together in launching the radiomics ancillary spin-off study from the ACC lung protocol. The weak points are, of course, the fragmentation risk of subspecialty in terms both of clinical environment or technical aspects, the technical dishomogeneity of the different scanners that we have in our institution, but these represent also an occasion of very robust radiomics model setup, the storage and analysis platform activity and access, which was quite not very clear and straightforward at the very beginning of the working group activity, and we hope we'll then be able to make it more easy for all the attendees to have access to the storage, protected storage space, and of course the need of authorship rules, which are currently object of debate and of modulation, having such high number of involved people and specialties needed for this kind of analysis. The 2021 specific scientific roadmap mainly relies on empowering the existing research protocols for the Ricerca Corrente Reti 2019, and of course studying new ones. The facilitation of the storage and web service access for, of course, IT solutions, and also image exchanging and merging of multi-omics platforms. launching an internal radiomics-oriented newsletter that will take updated all the members of the working teams, and that I plan also to share with the other working groups. And of course, the revision and optimization of the working teams, trying to tune at best the clinical aspects and the technical innovations, and offering also to other pathologies which were not represented in the standard working groups of Alleanza Contro il Cancro, also the occasion to be object of studies and of more deep radiomics analysis. And having said that, I want to acknowledge, of course, the Alleanza Contro il Cancro board and staff for the very nice cooperation, the team leaders of the radiomics working group, who really are enthusiasts of their work, and they're doing their best in order to coordinate such a huge amount of people and different backgrounds. Once more, Arturo Chiti for the first steps as PET-CT team leader, Vanessa Gregorch and Luca Mazzarella for the activity of joint working group leaders in our study about lung cancer radiomics, and then Philippe Lambin, Pablo Rodriguez Carnero, Lucia Orlandini, Inji Lang, and Annette Edwards for the international dimension, of course, of our working group. Having said that, I'm happy to answer to all your doubts and indications. You have my email there, and I thank you for your kind attention. Thank you once more.
Thank you very much, Luca, for the very good presentation and for the excellent work you have done so far, actually, and the punctuality in meeting your deadlines. And it's a very ambitious program that you presented, so congratulations again. Are there questions from the audience or from the panel? Comments, questions for Luca? No? Okay, so we do have a general discussion session at the end of the session, so perhaps if there are questions, you can keep them until then, and we are running late. So if there are no questions, I will go on to the next speaker, who is Giulia Buzzati from San Martino Hospital, who is sitting in for Lucia Del Mastro and will tell us the progress of the breast cancer working group. Thank you very much.
Good morning, and thanks to the organization for this opportunity. On behalf of Professor Del Mastro, the secretary of our group, I will give you the update of the activities that are currently ongoing on the breast cancer working group. Breast cancer is the most common malignancy in women, and in Italy, an estimated 55,000 new cases of breast cancer are expected to be diagnosed in 2020. The secretary of our group is Professor Del Mastro, the clinical coordinator, Professor Bianchini, and the preclinical coordinator, Professor Daidone. Thanks to this network, almost 86,000 and almost 10,000 patients per year are estimated to receive surgery in these centers. This is based on a survey of 2016. Today, I show you the ongoing project of our group. First of all, the NeoGene trial, which is a genomic test aiming to identify actionable mutations in hormone receptor negative, HER2 positive, or triple negative breast cancer resistant to neoadjuvant therapy. The coordinator site is Ospedale Policlinico San Martino. The study coordinator is Professor Del Mastro, and 15 centers are involved in the study. Is it currently known since Cortazar meta-analysis published in 2014 that the patients who did not achieve a pathological complete response have the poorest outcome? These occur especially in patients with aggressive breast cancers, subtypes like triple negative breast cancer and hormone receptor negative HER2 positive breast cancer. Thus, in these settings, it's crucial to identify and validate biomarker associated to treatment resistance. In metastatic breast cancer, thanks to the molecular profiling of cancer cell, potentially actionable mutations have been identified, and the ESMO have developed a clinical actionability scale for the molecular targets. Moving to the early settings, in 2013, Balco et al. Profiled the residual disease remaining after neoadjuvant chemotherapy of triple-negative breast cancers. And they identify that with higher frequency alteration on MCL1, 10, and JAK2. In a recent study, Di Cosimo et al. Characterized 19 patients for somatic mutations in primary tumor biopsy and residual disease surgery. The most common mutated gene was TP53, and no recurrent mutation significantly associated with pathological complete response. In a residual disease, more common alteration were involved of the cell cycle progression, PI3K, AKT, mTOR pathway, and the EGFR TKI resistant pathways. PI3K was the most altered gene in residual disease. In the NeoGene trial, we enrolled patients with the locally advanced triple-negative breast cancer or HER2-positive hormone receptor-negative breast cancer undergoing neoadjuvant chemotherapy, and that are treated at Alliance Against Cancer Breast Cancer Working Group. The patients, during their neoadjuvant treatment, blood samples are prospectively collected, and in the patient who did not achieve a pathological complete response after surgery and enrolled in the retrospective project. As per Amendment 1 in 2019, also the patient treated between 2016 and 2019, for which are available tissue sample pre and post chemotherapy, can be enrolled in the retrospective project. Primary objective of our trial is to analyze ctDNA, the presence of circulating microRNA on blood sample collected before, during, and after treatment, and to compare tumor infiltrating lymphocytes on primary and residual tumor specimen. The objective of the retrospective genomic project are to perform a genomic analysis and compare mutational status of primary and residual tumor tissue, and to identify specific mutations potentially actionable in residual tumor disease post-neoadjuvant therapy. In the retrospective project, the genomic test on residual disease will be performed on primary and on residual disease will be performed for the first 100 patients with the Foundation Medicine profile, and for the further 100 patients with the Alliance Against Cancer Gerson panel. The first interim analysis is expected at the end of the accrual of the first 100 patients. The current status is that 12 sites are currently open and 7 are currently enrolling patients. 2 sites are waiting for opening, 1 site is in standby for internal reason, and 1 site will never open. Actually, 52 patients are enrolled in the prospective project, and 15 patients are enrolled in the retrospective project. We have seen that there is a very slow recruitment, and that's why we have conducted on July 2020 a survey to better understand the reason of this slow recruitment, a part of the SARS-CoV-2 infection. And from the 7 centers who replied to the survey, we have realized that at least for the retrospective project, so the patients that have been treated between 2016 and 2019, almost 89 patients can be enrolled, thus supporting a future faster recruitment. The second project is the follow-up trial. The follow-up trial aims to understand the impact of intensive follow-up and liquid biopsy on diagnostic anticipation in high-risk stage 1-3 breast cancer patients. It's a randomized multi-center phase 2 trial. The coordinator site is Ospedale Policlinico San Martino, and the study coordinator, Professor Lucia Del Mastro. We know that the annual hazard of recurrence peaks in the second year after diagnosis and is higher in patients with node-positive disease than patients with node-negative, and especially in hormone receptor-negative patients. Current guidelines advise against routine surveillance apart from physical examination and mammography, regardless of the risk of relapse. However, these recommendations are based on 2 randomized studies completed almost 20 years ago, and considering the new, more sensitive tests and more active treatments currently available for the metastatic disease, the role of follow-up in high-risk patients needs to be re-evaluated. Moreover, recent data support the detection of circulating tumor DNA in the blood plasma may be a potential non-invasive approach for disease surveillance in order to identify preclinical metastases. In a recent paper published by Garcia Morillas, primary tumor from 101 patients was sequenced to identify somatic mutation, and personalized tumor-specific digital PCR were used to monitor this mutation in serial plasma taken during the follow-up. The detection of ctDNA was associated with a relapse in all breast cancer patients, and the median lead time was 10.7 months over clinical relapse. This is a multi-center, open-label, randomized comparative phase 2 trial. Patients at high risk, defined as patients with 4 or more positive lymph nodes, with tumor size of 5 centimeters or greater, patients with triple-negative breast cancer or HER2 positive hormone receptor negative breast cancer who did not achieve a pathological complete response, or patients with a risk of relapse after adjuvant treatment estimated more than 25% can be enrolled in this trial. Patients will be randomized 1-to-1 to intensive follow-up, where total body scan is added to standard follow-up. Liquid biopsy performed every 6 months is also done in every patient enrolled in the trial. Primary objective of the trial are to evaluate if the intensive follow-up can anticipate the diagnosis of distant metastases or breast cancer local regional relapse compared to the usual follow-up practice. Secondary objectives are to evaluate the false positive rate, the rate of interval relapse, the rate of resectable or unresectable relapses in the intensive follow-up compared to the standard follow-up practice, to explore the impact of liquid biopsy on diagnostic anticipations, and the difference in terms of quantitative ctDNA at relapse, compared to matched non-relapse patients. Also, a translational ancillary study will be performed to analyze ctDNA detected gene variants, to characterize circulating microRNA, to characterize cancer-derived extracellular vesicles, and to analyze the metabolomic profile. What is the current status? This year we have prepared the protocol, we have written the standard operating procedure and identified the CRO. And currently the protocol is under submission to the AP committee of the coordinator sites. The last project I will talk about is the STRA-RNA, which is the PI is Professor Enzo Medico, and the coordinator site is the Istituto Candiolo in Turin. The STRA-RNA is focused on the clinical deployment of RNA-seq for molecular stratification of breast cancer. Multigene expression assays have a validated prognostic utility in hormone receptor positive, HER2 negative early stage breast carcinoma, where adjuvant chemotherapy could be spared in approximately 25% of patients. However, the most widely used tests like Oncotype DX, MammaPrint, and Proxima are based on different methodologies, measure different genes, and have been validated in different patients' cohort. Thus have limited the prognostic concordance on the same patients. Global RNA sequencing analysis measure all expressed genes for a reasonable cost, which could be below €500, and that is expected to further decrease in future. In principle, all existing transcriptional classifiers can be reconstructed by RNA-seq. Global RNA-seq profiling would allow to apply multiple classifiers to the same sample for comparative analysis and most robust prediction, and the development of novel integrative classifiers. What has been already done, so the RNA extraction and RNA sequencing protocol optimization, finding the Illumina protocol as the best protocol. Second, a larger scale RNA sequencing profiling of retrospective breast cancer samples already classified with a multigene prognostic assay. 71 cases have been profiled with available MammaPrint classification, and 51 cases with available Oncotype DX classification have currently been processed. Third, the emulation of multigene prognostic classifiers and the evaluation of their performance using PAM50, MammaPrint, and Oncotype DX have been emulated. Moreover, it's been done the reconstruction of existing classifiers and the development of new methods, like the machine learning methods, which has led to this publication this year. Considering the development of a machine learning approach, we should know that it is not clear whether emulation with RNA sequencing or the classifier originally developed using PCR or microarrays or reconstruction through machine learning is preferable. Hence, they have focused on the robustness and portability of PAM50 to identify 5 breast cancer intrinsic subtypes. And then they have explored 5 supervised learners to reconstruct a robust single sample PAM50 colors via RNA-seq. Looking at this imaging, for the purpose of this work, has been used RNA sequencing profiling from 4 published RNA-seq database for a total of almost more than 4,700 breast cancer samples. They have explored 4 different ways of emulating PAM50 and 5 different machine learning approaches. They have selected the best machine learning strategies based on concordance with available PAM50 classes, and then compared emulated and reconstructed classification with the published ones, also considering prognostic robustness for dataset in which disease-free survival and overall survival data were available. Machine learned-based classification reached 90% concordance with PAM50 without the need of our reference samples, and machine learning-based classification improved prognostic performances and is therefore a valuable single sample method to strengthen breast cancer subtyping. Which are the next steps expected for the next year? The RNA-seq of additional MammaPrint sample, the RNA sequencing of Oncotype DX samples that already started, the training and validation of MammaPrint and Oncotype DX RNA-seq classifiers that already started, and comparative analysis and emulation of additional classifiers. And in my conclusion, I really want to thank all the clinicians and pre-clinicians that are part of the family of the Breast Cancer Working Group. Thanks for your attention.
Okay, thank you very much. Giulia, that was a very good and clear summary of the progress of the breast cancer group. Are there questions from the audience or questions or comments from the panel? Pier Giuseppe?
Yeah, I have a question. I don't know if Julia is connected.
Yes, I'm here. Can you hear me?
Hi, Julia. I have a question for you and Enzo, I guess, regarding the RNA-seq part. It's more a curiosity. I mean, the statistical power which is needed to benchmark RNA-seq against the known classifiers is much different than doing the same machine learning approach, for example, on retrospective cases with known clinical output.
Okay.
Go through the classifiers.
Yes,
so different.
I have understood. However, I would say that this is the Enzo Medico project. Yeah. I don't know if Enzo is connected as he followed the trial, so he can answer you, of course, better than me, that I currently don't know exactly the data. But I don't know if Enzo is connected.
If not, we discuss it in the panel discussion at the end.
Okay.
Thank you for your presentation, Julia.
Okay, there's a quick question from Luca.
Yes, ciao Giulia. Great. I mean, really nice. I really like the follow-up trial. I was just wondering which liquid biopsy tool are you actually going to use in that context? And please, can I note that, I don't know if you followed this morning conversation, but we have repeatedly discussed the liquid biopsy as a main priority of the consortium for a number of projects. So it may be great to integrate, if it's not particularly time and sample consuming, also other goals of the other projects within your framework.
Okay, I'm sorry I couldn't follow the intervention this morning, but Barbara, which is with me, she followed your speech early this morning. So she will give me a feedback about. And clearly, I think that this project is still under submission. And if we can have your, I can see your feedback about, I can kindly send you also and see with you if there is any improvement that we can do the best. Lorenzo Geratano and Fabio Puglisi from Aviano, they helped us in this part as they will be the center which will perform the analysis. So if you want, we can see with you if you agree with our what we have already done and see if it is a part to improve the liquid biopsy part.
Okay, so we have a question from Giovanni Martinelli who asks, Giulia, could we use the data for fusion transcripts?
What is that about for the RNA-seq trial? Yes, I think exactly. And I'm sorry about this as well. That this is the Enzo Medico project, as I told you.
Okay, well then maybe we can discuss it at the end.
We discuss in the general discussion at the end after Enzo Medico presentation.
Perfect. Yes. Okay. Okay, great. Thank you very much, Giulia. We proceed to the next speaker, who is Gaetano Finocchiaro, who will update us on the progress of the glioblastoma working group.
So, hello everybody. Thanks for allowing me to present data from the working group glioblastoma. Our main project this year, that was approved this year, is about liquid biopsy. Essentially mostly on cerebrospinal fluid, but also from plasma. Here you can see the institutions that are involved at the clinical level: Humanitas, Vesta, Bambino Gesù, and quite soon Regina Elena. At the level of nucleic acid extraction and quality control, Candiolo, Vesta, Istituto Europeo di Oncologia, e for sample analysis, ancora Candiolo, l'Istituto Oncologico Veneto, il Vesta e l'Istituto Europeo di Oncologia per quanto will be involved about microvesicles. Before I go into the presentation about liquid biopsy, let me tell you something about the previous projects that are still ongoing or essentially closed. The first one is about sequencing of more than 100 glioblastoma-derived cell lines with whole exome sequencing results. What is interesting here is in particular the fact that we included 16 pair samples that were sequenced and additional sample from recurrent glioblastoma from Besta Istituto Superiore di Sanità. And in one case in which we detected the V600E mutation of the BRAF gene, an in vitro treatment with inhibitors is ongoing. A paper is in preparation and Stefano Dracula is taking care of that. So having said this, the other project of the previous 3 years is the one about long-term survivors, that is patients with glioblastoma surviving more than 5 years. Here, an important goal is the connection, to find a connection between the neuropathology and molecular data and immunological data and clinical evaluations, which include the monitoring of quality of life of a live patient and their caregivers 5 years after the first diagnosis and later on with 2 time points at 6 and 9 months after the baseline. The number of patients involved in this study is sizable. 105 were screened and 86 enrolled, with a median survival time of 82 months, so much longer than 5 years. And the ongoing activities at this point are the neuropsychological and quality of life evaluation at time 1, which is okay for 13 patients, and only 1 patient so far, because of the COVID interruptions, has been tested at time 2. Immunohistochemistry evaluation and the study of immune infiltrate is in progress, as it is also in progress the sequencing of available specimen with a 400 gene panel at Istituto Oncologico Veneto, GSIMP analysis, methylation profiling at OPBG in Rome, mRNA immune signature profiling at ERST at Mendola, and traditional, let's say, markers of glioblastoma that are evaluated at Besta. Now going to the present part of the project, the one related to the liquid biopsy. Let me start with the contribution of the Istituto Clinico Humanitas, who drafted the protocol that was the starting point to prepare an updated protocol that was approved by the ethics committee at Besta on July. So far, Humanitas collected 14 cerebrospinal fluid samples, and more samples will also provided by Istituto Oncologico Regina Elena in Rome, that is joining the project and will contribute plasma, CSF, and tissues from glioblastoma patients. One minor issue here is the way we should present the protocol that has been approved at Besta to other institutions, whether this has to be done by scientists at relative institution, or whether the presentation should be centralized by ACC. That we will find out soon. The idea of the study design is that we have samples that is a fresh tumor or a formally fixed paraffin embedded tumor, from which we may extract the DNA and RNA. We have CSF that will be centrifuged essentially from spinal tap. I mean, subarachnoid CSF was used in the early phases just as a proof of principle, but clearly from the clinical standpoint, what we need to have is information from spinal tap, and there we will get DNA from CSF, and also we will have blood, which is very important to get data on constitutive DNA, but also relevant to see whether microvesicles isolated from peripheral blood may contain tumor or tumor DNA or tumor RNA, which is an ongoing part of the story. Last year, before starting, the kickoff meeting of this project was early February here in Milano Pesta. Before that, a number of samples, mostly from subarachnoid space, were obtained at Humanitas and studied at Candiolo as well as at the IOV in Padua. And the CSF DNA were available in 13 cases. This year, the number of cases that were piled up by these three institutions, which is Humanitas, Candiolo, and IOV, is of 13, and their investigation on these specimens are ongoing. At Pesta, we have collected 11 lumbar CSF. 6 were deriving from patients that had surgery for brain tumor. So we had both CSF and tumor DNA, and we will call this CSF-T. In 5 cases, the CSF was obtained when brain biopsy was not possible because too risky, or were already performed but was not informative. So 10 of these CSF were analyzed by NGS with a small panel of 28 genes covering 231 kb on the ION PGM instrument. In one case, we only performed digital PCR and Sanger sequencing. This was patient 7. And you can see here result of Sanger sequencing in which you can see a mutant nucleotide, which suggests that the mid88L265P mutation is present. This present is a kind of signature for peripheral central nervous system lymphomas, and was confirmed by digital PCR. So in this case, we could make a diagnosis and start treating the patient. This is the sequence of genes that are represented in the panel. As I said, 28 glioma genes. And the coverage we had in the 10 patients examined was 480X for the tumor and 954X for the cerebrospinal fluid. Here are the results. In the 6 cases in which CSF plus tumor and blood, of course, was available, we had one patient that was not informative. We could not find mutation, but we are revising. We are rechecking this, and we will find out. The patient had multiple meningiomas, so it was quite likely an NF2 patient. In 2 cases, we could find same tumor variants in CSF and tumor, although in one of the 2, the frequency in CSF was quite low. In patient 6, we had the same tumor variants in tumor and CSF, but a new variant in CSF, which was the BRAF V600E mutation. Patient 3 was interesting. In this case, he had surgery at recurrence, but the recurrence, was at histology. It was obvious that the majority of cells were macrophages and other kind of immune cells, because this patient had an inflammatory reaction after her surgery. So what we found here is in the CSF and not in the tumor was the IDH1R132L mutation. Patient 4, in patient 4, we had an interesting finding, a mutation of the SMARCB1 gene, which is, according to the program we checked, could be pathogenic. SMARCB1 represses cyclin D1, and there are very recent data of this year suggesting that it might be involved also in the development of glioblastoma, although this is a very specific situation in familiar cases of glioblastoma. So this can be interesting, but it has to be double checked again. So in the 4 cases in which we only had CSF, we had in one patient, patient 10, mutations that are quite frequent in gliomas. In one patient, just the R132L mutation, and it's in red here because we found it also in this other case. This is a very rare variant, and so we have to make sure about its origin. Another patient had a mutation of the IDH2, which has never been described in gliomas. So it also had mild positivity that has to be double-checked, possibly by a second round of digital PCR of the MYD88 gene. So it is possible that this patient has a lymphoma. This is coherent with the MRI situation. And finally, we have this mutation of patient 9 that again seemed to be typical of gliomas. Overall, we can say that in this small panel, we, in that sequence, can be information sometimes unexpected. Overall, at this point, we can say that we got data in 10 of 11 the patients examined. We have to double check about the possible artifacts that might be also favored by the high coverage we have been used. And we believe that the use of unique molecular identifiers, UMI, might help to dissect this and make sure that what we are seeing is really present in the tumor and not just the consequence of PCR. Let me switch now to the contribution of the pediatric hospital Bambin Gesù in Rome. As you may know, pediatric gliomas are significantly different, genetically different from adult gliomas. The possibility to identify mutations is very important because in most cases the tumor are in the brainstem, so surgery and even biopsies are very difficult. So if we have data from CSF, that would be great. Overall, these are the patients that provided CSF. There is 21 patients, although the number of CSF samples is higher because longitudinal follow-up was performed. And in the majority of cases, the CSF derives from the lumbar puncture. There were limitations in obtaining information from this specimen, possibly because the maximum amount available was 500 microliters. So 100 CSF. So one possibility is to investigate microRNA in this patient, as this microRNA can be informative. They are stabler than other molecules, and so they could be important to get more information. But I mean, this is very preliminary at this point. As I mentioned at the beginning, there is also the effort to verify whether in plasma we can find extracellular vesicles deriving from the tumor containing tumor DNA or RNA. So the extracellular vesicles in vitro, starting from 2 GBM cancer stem cells lines, this mutation in the vesicles could be detected. But in the 5 patients analyzed so far, the amount of DNA in extracellular vesicle was extremely low, and no detection of mutation was possible. So the ideas here from the group at Yale, led by Giuliana Pellicci, to improve this are related first to the use of larger amounts of plasma, and second to the use of size exclusion chromatography rather than differential ultracentrifugation to obtain amounts of extracellular vesicles, and also to the use of digital PCR, which is extremely sensitive, as you may know. So a new cohort of GBM plasma sample is available, 12 patients for this analysis. As I said, the tools, the hardware for this is now present at Yale for size exclusion chromatography and also for quantification of the extracellular vesicles. Of course, this is very, very important because we would all love to have information from peripheral blood rather than CSF. I guess I can close here. I thank you for your attention, and I will be happy to address issues or questions that may arise from the audience. Thank you.
Okay, thank you. Thank you very much, Gaetano, for your update. So are there questions? Let's see if there are questions from the audience.
I have a question.
Okay.
Data on the liquid biopsy, the spinal fluid, are really exciting, Gaetano. I have a very technical question. Regardless of concordance and discordance, all the variants that you described were somatic variants, right? Were called against the putative peripheral blood germline DNA.
Yes, they were not present in germinal.
Okay, so no matter what, in the case where there is no concordance, those reflect clonal expansion, no? Gaetano, non ti sentiamo.
You are muted, Gaetano.
That's also my guess. In particular, the R132L
Right.
mutation is very interesting because it seems to be catalytically even more active than the most frequent mutation. And so we will triple check. Rosina is here with me and she is very careful. But I doubt this is a PCR artifact. I rather think that this is reflecting something that we cannot see in the tumor. Also because the CSF compared to blood is very specific. I mean, it's very clean there.
Yeah, and the other comment, Gaetano, I totally agree with you based on our experience. I mean, 1 UMI, even 2, are very useful to ensure the consideration of PCR added late during the cycle mutation.
Can't hear you very well.
I was saying that the idea of using 1 or even 2 UMIs.
Oh, the UMI.
Yeah, it is very good. I mean, they work very nicely. I mean,
yeah, I think that's the first thing we will check.
Absolutely. Thanks.
I have a question. Can I ask, Miriam?
Yes, of course, John.
Yeah, very nice findings, Gaetano. So I have one, more medical question. So when you find variants in CSF, which are not in tumor, actionable variants, like if I understood correctly, you had one patient in which you found a V600E mutation in BRAF. So how do you react? So would that, at that point, that mutation instruct, say, for a molecular tumor board decision to give BRAF and MEK inhibitors to this patient?
You know, the ideal scenario would be to have something to double check on that. For example, in case of IDH, you could check on hydroxyglutarate level in peripheral blood, because you know, as pathologists can tell you easily, the amount of tissue that they analyze is very minor. Most of the surgery these days for brain tumors is using ultrasonic aspirators. So it might very well be that what the pathologists cannot see, we can see it in CSF. Of course, before starting a treatment, we will need some confirmation.
And what type of confirmation if there is no tissue available at that point? So would you risk making a therapy off-label in this case, in the absence of additional information?
In case of the lymphoma, I show you the one identified by digital PCR. We treated the patients because that mutation were very straightforward and very typical. In case of the BRAF, it would be difficult. We should think of ways to maybe double check in the peripheral blood to see whether we could find that, for instance.
Okay, thank you very much. Are there further questions or comments? Let me just double check. No. Okay, thank you very much, Gaetano. I think we can move on to the next speaker, who is Giandomenico Russo, who will update us on the progress of the melanoma working group. Hello, Gianni. You have 15 minutes plus 5 of discussion.
Okay, thank you. Can you hear me?
Yes.
Okay, so the working group of melanoma is working on organoids recently. And I will present some of the results that we have obtained during these years, during this last year, were actually very few because of the COVID story, okay, who has infatuated us really a lot from many points of view. However, at least partially in each institute have been performed some progress, either in terms of biobanks accrual or in terms of setup of the conditions for organoids. Let me start with the people who is inside this group, because this group was born at the first and the project of the ACC lung to use in melanoma, and then has switched also to organoids, and so has seen many actors and who have actually participated in different way to the group. So the work that I am presenting, the results I am presenting today, they regard mostly organoids, and will have a very, very small part on the other two projects we have been doing. So the idea was to apply to model disease by PDOTs, to make biobanking, to make then analysis for genome, transcriptome, proteome, any omics that we can have, even in single cell, as well as then to use these PDOTs for drug testing. This is quite general wish from everybody, and this disease modeling is, as we know, is important, but is also difficult to obtain. So when we decided to keep a different approach, also to maintain, not to force anybody inside groups to have one single approach to the technique. There were groups who were working already on PDOTs. So we decided to have everybody working with their techniques and then to compare these techniques. Things that we would have liked to have done, or things that we will do probably in the second year, COVID permitting. So if we take a piece of tumor and we make small fragments of it by 70, 100 microns, we can use this on collagen beads or new hydrogel, like people is been doing at Yale mostly, and Luisa Lanfrancone and Fiorenza Lotti have worked a lot on this, and I will show you some of the results that we have obtained, that they have obtained recently. Has been, and also at Istituto Nazionale Tumori of Bari, where new hydrogel matrix have been used by Letizia Porcelli and Amalia Zerita. But if you then dissociate yourself to single cell suspension, in this first approach, you can also use microfluidic system, who have been our first choice, but then have been abandoned because of the difficulties of finding really microfluidic system were actually available. We had an agreement at the beginning with Politecnico di Milano, but then it didn't work out, all the things. So we have almost abandoned this. And the second thing is here, as you can see, is if you dissociate cells, and then you can use those dissociated cells on dermal scaffold, then you can also generate PDOTs. And that has been the work done by Marcella Tazzari at Irst in Meldola in the things. But then if you take big punch of the tumor, then another approach is then to use a bioreactor, and this is approach has been used by at Istituto Nazionale Tumori di Milano by Monica Rodolfo and Licia Rivoltini. And so on each one of these we have done several progress. Each group have done several progress. The other things that we have done is, first of all, so let's start with the first, that is the 3D culture in collagen beads. We have designed the protocol, which is available to all the centers who have participated. The protocol is actually on the server that is running for ACC and is available. The wish was to also to make a video of this protocol, who COVID has impeded us to do completely. We couldn't move, our lab were closed, and so all were open in time in which we, anyway, I don't need to go ahead on this. But the protocols are there, have been diffused to several institutes. This was one of the goal of the network goal that I think is very important in this working group. So this allows us to have chosen reagents that and to have tested reagents and panels. So to make immunocell markers, or to make also tumor markers as a melanoma, and then to have and then to use them the PDOTs for treatments, the cell recovery for FACS, immunopanel, and so on. So I told you that the microfluidic system has been abandoned. So as these are the results who have been done mostly at Yale by Luisa and Fiorenza Lotti. So this definitive protocol, the recovering and freezer condition of the tumor material were also standardized. So we have now 5 markers that we use currently, that are CD45, CD3, CD4, CD8, and pan-mel on fresh tissue to verify the expression levels. And we do confirm that that we have a good expression levels on these P dots in during also after the freezing process, who is a very important technical question. This analysis who has been conducted only on 4 melanoma as actually will be extended to other 4 samples and the shared protocol. So the comparison that were made by mostly at the EO and ED in 8 patients between the lymphocyte profile obtained from the FACS analysis on organoids and that from the pathological analysis in order to assess the accuracy. And then we have also generated PDX from some of these tumors. At the EDID in Rome, we have continued during this period the accrual of biobanking, and now we have a total of 21 metastases of melanoma, and we have also tested the shipment of this, and we have this max tissue storage solution, which is a very it's a bit expensive, but it's a very good solution to work on it. So OTS will be also set in our institute. IFO and Pascal in Napoli have just started to work on this, acquiring the necessary materials to do this, and also some instrumentation was acquired at IFO. I told you that at the INT in Milan, they are using these 3D cultures they make generational melanoma with this bioreactor. So here is illustrated what they do. So they cultivated this ex vivo culture of tissue explants from 3 mm biopsy puncher. Don't ask me the detail, but of course are available from Monica and Licia. And after 72 hours of treatment, supernatants and tissue explants can be analyzed. So their results were obtained on 17 melanoma biopsies, and they can keep in culture for 3 days, preserving structural characteristics of the tissue and the vitality of the tumor cells. And these PDOTs obtained in this way will allow the evaluation of the effect of drugs and so on. So right now there is a biobank of PBMC biopsies that has been generated that includes also samples from 31 patients or in progression therapy with BRAF, MEK, and so on, who have been analyzed with different things. Another nice way was done by Marcella Tazzari at Erst in Meldor, was this one of generative of patient male melanoma organotypic culture using dermal scaffold. So at this point, you do single cell, and then you put on dermal scaffold, and then you generated this different type of organoids. So in 2020, another 7 samples have been analyzed in this way. And to show you the results they have obtained is that, first of all, these samples can have a very good vitality on when you use, for example, those solutions also for shipment, like the Max solution from Milteni. You have also they have also generated protocol for. The protocol, of course, is available. They have also set the condition for the cryopreservation of these cells, works quite well. The vitality of both CD4 and CD45 over the culture, as you can see here, it's very good. And it's even better, at least in 2 experiments done on what Marcella was able to obtain on collagen beads. By having a 74% vitality after, this is an experiment done only on two samples, must be repeated, but comparing to what she has obtained by freezing and thawing collagen beads, you can see there is a nice increase of percentage of light cells at day 3. And as you can see also, when you from this immunohistochemistry analysis, this tumor looks very nice with a reconstituted dermal scaffold over here. So what's next also now is to pass to single cell analysis from them, to go on also on multiplex immunohistochemistry, and then start what is really the second part of our project, with treatment. We have another group in Bari, okay, who is using a new hydrogel with structural flexibility and control the gelling by applying, for example, external stimulus like temperature. Also make hanging drops and matrix gel in this way. And they have obtained also some results on 8 biopsies of metastatic melanoma, in which after 6 days of culture, comparing to start, you, however, see still a reduction of the immunocomponent. They are also looking at the new ketosine, I don't know how to say ketosine, I think, based hydrogel, and also to the creation of the biobank. We had a project before of bioinformatic analysis who are being completed after, as you know, we were left at the end of the project by our very good bioinformatician in Candiolo, and then Gennaro has suggested to have the data looked by Matteo Pallocca. We have shared with him, and he said to me this morning that together with Enrico Berrino, always at Candiolo, has generated the data. They will be ready by the beginning of December. And then we have presented the project for the germ of melanoma, who has been actually taught and proposed by Paola Ghiorso and Stefania Datri. Paola is a person who is very expert also at the IMI, at the Intergroup of Melanoma, Italian Intergroup of Melanoma, on familial melanoma. So since the use of the germ panel would be not really competitive in our opinion on the diagnosis or on on the diagnosis of the predisposition to melanoma, to familial melanoma, we have decided to conduct a mutational analysis on tumor DNA germline in patients with metastatic melanoma derived with a personal and family history, to fully use the ability of the germ panel, who has a lot of information on actionability. And so this has been presented to ACC and is pending to start. And also, we have also designed a custom Thermo Fisher panel for the mutation of at least 30 genes to share also with IMI together with the Paola Ghiorso. I'm finished here. I'm ready for questions, and I apologize not to be present to the general discussion because I have to leave. I have some important stuff to finish because we have quite an outbreak of COVID here.
Okay, thank you very much, Gianni, and thank you for making the effort to be here with us tonight. Are there comments or questions from the panel?
I do have a question for Gian Domenico.
Okay.
Okay, so actually I have several questions, but I will limit only to one, which is more a suggestion than a real question. I think that some of the projects that you have outlined, especially the one which is now projected in the last slide, might be useful to suggest to the Italian melanoma, to IMI, the group, because they support several activities in this sense, and especially for familial melanoma, they might be interested in this project, and we can get additional extra funding to run this project through the involvement of additional centers in Italy.
Absolutely, that's a very nice suggestion, and yeah, it's the idea at the beginning. I don't know if you remember, but I co-opted the past president of the IMI inside the group.
Yeah, but Giuseppe Palmieri, the current president, is very open to this kind of.
Yes, exactly. Paola, it's our link to them. Yes.
I can also act as a link. I am also an IMI member, so.
Oh yeah, okay, yeah, sure.
And
of course.
And Virginia Ferraresi is in the board, so.
Yes. Okay, that's good. That's absolutely. That is absolutely important, because I think it's usable in that we have focused on that idea to use on metastatic melanoma to see if there are also differences with the other sporadic melanoma in the familial part. If we can use the actionability, the potential of the gerson, no?
I have a question, Gian Domenico. I'm impressed by the effort you are doing with the 3D cultures of melanoma biopsies or cells. Where do you want to bring the project, Gian Domenico?
To the testing of pharmacological compounds.
But what is the average length of cultures? How long does it take?
We have several. We have been able to do also 7 days.
Sorry, I didn't hear, Gian Domenico.
I say we have been able to do even 7 days, but Luisa can be much more precise on this than I can do. Okay, so but I have the idea now is to have at least we have reached 7 days, even 10 in some cases. Okay.
But listen, the very last.
The idea, of course, of using also animal models and growing them in animal models could be another,
No, yes, that is clear. But I mean, the 3D short-term cultures are really powerful if they're representative, and this goes without saying. No, but if I understood correctly, but in one case where you addressed it. I'm not sure I understood whether or not the immune infiltrate is stable over days. In one case, you clearly said that was not. In the others, was not tested, or
No, I don't think it was tested yet. Yeah.
Okay. Thank you very much. Very nice project.
Thanks, guys. Sorry for leaving. I give you a big hug from me, and please, I apologize for this.
Thank you, Gianni.
Ciao.
I understand. Okay, so we go on with the program. The next speaker is Roberta Maestro, who will tell us about the progress of the sarcoma working group. Roberta?
The project that the sarcoma working group has been working on the last months is related to the setup of a collaborative platform that has the aim to maximize the efficacy of clinical and translational studies on sarcomas. Just a couple of words on sarcoma to frame our project. Sarcomas are rare tumors. They account for about 1% of all tumors in the adult and are slightly more common in children. They are highly heterogeneous. The most recent WHO classifications lists over 70 different tumor histotypes that correspond to over 70 different biologically and clinically distinct entities. Sarcomas are quite aggressive tumors. The overall survival is about 60%, but it drops dramatically in advanced and metastatic forms. The fact of being rare and highly heterogeneous, of course, has an impact on the clinical management of sarcoma patients. First of all, diagnosis is challenging. The diagnosis of sarcoma requires a trained eye for a sarcoma to be recognized. And so it has been calculated that about 1/3 of sarcoma diagnosis done by pathologists that are not experts in sarcoma fields are either wrong or inaccurate, with of course impact on the therapeutic choices. Prognosis. The clinical outcome of a sarcoma is hardly predictable because the conventional clinical prognostic criteria that work well in other sarcoma types, tumor types do not perform well in sarcomas. And finally, therapy. The therapeutic portfolio for sarcomas is limited. And the fact of being rare and highly heterogeneous complicate the conduction of robust clinical trials for sarcomas, which, by the way, are also poorly appealing for the big pharma, being rare and heterogeneous. So there are a number of unmet clinical needs that need to be addressed. The availability of more accurate and effective diagnostic and prognostic parameters, and of course, new therapeutic options that may be tailored for the diverse histological subtypes. So bearing this in mind, we thought that for properly tackle these issues, we needed to have basic scientists, pathologists, and clinicians working side by side on the same project and for the same goals. So we established a strong interaction with the Italian Sarcoma Group to set up an integrated platform to maximize the efficacy of clinical and translational studies on these tumor types, with a progressive alignment or laboratory investigation with the ISG clinical studies. So with this aim, we identify three major trajectories: diagnostic, prognostic, and bioclinical. The first trajectory is essentially the natural follow-up of the PROVEST project carried out by the previous triennium sarcoma working group. And it deals with the setup of next-generation sequencing-based approaches for the identification of pathognomonic fusion transcript in sarcomas. We know that about 1/3 of sarcomas express a fusion transcript, and these have diagnostic, prognostic, and predictive values. We know that in most labs, FISH or real-time PCR-based approaches are used to identify these fusions, but these are essentially reflex tests that are aimed to confirm or reject a priori hypothesis developed by the pathologist. So we thought that nowadays diagnostic needed more comprehensive and unbiased approaches. And so during the previous triennium, we compared commercially available NGS panels focused on fusion transcript to assess their implementability in routine diagnostic for sarcomas. And the result of these studies have been recently published in Frontiers Oncology. And we found that the different panels that we compared performed equally well in terms of sensitivity and specificity, but definitely the anchored multiplex PCR-based approach was more compatible with a routine diagnostic, being easier to run and easier to analyze compared to the other assays. Unfortunately, this assay has a limited breadth because it covers only 26 genes involved in sarcoma fusions. So we thought, as part of our plan, to design a more comprehensive assay. And that's exactly what we did during these last months. We designed two different assays, one of mid complexity and one of larger complexity. This latter covers about over 55 sarcoma gene targets. And of course, this panel is going to be run during the coming months. The second trajectory is prognostic. As I mentioned earlier, the histological grading performs poorly in the prediction of the outcome of sarcomas. So this has been long known, and about 10 years ago, the French sarcoma group proposed a molecular signature called SynSARC, composed of 67 genes, that according to this group, predicts pretty well metastatic outcome in sarcoma patients. So we thought to take advantage of this signature and try to simplify, to make it more compatible in the routine diagnostic, because despite the fact that the French sarcoma group claimed that this signature was efficiently capable of predicting the outcome of sarcomas, this signature didn't gain ground in routine diagnostic because of the complexity of application. So we thought to take advantage of the data of our RNA-seq profiling that will pile up during this triennium to come up with identification of minimal signature applicable in the diagnostic lab. But in the meantime, we wanted to test the proof of concept of designing a minimal signature applicable in the routine diagnostic. And to do so, we took advantage of a set of about 90 samples that were previously profiled with a SynSARC signature, and for which we had the classification in a high risk and low risk according to this signature. And by comparing the SynSARC signature with other signatures, prognostic signatures in other tumor types, we came up with a minimal 18 gene signature that gave us a score which was calculated with a very simple algorithm. And as we see in the plot, we see that the area under the curve of the 67 SynSARC signature and our signature is pretty good. So there is the proof of concept that the complexity of the SynSARC signature can be reduced to a more compatible with the routine diagnostic signature. The last trajectory is what I call bioclinical, and it deals with the alignment of laboratory investigation and ISG clinical studies. And essentially, it relates to the molecular profiling of sarcomas, including ISG clinical studies, with the rationale that by better knowing the biology of the natural history of sarcoma, we may come up more faster and more easy with a new diagnostic, prognostic, predictive markers. We know that this is doable because we previously demonstrated by collaborating with the Italian sarcoma group and other European consortia that by aligning molecular profiling, we could provide to clinical trials molecular signature predictive of clinical behavior. So for our group, we identified two initial clinical studies to work on, the EPISOD study and the Thomas 2. The EPISOD study is a study on epithelioid sarcomas, is an observational study. And epithelioid sarcoma is a very rare sarcoma type. It accounts for less than 1% of all sarcomas. It's typically an epigenetically driven tumor because it's characterized by MARC1 inactivations. And from a pathological standpoint, is distinguished in two forms, clastic and proximal, with the proximal type behaving, as we see in this curve, clearly more poorly than the classical type. However, a very recent study conducted by the Italian sarcoma group demonstrated that even the classical type, the so-called more benign form, can be subdivided in two forms, high grade and low grade, with a high grade essentially overlapping the curve of the proximal aggressive type. So there is much to know yet about the biology of these sarcomas, and we need more efficient prognostic parameters. So bearing this in mind, we profiled during the last months 22 samples of epithelioid sarcomas included in the EPISOD studies, 9 proximal and 13 classics. And these were the preliminary results, where we see that the two sarcoma types essentially are clustered apart according to both the principal component analysis and hierarchical clustering. But one sample, but this was prior histological revision. After histological revision, also this sample that was eccentric essentially was reclassified as a classic type tumor, and then The results of our ANC profile perfectly match with the histological classification with a netted separation between classic and proximal type. The high group classical type tend to cluster together, while the low risk group tend to be more separated apart. We analyzed the functional notation of the genes differentially expressed, and we are currently addressing the significance of this pattern. And we are performing also MIRNA profiling and planned to do mutation profiling by using the Gerson panel. The second study is the TOMAS-2 study, is the follow-up of a previous study called TOMAS-1 or TOMAS. That was a phase 1b study focused on non-resectable bone and soft tissue sarcoma, advanced and non-resectable bone and soft tissue sarcomas. And in this study, two first-in-class drugs, a minor groove binding activating agent, trabectedin, and PARP inhibitor, olaparib, were combined. The results of this study were promising, with 14% of partial responses, laying down the basis for the phase 2 trial, which is called TOMAS-2. An interesting result of this study was that BRCA1 and 2 mutation status did not seem to impinge upon the observed clinical activity. So the hypothesis was that other DNA damage response genes were probably involved in the response observed during the TOMAS study. Initially, we thought to design a DDR-specific NGS panel, but then it turned out that the Gerson mutation panel would have become available also to other groups besides the initially identified ones. So we decided to not to follow this project, this part of the project, but to wait for the Gerson panel to be available. But in the meantime, we decided to gain data about the mutation profile of these patients by doing exome sequencing profiling on a set of 12 samples included in the TOMAS studies. And so these samples will be used as a reference for the subsequent validation on the Gerson profiling. Of course, our studies, our profiling study will yield hypotheses that need to be tested and validated by in vitro and in vivo models. So part of the group was engaged in developing in vitro and in vivo models, PDX, zebrafish, and 2D and 3D cell models, particularly on sarcoma histotypes that were included in ISRG studies. So to conclude, we will keep pursuing our task of aligning our translational investigation with ISG clinical studies during the coming months. Besides the first two studies, EPISOPS and TOMAS-2, we have other studies that just were launched by the ISG, and we will focus also on these sarcoma histotypes. And to conclude, I want to thank all the members of the working group sarcoma for their contribution. These are the list of the members of the sarcoma working group. And of course, I want to thank you for your attention.
Thank you very much, Roberta, for your update. And are there comments? Or yes, Pier Giuseppe?
Great job, Roberta. Thanks.
Thank you.
I mean, it looks like you are pushing RNA-seq into the clinical routine. I mean, clearly expression signature are emerging now much more than before. That's great. But I know that we discussed already that, but I want to know your opinion on what do you think about trying to set up some robust pipelines to call translocations from the RNA-seq?
Well, we've been working quite long on that field, and indeed, I just wrote to Luca Mazzarella to suggest to be involved in this part of the project. So we have experience on different tools involved, used for fusion calling, and it's something that we are doing routinely. That's what I'm saying.
Yeah, because, you know, I mean, it is claimed that it's possible, though, I mean, I think is challenging, but it would be really great if we could routinely collect RNA, which is not a great deal. I mean, technologically, it is much easier than other approaches. I mean, a gene panel is much more difficult than an RNA-seq in terms of workflow, and the cost is getting really down. So it would be nice if from a single assay we could not only derive expression signature, but also fusion proteins. Maybe
it was part of the publication that we published.
Yeah, I said fusion proteins, but I mean, we are late in the night.
What I was saying is that it was part of the publication that I mentioned on the fusion, the validation of the fusion panels to run also transcriptome and fusion calling on the samples. So it's something that we do routinely. I want to mention that the samples that we are profiling are all archival. We work just on FPV samples because the rarity of tumors is such that we cannot be picky and just select for frozen samples.
Of course. Thank you.
PG, just since Roberta called me in question, yes, so I confirmed that she sent me an email earlier today. So definitely within the GERSOM trial, we can, I mean, the framework is there to validate different methods for detecting fusions, because with the new amendment, we are going to collect RNA samples as well for the transcriptomics. So if the amount of RNA required is permissive, we can definitely test different assays.
Roberta, this is Gennaro. Nice work. One also suggestion, given the rarity of the cancers that you are studying, have you considered the possibility of aligning with making an alliance with other networks outside Italy? For example, we can talk of Yaniv Blay.
I could barely hear you, but I understood you mentioned Yaniv Blay. We, for example, we have a paper that just came out a few weeks ago with Yaniv Blay. We have been collaborating with the groups in France, also in the United States. Of course, there are issues about sharing samples. That's a major issue. But about the technology, that's definitely.
Exactly. So I was not considering of sharing samples, but technologies and programs, just to have an exchange of ideas and plans.
And I have to say that Yaniv is a very nice and open-minded person, so it's easy to collaborate with him.
Good.
Okay, thank you very much, Roberta. We go on with the next speaker, who is Katia Scotlandi, who will introduce the new working group of ACC about skeletal muscle tumors. Katia?
Good morning. It is a privilege to present this novel working group. This is a working group that is devoted to the study of musculoskeletal tumors. And you can see in this first slide the people and the institute that have already accepted to being part. But of course, all the other researchers and institute that can be interested in working in these rare tumors are more than welcome. As I said, this was a working group that was approved at the beginning of this year, but due to the sanitary emergency, we couldn't start our activity. So actually, I'm not in the position to present you a reporting activity, but rather I would like to share with you our ambition and our aims. So we have defined three major ambitions inside this working group. The first ambition is to run biological and genetic studies inside international clinical trials. Bone musculoskeletal tumors are substantially rare tumors. So this means that we have to search for an international level in order to run clinical study in an adequate time frame shift and in order to obtain results in few years. And we are already involved in several national and international studies, and I think we have the great opportunity this year to have a new international study on Ewing sarcoma started in 2021. This will give us the great opportunity to run the biological and genetic studies in real time in parallel with clinical annotation data. The second ambition is to exploit what we already have done in our institute, and to exploit the models the experimental models that we have already obtained in order to test novel drug combination in an appropriate genetic and cellular context. The third ambition is certainly to improve our understanding on the immune microenvironment of this tumor, because these are tumors that are usually defined as a cold or even desert immune disease. So the working group, of course, is devoted to study diseases that affected muscles and bone. If we are speaking to bone, we have a large proportion of benign tumors, but we also have malignant primary tumor and bone metastases. Everybody who would like to help us to organize a subgroup working on bone metastasis is more than welcome. But right now, to start with, we have decided to focus on primary malignant tumors. And this mainly means to focus on three major diseases: osteosarcoma, chondrosarcoma, and a small blue round cell tumor of bone, which include Ewing sarcoma. These tumors are rare, but they also involve mainly children and adolescents, so younger patients than most of cancer. This means that these tumors have an important social impact, and certainly these tumors are in a desperate need of definition of novel therapeutic strategies. The recent clinical history of this tumor has been very disappointing. Patients do not benefit from targeted therapies, do not benefit from the recent introduction of checkpoint inhibitors. And we are dealing with tumors with very complex genetics that prevented us to identify common druggable mutations. When I say complex genetics, I mean on one side either the extreme intra and genetic heterogeneity that characterize, for example, osteosarcoma, which is due to measured chromosomal alteration and really chaotic rearrangements. But I also mean that the fact that we are dealing with tumors that are mainly driven by epigenetic mechanisms. And unfortunately, we still know very little about epigenetic regulation of gene expression. So our aims, the aims that we have defined for the next 3 years, are mainly to provide the most comprehensive genetic information that we could achieve, that we can achieve, and to provide that information to patients and to clinicians, to exploit, as I said, the models, in particular the patient-derived xenografts and patient-derived xenograft cell lines that we have already developed in order to design novel therapies and to assess immune infiltration. And the fourth aim is to provide again, a general and comprehensive information about the cytokinomes and the exosomes-related microRNA and proteins in the blood of the enrolled patients in order to identify circulating biomarkers. Actually, we can start with something because in the last 3 years, starting from 2016, we have three centers under the umbrella of the Italian Association, the Italian Alliance for Cancer, have worked for this international European project. This project was mainly devoted to the development of models for pediatric tumors, and actually we have obtained several patient-derived xenografts and several patient-derived xenograft cell lines, which proved to be much more efficient than cell lines that were developed directly from patients. And this helped us to have right now 12 complete models for osteosarcoma, 5 models for Ewing sarcoma, 4 models for rhabdomyosarcoma, and 5 models for synovial sarcoma. All these patient-derived PDX and all these patient-derived PDX-derived cell lines are fully genetic characterized, and some of these PDX have already sent to PharmaMar, to the company, drug company PharmaMar, in order to provide us with also a pharmacological characterization. So in addition, with the work package can also take advantage of some specific expertise and models that are already in place in participating centers. So in particular, we can take advantage of immune-competent mouse osteosarcoma models that have been developed in Milano. We can take advantage of the expertise in 3D models that is developed in Napoli, and we can also take advantage of the development of zebrafish models in Meldola. All the researchers that participated to this working group has also already in their hands solid and robust evidence, preclinical or even clinical evidence about possible novel therapeutic approaches. And we are going to test all of this. In addition, of course, inside the working group, we can have upfront technologies that is spread in the different participating centers. And we can also take advantage of a very strict collaboration and a long-lasting, actually, collaboration with the Italian Sarcoma Group. I am also in the board of the Italian Sarcoma Group. This is an association that was created more than 30 years ago. It includes 68 centers, including a national institute, but including also university center and hospitals and general hospitals. And as you can see, all the centers are very well spread around the country. And so we can say that through the collaboration with the Italian Sarcoma Group, we can really reach all the patients, or the great majority of the patients that are treated in Italy. The members of the Italian Sarcoma Group have different expertise, and this also means that we can provide a multidisciplinary vision, a multidisciplinary approach to the treatment of these diseases. And again, the Italian Sarcoma Group is very active in running clinical trials. As you can see, there are at least 23 clinical trials open and ongoing right now. Some of them are national, some of them are international, and this is a great opportunity for us. For example, in this case, this is an international clinical trial that was run in collaboration with the Spanish Sarcoma Group. We had the opportunity to have biological material, blood and tissue material in patients that were enrolled in these studies. And so to start with our activity, we plan to use this trial as a common platform, as a common initial platform to study what I have said before. So to provide to clinicians with a comprehensive genetic information, and we are going to perform a gene expression profile of sample pre and post treatments. And of course, we are going to use the RNA sequencing because we need to know also, to have information also on epigenetic mechanisms. We are going to characterize the immune infiltration with a specific information, with a specific attention to macrophages, which constitute the major population in this type of tumors. We are going to evaluate and also to validate biomarkers that have been selected inside the international committee. And as I said before, with the blood samples, we are going to study blood samples either for the definition of a cytokinome profile, but also for the definition of microRNA and proteins that are associated with exosomes in order to identify possible circulating biomarkers. In addition, of course, in the next years, we are also going to perform functional studies. In particular, we are going to perform a drug efficacy test with these agents that have been already studied inside each center. So the general idea is to have a common platform and to spread the samples and the studies in the participating centers according to their point of strength, because we are all well aware of the fact that we are dealing with rare tumors, and for this tumor, collaboration is certainly an essential point. We are also very well aware of the fact that for this tumor, we still have a long way to go, but we are also very positive about the creation of this working group. We truly believe that this working group represent a unique opportunity for study the samples in association with clinical information in real time. And we really thank the Italian Alliance Against Cancer for giving us this opportunity. Thank you.
Okay, thank you, Katia, for sharing with us your ambitious projects. Sounds very interesting. Are there comments or questions? Pier Giuseppe?
Yes, ciao, Katia. Good luck. A simple question, Katia. Maybe you said and I lost it and I apologize. How you want to genomically characterize the osteosarcomas? You think to whole exon sequences or gene panels?
No, actually, we would like to deal with RNA sequencing because actually, even in osteosarcoma, all genome sequencing have been done quite in a large series of patients, and the definition of subgroups is still quite difficult. So we think that we have to choose, and we think that we are trying to go for RNA sequencing in all these tumors.
Okay, thank you, Katia.
Okay, any other comments or questions? Okay, then maybe we can move on. We are really running late. The next speaker is Enrico De Renzini, who will update us on the progress of the oncohematology working group.
Good evening to everyone. So it's a great honor for me to be part of this 2020 annual meeting of Alliance Against Cancer. So in this presentation, I will summarize what has been done during last year for the working group of hematology. In the second part of the presentation, I will leave the stage to Dr. Matteo Della Porta from Humanitas Cancer Center, who will present a very specific project that I really hope that will be actually expanded and implemented in our working group. So first of all, the working group has some specific objective and a 3-year scientific project. It's very focused on AML and DLBCL, so only 2 diseases to start. And the main aims are basically 2. So first of all, the institution of a high-tech consortium for innovative clinical trials, which is going to be the last goal. And second, the 3-year scientific project is going to be a multi-omics profiling of DLBCL and AML with TP53 genomic alteration, focused on functional characterization through single-cell RNA sequencing, metabolomics, and whole exome sequencing. So right now, the consortium actually accounts for 19 centers distributed across all Italy. So this is the action plan and milestone slides that we proposed last year during the past annual meeting. Of course, as you can see, this is a 3-year project as all working groups. First of all, we plan to basically to create the network and activate the project to start the multi-omics profiling of the TP53 dysfunctional malignancies, and to carry on some projects such as clonal hematopoiesis and study of genetic predisposition, which is basically carried out by Matteo. And so we were planning to have a kickoff meeting in the early 2020, and then to go straight to the EC approval of the main protocol. And then during the second part of the year to start some diagnostic profiling and some multi-omic profiling of the LBCL and AML. So the backbone, as we will see in the next slide, of the working group of the 3-year project is this INTEMA trial, which is a flexible platform able to be the backbone of multiple different projects. And basically, this year, not only for the COVID-19 emergency, have to be honest, but also for some difficulties, some legal issues in the implementation of the INTEMA trial, basically our work plan actually shifted by 4 months. But for this reason, in order to achieve the targets actually that we were planning for the first year, we promoted some preliminary collaboration between centers to effectively start diagnostic profiling and multi-omics profiling. So what is the INTEMA trial? The INTEMA trial is a retrospective and prospective collection of clinical and biologic data, also for multi-omics studies, which actually makes possible the proposition of multiple different studies. This is going to be the backbone of our 3-year scientific project on TP53 dysfunctional malignancies. And in this trial, which was originally promoted by the Medola Cancer Institute, the working group hematology of ACC is going to be co-promoter, and each center will retain a scientific leadership of the proposed studies. And ACC and the involved centers will retain full ownership of the data. The main advantage is going to be that we're going to have a unique informed consent form, and then we're going to have a specific consent form for the centers actually affiliated to the VG EMA network. And the ethics committee notification will be required for most studies. So when the patient is initially enrolled at diagnosis, the patient signs a comprehensive and informed consent. And then when each center actually proposes a different study, most study basically will start only with a notification from the ethics committee. We're going to have also a specific scientific committee from the ACC network that will evaluate each project, both on the scientific side and also on the funding side, basically to approve them. And regarding the data, so we have an open clinical database which is implemented in the INTEMA trial. And for genomic data, we're going to use the ENFN storage for of the ACC network. So this will be a first-year report. Will be divided in 5 sections. First of all, I will briefly talk about the kickoff meeting and establishment of the network. Then I will talk about the technology survey that we have, and then I will talk on the difficulties of the main steps of the implementation of the INTEMA protocol. And then I will touch some specific points, such as definition of the targeted NGS panels in the LBCL, and also the project of diagnostic whole exome sequencing in AML. And then finally, what has been done right now regarding our 3-year project on multi-omics characterization of TP53 dysfunctional lymphoma and leukemias. The kickoff meetings actually was originally planned early February, but then for the COVID-19 emergency, so was postponed, and we had 3 virtual different meetings back in April. So the discussed topics were, of course, the work plan, the establishment of the consortium, the implementation of the multi-omics 3-year project, and of course, the discussion between centers on available infrastructures and proposition of additional projects. So I think these meetings were a success because multiple centers actually were very interested and actually proposed different projects that are outlined on the left side of the slide. And today, actually, Matteo Della Porta will present his own project on genetic predisposition of myeloid malignancy, which I really hope is going to be actually expanded to the centers of the network. Second, the technology survey was a step really important because we needed first to confirm the feasibility of our plans by assessing actually the technology platform available in the different centers. From this slide, you can see that actually most centers, like basically 90% of the centers, have the MySeq sequencing platform, and then 60% of the centers have the NanoString platform for gene expression profiling, and almost 40% of the centers have a single-cell sequencing technology with the Chromium Controller 10X. So basically, these slides tell us that what we want to do is actually possible. Regarding the metabolomics, this is going to be performed by Simone Cardaci at San Raffaele Hospital. And this slide actually also supports the choice to employ actually the Illumina platform for targeted NGS of lymphoma, as we will see later on in the presentation. So I was talking about the implementation of the INTEMA trial. So the INTEMA trial actually is an already ongoing trial, which was actually originally promoted by Meldola Cancer Institute. And actually insert the ACC VGMA network inside the INTEMA trial actually is a substantial amendment will be necessary. So first of all, we had to review the trial to include the network. Then we had to go to a first approval of the DPO of the Meldola Institute, and then a first revision from the ACC legal consultant. and then a first revision from all participating centers, that was June, and then a second revision from the DPO of the Meldola Cancer Institute, and then a second revision from the legal consultant of the DPO, and then a revision from the ACC GDPR, and again, ACC legal consultant, and then a final revision of the ACC legal consultant, and then a final revision from all participating centers. Actually, we are now at this stage, and we actually were really close to the final submission of this amendment, which will basically introduce the ACC network of this INTEMA trial. So regarding some specific point, the NGS of AML. So last year, actually, I presented this targeted NGS panel, which were actually implemented by Pier Giuseppe Felici and Chiara Ronchini during the past few years. But I want to focus right now on this project that actually we discussed during the kickoff meetings, and actually to explore the diagnostic potential of whole exome sequencing in AML. And these are some preliminary, very preliminary data actually demonstrating that whole exome sequencing and the performance of whole exome sequencing versus targeted NGS, which actually is very high. So basically, more than 90% of all the single nucleotide variants were actually picked up by whole exome sequence. This is a cortex of Chiara Ronchini. And then I want to share also some preliminary data that were generated at the Regina Elena Institute. So as I had mentioned before, to start basically profiling some AML and DLBCL, we promoted this preliminary collaboration between institutions. Then at Regina Elena, 15 AML samples were sequenced with whole exome. Many of them were relapsed, but also 5 or 6 of them were at initial diagnosis. And as you can see here, this is the oncoprint of the top 25 mutated genes. One of the recurrently mutated is P53, which is 40%. So this is the distribution of the mutation in the TP53 gene. And this really strongly supports actually also our intention to characterize from the functional point of view this TP53 dysfunctional AML with a multi-omics profiling. And then regarding the B-cell lymphoma, so we actually established a new targeted NGS panel, which is a 146 gene panel. And then this was done in collaboration with multiple centers, actually that are actually leader in this field, such as Bellinzona, Novara, Columbia University, Yale Institute, and also Aviano. And there is an ongoing agreement with Sophia Genetics also for to have a bioinformatic platform. And this is some preliminary data actually that Professor Pileri sent to me on the targeted sequencing on aggressive mediastinal lymphomas, which can be of help in distinguishing some very similar forms such as primary mediastinal and gray zone lymphoma versus Hodgkin lymphoma. And then regarding the targeted gene expression profiling, so we have some nanostring panels, so we can implement a 22 gene panel providing all the cell of origin classification plus targeted genes. And we recently published that only 3 genes are necessary to discriminate the prognosis. And then this signature actually is characterized by also a different metabolomic features, but these are still ongoing. This is ongoing research. And we really hope that this can be implemented in the working group. Also, we decided to go with the IO 360 gene panel, which is also a very, very interesting gene panel from nanostring technology, which actually encompasses more than 700 different genes, and so can be really suitable to do some discovery. Finally, regarding multi-omics characterization of TP53 dysfunctional lymphoma and leukemia, so we had a preliminary teleconference to decide the protocol, because this is not really easy. We will need to basically isolate leukemic blast and lymphoma cells, and then subject these cells to single cell RNA sequencing and to metabolomics. So we decided to restrict the analysis to CD34 positive leukemic blast, which will be separated through actually the employment of magnetic beads with a Miltemi instrument, and then same thing for lymphoma cells. These cells will then incubated in a plasma-like medium called Plasmax, and then after 6 or 12 hours, cells will be subjected to metabolomics, single-cell RNA sequencing, and whole exome sequencing as well. And now there are 5 samples ongoing. All these things actually were done in order to be able to start really with at full speed once our INTEMA trial is finally approved. So in conclusion, the approval of the INTEMA trial is imminent. There was a substantial amendment needed to include the ACC network as co-promoter. The data storage platforms have been defined. We defined also a new targeted NGS panel for B-cell lymphoma, which is really new. And there are ongoing negotiations with Sophia Genetics for the bioinformatics support. And then we really promoted in the last 2 months preliminary collaboration between participating centers, basically to be able to meet actually the deadlines that we proposed last year in terms of number of DLBCL and AML profiles with sequencing or targeted gene expression profiling. And now we also started the metabolomics project of TP53 dysfunctional malignancies with 5 ML cases actually that are ongoing. Finally, we defined also future collaborative studies. For example, besides the diagnostic whole exome in AML, but also Genova, Professor Lemoli proposed a study to investigate the BPDN-like AML. Dr. Pinto from Pascale Napoli proposed a nanostring study on PMBCL. Carlo Stella from Humanitas is very interested in liquid biopsy, and then we have an ongoing study on clonal hematopoiesis, and Attilio Guarini from Bari proposed a study on long-term survival. So I think there is a lot of interest in this working group. And finally, Matteo Della Porta will now present his project on genetic predisposition in myeloid malignancies, which is really, really fantastic project. And I really think that it's a great opportunity for all of us to have this project in our working group. Finally, main points that will be very important for next year: implement collaboration with established networks such as GMEMA, PhilCIE, and GITMO. And of course, we need extra funding because all these things are otherwise impossible. So we need to implement collaboration with pharma companies. And we also need to participate in cooperative grant application. So thank you for your attention. And I really want to thank all participating centers. A special thanks to Giorgia Simonetti. She has really done a fantastic job in the implementation of the INTEMA trial, and to Professor Fabio Ciceri for his strong support. Thank you.
My name is Matteo Della Porta from Humanitas University. And first of all, I would like to thank the Scientific Committee of Alleanza Contro il Cancro, and in particular Enrico De Renzini, for the opportunity to discuss a possible research initiative on the study of genomic predisposition to myeloid neoplasms. As you know, recently, the WHO classification recognized a new category of diseases that are called myeloid neoplasms with germline predisposition, that refers to a subgroup of cases in which the dermatological malignancy is associated with a germline predisposition, and this category may account for about 5 to 10% of adult patients with myeloid neoplasms. The recognition of these cases is relevant not only for a patient perspective, for instance, for the correct choice of a donor for a transplant procedure, but also in order to define the possible clinical implication in family members. So the knowledge about genes that are associated with the predisposition to myeloid neoplasms is quite limited to date. As you can see here, the WHO classification currently stratify these diseases from a biological point of view on the basis of the presence of a pre-existing platelet disorders associated with specific germline mutations, or the presence of organ dysfunction that refers to specific genomic abnormalities or syndromes. So recently at Humanitas, we created a center for the study of genomic predisposition to myeloid neoplasms in order to provide evidence for the clinical implementability for a genomic screening in order to detect these specific germline aberrations. The center is coordinated by a multidisciplinary team, as you can see here, that is able to provide a genetic and hematological counseling, of course, together with a specific team of molecular biologists and bioinformaticians that are able to provide NGS screening and variant calling. So the first point is to correctly select patients that are candidate for this type of genomic screening. And as you can see here, we developed a specific flowchart in order to provide objective criteria to have access to this type of genomic investigation. Major criteria are based, of course, on familial history, on the presence of multiple neoplasms in the same patient, on the presence of platelet disorders, diseases associated with specific germline mutation, as you can see here, on the basis of the presence from a previous mutational screening on tumor cells of a somatic mutation that can be associated with genomic predisposition, such as DDX41, for instance. And finally, on the basis of a presence of peripheral blood cytopenia that is possibly associated with congenital abnormalities or syndromes. This slide represents the NGS panel that is currently in use in our center. This panel is based on 106 genes and includes all the genes that are recommended by WHO classification, together with another small group of genes that are recognized to be specifically associated with the risk of developing myeloid neoplasms. So basically, as you can see here, the patient is firstly evaluated by an hematologist, and a genetic and an hematological counseling is performed. Then a peripheral blood sample is collected together with a skin biopsy in order to have access to a normal control sample. And then the screening by using the panel that we have previously described is performed. After 2 months, the results of the screening are released and communicated to the patient. So this is, of course, a pilot initiative, but that provides evidence for a clinical implementability of the study of genomic predisposition in a clinical context. And I hope that this first initiative can provide the rationale to define a project within ALLEANZA CONTRO CANCRO Alliance in order to create a national network of centers that are able to collect this kind of information in a large patient population. Thank you very much for your attention.
Thank you very much, Enrico, and thank you, Matteo, as well for this very interesting presentation. And the perspective of these studies is very interesting. So we have a question from Pier Giuseppe.
Yeah, I mean, a question for Enrico and a comment on Matteo's proposal. Thanks, Matteo. I think this is very important. You know that it is in the DNA of ACC to generate panels which explore the genetic risk of cancer, including hematopoietic tumors. So we must absolutely structure a project on that. Thank you very much. And I have a little question for De Renzini, Enrico. Enrico, very nice setup, much better than I was coordinator. Good, was good for me to live. And the WES validation, it is really an important step. It can be then used for also other applications. Where do we find the money, Enrico?
So this is why I think it's crucial, really crucial, to get extra funding. So of course the funding from the ricerca corrente is enough to start doing something, but if you really want to achieve your goals, then we need to participate to European grants and also have some support from pharma, I guess, because the final endpoint of the working group actually is to run some clinical trials. So that's the endpoint of having a high technology platform, is to be really strong as a hematology network to propose trials and to get support also from pharma to do that.
Maybe, Enrico, we should go again to talk also to AIL, because when we proposed this WES validation, that was 1 year ago, they were interested in principle. So maybe we can again try to establish a tight connection with AIL and with the AML group, no?
That would be great, yeah.
Thank you.
Okay, Pier Giuseppe, did you have any comments for Matteo?
No, I already said that I like.
That was it, okay. So thank you again, and we go to our last speaker. We're very particularly happy to see you here today. Enzo Medico, who will tell us about the progress in the colon cancer working group. Enzo, you have 15 minutes and 5 minutes of discussion.
Are you seeing my screen?
Yes, we are.
Yes.
Okay, perfect. So yeah, I'm really happy to be able to present this, improving a lot. And so unfortunately, I did not have the opportunity to complete everything as I wished, but I think we have a very good update anyway. So the objective of the strategic program for the working group colon is to implement in a multicentric, let's say real world context, the generation and analysis of multidimensional NGS profiling on colorectal cancer. So the objectives are standardization and implementation of procedures to perform multidimensional next generation sequencing on FFPE tumor samples, so some standard source of material that could be used in routine analysis. And the 3 types of profiling are mutational profiling. We are thinking of the Gerson panel, or we could decide otherwise if there are problems in using the Gerson panel, but this has to be defined together with Luca and the Gerson board. And determination, of course, of the mutational load, as you have heard from Luca. Second is copy number profiling, so to assess gene amplification and deletion using shallow whole genome sequencing. We and Candiolo have been running that already for more than 300 samples with all the analysis, so we know how this has to be done. Third, transcriptomic profiling by global RNA sequencing. I want to stress that the protocol chosen, because of the quality of the result on FFPE, is ribodepletion. Ribodepletion leaves everything except ribosomal RNA, so it is very good for characterizing the transcript at multiple levels, including non-polyadenylated transcripts, including circular RNA. So we are going to build a resource of RNA sequencing, which can be used for the years to come to explore many different things. And then, of course, the final objective is to integrate these things for optimal prognostic and therapeutic characterization of the patient subgroups, and also for each individual patient in a perspective of precision medicine. So we had planned a number of activities. Of course, the first task was to define the set of samples to be analyzed, 65 per year, 5 samples per participating center. So not a big effort, but consider we have to set up everything. This is going to be demanding anyway. To address, this set should address an unmet clinical need and a scientific problem. The sample should be available at all centers with sufficient material for the profiles. And they should be defined, they should be retrospective for feasibility with appropriate clinical annotation. Then definition of the standard operating procedures and training were necessary. Finally, the samples will be selected. DNA and RNA will be extracted at each center. And then Candiolo will perform the QC or also the single centers. Each center will perform library preparation and run on Thermo Fisher systems the mutational profiling, prepare libraries for shallow whole genome sequencing and RNA sequencing. Finally, all these reagents will come to Candiolo, where Candiolo will perform RNA sequencing, which is not actually written here. And then, of course, all the data will be processed, quality controlled, and the idea is finally to set up a shared archive, of course, in collaboration with the ACC platform, and to evaluate the success rate and to make some benchmarking. So these were the strategic program activities, and we also had an institutional program, so that every year we would take 65 selected cases from the Gerson project for colon to be further profiled by shallow whole genome sequencing and global RNA sequencing. So to add more layers of profiles to the Gerson samples. The selection, in this case, will take into account possibility to answer specific questions, like, for example, to highlight amplified genes or transcribed fusions for cases that do not have obvious targets in the Gerson profile, so to find some possible actionable target. And stratification in transcriptional subtypes and the potentially predictive or prognostic immunophenotypes. The analytical procedures will be the same as the strategic program, so there is no big effort here, except to find and select the appropriate Gerson samples. So the project is really distributed. As you can see, 13 centers will all perform RNA extraction and processing. Will all perform the Gerson profile on Thermo systems that are available in all but one center. Preparation of libraries. So everything will be carried out by each center so that the procedure will be really implemented in a multi-centered way. Then, of course, sequencing will be centralized at Candiolo. So we had all these planned activities. Then, as you know, the unpredictable always occurs. So the COVID impacted severely on the activities, first of all, because we had the personnel of the facilities on which we counted for a number of things related to the project was somehow reorganized for COVID lab. So we could not count on the facility. Personnel was reduced, the possibility of access to the institute. So we could really not lead to too much activities right now. So we couldn't even recruit the personnel, the unit of personnel that was meant to carry out this at Candiolo. Now we have it, so we can start now with the practical activities. What did we do in the meantime? So the possible studies that were that emerged from some discussions were essentially 3, and the one will be prioritized and selected in a TC plan for mid-November. First possible study, intratumoral heterogeneity. So in this case, each center having like 5 samples per year would actually perform 5 samplings of appropriate cases to study intratumoral heterogeneity. This has biological and clinical relevance because typically molecular profiling or genomic profiling is carried out in 1 sample. But actually how intratumoral heterogeneity is present and how it affects the diagnostic response of molecular profiles is still a pending question, although there are many works on that. The second possibility is to choose right colon T3 with early relapse. So in this case, the standard surgery would not be enough. Prediction of those cases with worse prognosis in the T3 settings, in particular N+ setting, would indicate a more radical and aggressive surgery, which should not be performed on everyone, which is total mesocolon excision. The third option would be to choose, again, right colon, but T2 and 0, so substantially good prognosis cases, but that we have early relapse, half with early relapse and half without, so that predicting early relapse in T2 and 0 would actually indicate adjuvant treatment. So as you can see, all the three settings have good reasons to be chosen, but it will come to a practical issues of feasibilities and interest of the various center to finally select one of these three. So about the standard procedures, we progressed in particular for RNA. We also have standards for DNA extraction, library generation, sequencing, and the analytical pipelines. So let me briefly outline, in particular, the organization of the RNA sequencing workflow. So we organize operational modules where you have an input, a procedure, and an output. So the first step is the procedure is RNA extraction. You can have different inputs. FFPE is particularly our interest. Protocol for extraction. From that protocol, you obtain tissue and data, okay, and data sheet. And the output is RNA and data associated to the RNA, in particular QC data that have been encoded precisely what should be the QC of the performance. Then, of course, the RNA and the data would go for library preparation so that every center could perform based on the protocol and on the RNA, in particular ribodepletion, prepare the library and attach the data, QC data of the library, frozen aliquot of the library. And then this would come to Candiolo for the other procedure, which are pooling of the libraries, run the sequence, and process the data. So again, in this case, the output of the data have been defined as precise parameters that would allow to assess the quantity and quality of the RNA here, and also the quantity and quality of the libraries. Then in Candiolo, all the procedures related to library pooling, sequencing run, and data processing, again, according always to this operational module. With more or less everything in place so that we know exactly what goes in every place, we actually faced a couple of problems with this procedure. The first one is that the RNA extraction that we have defined as the best requires a system, which is a Maxwell apparatus, which is not available at several centers of the working group. Second, the Paola Nistico and Regina Elena tested Maxwell. They wanted to obtain it, and so they tested with the Qiagen and found actually with Promega, and they found no significant difference with the all-prep kit, while we observed significant differences. So we don't know exactly what's been going on, but all-prep doesn't need a dedicated instrument, so it's easier to implement. So I mean, it can be implemented even without dedicated instrument. So we planned a new benchmarking experiment on 2 tissue types. So the lung cancer from Regina Elena, 5 samples, and 5 samples from CRC. Each center will extract DNA and RNA with own method on all 10 samples. We with Maxwell and Paola with all prep. RNA samples are shipped from Regina Elena to Candiolo, and Candiolo performs comparative QC and RNA sequencing on all of them. We will also be willing to perform RNA sequences to some of the previously extracted RNAs with Maxwell and all prep, where they found no particular difference. We will want to see whether finally the library is coming out as well from both kits. So now the samples have been identified, extraction started. I'm just providing here some preliminary data thanks to the work of Caterina Marchiò in Candiolo, Enrico Berlino and Daniela Cantarella, and of course, team of Paolo Nistico. So this is the samples of Candiolo. So actually they are from a series of couples, primary and metastatic colorectal cancer, 5 couples, part of 23 couples that have been selected for the older project of paired primary metastasis, which is ongoing. I don't have updates, unfortunately, I couldn't put that together, but it's progressing also. So as you can see, this is the total RNA yields, and the minimum acceptable yield would be 500 nanograms. And this is the fragmentation. The lower the fragments of the fraction of fragments above 200 bases, and the lower is the quality and the poorer the yield. And the threshold would be 30. So all samples are above 30, as happens for the previous samples, so no big problems with the quality and also with the yield. And these are the primary samples. You can see they span a very good range of DV200 and of yield. So the same samples will be extracted by Allprep, and we will do the opposite with ROM samples. So we have contingency plans in place. So clearly the sample processing has been delayed by some 3 months, not really that much, but could be delayed until the end of the year. So we would consider processing 10 samples per center instead of 5 at once, because this could be feasible. So we would anticipate the processing of the 5 new samples of the second year, subdividing the sequencing cost between the first and the second year. The GERSOM enrollment has been delayed, so for the institutional program. So some centers might not enroll 5 adequate patients, so the right cases with the unanswered questions in time for the first year. So we would process more samples for the strategic program than GERSOM, and then compensate in the following year. So this is how we think we could anyway deliver within the proper time the results that are expected. So that's it. This is the team. These are the teams of clinical and preclinical reference people, and of course the people that helped us with the SOPs and with the benchmarking experiments, Daniela, Caterina, Enrico from Candiolo, and Paola from Rome. Thank you very much.
Okay, thank you very much, Enzo, for this very good summary of your work. Are there questions or comments for Enzo in particular? Okay, so I think that we can.
Sorry.
Okay.
Sorry, Miriam, this is Paola. Just a clarification. I just coordinated the work. All the work is at Regina Elena at NGS facility by Ludovica, Francesca Di Nicola, and Frauke.
I just didn't know the names, so.
No, sorry, no, it's just because I have to give credit to them.
Yeah, that's right, absolutely.
I just coordinate.
Okay, thank you for the clarification, Paola. And okay, so I would like to personally thank all the speakers of this session for the wonderful presentations, and I pass the word to Pier Giuseppe for the general discussion and the wrap-up of the afternoon. Thank you.
Thank you very much, Miriam, and I also would like to have Gennaro helping me in that for the next 20 minutes. Thanks a lot, Gennaro. Okay, so very briefly, thanks on my side too to all the secretaries of the working group. And I also would like to thank the attendees. It is really amazing. We have about 200 people connected with us during this marathon, which is really remarkable. And that's good. Okay, so very briefly, I think we had a great view of all the working group activity. My impression is that most of the working groups are at a critical point, which is pragmatically speaking very simple. Many samples will be finally, I would say, analyzed by multi-omic approach in the coming months. according to what I heard, the most of the working group are pretty at the point where they will increase the throughput and they will increase the granularity of the analysis. And I think that this point is critical because, first of all, it's due. I mean, this is what we worked for in the past 1 or 2 years. But it's also critical because we should not run into the risk of fragmenting too much the type of analysis and then losing the opportunity of gaining info from each of the working groups in terms of putting together data. I'm referring to a number of specific issues, which I will briefly mention. First of all, which we were already discussed during the session, but I would like to reiterate. First of all, standard procedures. I mean, finally, I mean, we're really doing a lot of efforts to have common standard procedures, which is the basis then to run multicentric studies clearly. And I guess Caterina is doing a lot together also with the efforts that we heard from Candiolo, from sorry, Enzo, I'm tired, from Enzo. And I think we should intensify this effort. Again, I reiterate what I said. Caterina, you should take the lead really of putting everything together, pushing people toward the very last experiment in order to have, as Gennaro was saying, the finally approved protocol that everybody can use. And again, Miriam, we should work on the website in order to make it until we don't have a place in the data center of ACC, a place where friendly and easily everybody can find and put things. The second thing is the approaches, the technological approaches that we are gonna pursue in the near future. So there are a number of issues. GERSOM, okay, the GERSOM has been mentioned by a lot of the working groups. Clearly, the GERSOM clinical trial, which is funded, as we know, encompass work for 3 tumor types: the breast, the ovary, and the colon. But the needs of a gene panel, high-density gene panel, I mean, are larger than those tumor types. No, we heard from the melanoma, but we heard also from other working groups for other tumor types. So that's for Luca. We should make an effort to start as soon as possible a survey of the needs of gene panels to understand exactly if the GERSOM can satisfy them and make it available. There is a little money, not that much, for extra tumor types to be analyzed, and so we should make an effort in order not to have the need for other working groups to reinvent the wheel with new gene panels, if the GERSOM is appropriated in terms of type of genomic space that is investigated. Then we have a lot of efforts on whole exome sequencing, RNA-seq, and radiomics. I mean, clearly, many working groups are trying to perform this analysis and collect data from the same patients. It's a sort of multi-omic approach. Again, there is an issue of where we sequence, there is an issue of cost. We should try to rationalize this in terms of making contract agreements with the Illumina for the reagents. It's ridiculous that we don't get important discounts on that. Illumina works not with institutions, works with own projects. These are projects, and we should try to make a common effort to obtain better price. We have to decide if we do need to hub a little bit the RNA-seq, for example. We have, fortunately, 2, 3, or maybe more, I don't even know exactly, NovaSeq around. They should be used, I mean, by our scientists. Then there is the level of the bioinformatic pipelines. I mean, I'm not saying that we should use always the same bioinformatic pipelines, but we should have common languages and common efforts. I mean, I remember at the beginning there was a pretty active bioinformatic community that was sharing ideas and problems and troubleshoot together, which maybe now is the moment to somehow to give a further push toward having people that communicate on this type of things. And then the last thing, and then I shut up, is the data collection. And this is, to me, looks like an emergency. In other terms, if we don't move fast, in a few months, we will have a different way of transferring data, storing data, and we already mentioned analyzing the data. And this shouldn't happen, in my opinion. And the other thing that shouldn't happen is that for any reason, the work of the working group is delayed because we want to do fancy things in order to have everybody working on the same structure. What I am saying is that we should make an effort to convey all these needs now that they are starting together with the Politecnico people in order to maximize the usage of the ACC data center. And again, I said in the beginning, there is some money there and we have to use it. I mean, no delay. We have to push toward having the data center as soon as possible to be responsible for the work. But I think we should make an effort to tackle all these issues now, because we really looks to me that we are ready to increase substantially the granularity of the analysis and the number of patients which are to be analyzed. So I stop here. I would be very happy to hear your ideas on these issues or others that I may have not seen in this afternoon session.
Okay, Pier Giuseppe. Can I?
Sure.
So obviously I appreciate the work being done by all the working groups and the tremendous amount of effort that's been put. However, as probably some people that know me, I also like to play the devil's advocate, and so I'm going to raise some issues which I think have to be raised in this situation, because there are not only lights but also shadows in this overview. So I think that we need to keep more focused in our efforts in general. I've heard a big spectrum of activities by the various working groups. And some groups are more focused than others. I come from a background also industrial one, and in general, the suggestion that people do is really to try to prioritize efforts and to keep the objectives very clear and focused. And certainly some groups are fulfilling this type of objective much more clearly than others, which so I perfectly understand that the ambition is to want to really create impact, but sometimes by spreading into several rivers, the risk is the one of not delivering on projects. And again, the second point is actually to deliver on projects. There are some projects which have been going on for a long time and have not yet produced papers, have not yet produced clear results. And this might be due to different types of reasons, and therefore, if we multiply efforts and open new lines of intervention, the risk of opening up several boxes without closing them is elevated. And obviously, I'm not going to mention which are the projects that have not been delivering or are at risk of delivering. Certainly, the reasons might be the design of the studies that we do, the difficulty of working as a network, and so on and so forth. But basically, this is a real thing that we can observe, that there are trials that are going on have been going on for 3 years and have enrolled only 20 patients, which is something that should lead us to think and to reflect about this. And the final thing is something which I can say as being a scientific director of one of the IRCCS, which is really dedicating a lot of efforts and to ACC, and which is heavily committed to ACC. And probably this is the view of a scientific director and the perception of a scientific director, which derives from talking to the representatives of the working groups in my institute. So the impression is that there is a spectrum of involvement of people and communication by working groups. There are some working groups which communicate and involve people in the projects very efficiently, and this involvement obviously is reflected by a higher degree of productivity and also happiness of the various centers. And while other working groups are less involving, less communicating. And this is something that, in my opinion, should be corrected. There are some working groups that have routine meetings. There are other working groups which do not have meetings for months and months. And I think this is something which requires, especially from the secretary. So if secretaries are very busy or have increased duties, they cannot participate themselves into the meetings, have to think whether this should be in some way, this role should be passed to other people who are less busy, in order to make that working group more efficient. I'm sorry to say this, but I think we need to be very, and to increase our efficiency even more than has been obtained so far. And I think at this point I will stop here.
Okay, so I think that we have a lot of things on the table. Gennaro basically made 3 points: focus, productivity, and involvement, which are pretty big. I make 2 points. Need of standard procedures, not only in terms of protocols and technologies, not protocols, but also technologies, and the issue of data sharing, structural data and clinical data. So I think we now can open the discussion on these 5 issues which are on the tables, and I think I would be happy to hear your opinions on that. Wow, I know. It is difficult to start. but
So Pier Giuseppe, maybe I will try to break the ice here with a couple of points. So I will start from a couple of comments that Gennaro put forward. Two in particular, the need to focus and the involvement on the institutes. So regarding the need to focus, I actually fully agree with what Gennaro just said, and I think that for a long time there has been a bit of a dialectics, let's say, within the different voices in ACC. There definitely have been some forces towards very ambitious projects that then have issues with delivering, and some other voices that try to focus on more realistic endpoints. I think that in a certain way, we are all trying to mediate between these two forces and somehow maintain projects that are not simply deliverable in the short term, because otherwise they would probably add little to the scientific advancement, but we are also trying to limit the ambition of some projects because in fact it is difficult to deliver, and there are, I think, some structural some systematic reason why it is difficult to deliver. And I think that very slowly some of these issues are starting to be solved. So what I'm thinking about is that, for instance, the administrative burden that has been necessary to launch some trials has been, for instance, significantly reduced. If I take my experience, for instance, in the lung trial and in the Gerson trial, it has been much easier to, and still difficult, but much easier in the Gerson trial to write down a protocol that could be digested by the different ethical committees compared to what happened in the lung project, because probably we built on our initial experience of what went wrong with the lung trial, and so we knew how to make things easier, although they are not yet definitely as efficient as they should be. Other issues that we have been, for instance, the data protection plan. This is something that is a well-known obstacle to a number, especially within the framework that we have been working in. The fact that we now have a DPO and we have a legal framework makes a lot of things much easier. Another major issue has been the data management. We did not used to have a platform for storing the data, and now we have it. So these have been major obstacles to delivering, because, for instance, if I take the work in my group, I can tell you that if I don't have a platform for sharing genomic data among the institutes, then it's legally impossible to work on that, because we would not have a way to share in a legally compliant way the data on which the bioinformaticians have to work. So I think there have been a number of structural issues within ACC that we are starting to solve at the moment. This morning, I raised another issue that we have not yet solved, I think, which is the logistics and the MTA. And as we said before, I think it's really necessary that we schedule a couple of meetings where we try to solve these issues, because again, this is a major hurdle. Share the samples for, let's say, the Gerson panel validation. We have been not in the possibility to obtain those samples because we were not sure whether we needed an MTA, how the intellectual property had to be managed, and all these things. So it basically takes 6 months to do something that may take like 2 weeks. And I am pretty confident that as we encounter all these issues, we will be able to manage it in a better way in the future. I come to the second point that Gennaro raised, which is the involvement of the institutes. I fully agree with you. And I know that there have been a number of instances where the involvement of the centers could have been better. It is actually very difficult to understand how to do it. So it's, if you want, it's very easy to pinpoint the problem. It's much harder to find out how to do it, because there have been multiple instances where there have been attempts at involving the different institutes at different levels. And this doesn't always work in the best possible way, because there are some centers that are heavily compliant and highly participating, some other centers that are less compliant and less engaging. And in a situation where you have to attempt to involve everyone, the bottleneck lies with the slower ones, of course. And these centers are not necessarily slower because they are bad or anything, but just because maybe they have different agendas in that particular time. And so when this situation arises, then you basically have two alternatives. Either you wait for all the centers to do exactly what was decided at the beginning, or you start somehow going on anyways without the active participation of those centers. And I must be honest, this is something that has happened in our group. And that's why in my attempt, I'm speaking personally, in my attempt to deliver while mediating, at some point I had to take decisions where it was more important to try and deliver rather than obtain everyone's participation. I can tell you, for instance, for collecting the samples for the Gerson validation, I had to repeatedly email lots of centers for having their samples sent over. We're talking about having 5 samples from each institute. So if you don't get 2 institutes, you only eliminate 10 samples. But if those 2 institutes take 6 months to send you the samples, then what do? Either you don't deliver or you don't involve those centers. So believe me, in the types of projects that we have been dealing with, the number of obstacles is very, very high, and it's not immediate to identify the right way to deliver while engaging all the centers. But definitely something we have to work better on. I don't know if this, if similar experiences are part of
Sure. Although I am a young in terms of secretary of the working group of immunotherapy, I confirm the position of Luca, because I obviously believe, as Pier Giuseppe told, that our major point is to collect clinical data. But you have to be convinced that is very, very hard to work with the oncologist. And there are some institutions who are completely out of the projects. We write so many mails, and it's really frustrating. So then all the network works only if you have the direct contact with the groups of the institution. So if Enzo is telling what is telling about the centralization, because I speak with him and then I speak just to simulate how is the way. If I was unable to submit still the paper for Immunoscore, it's because National Institute of Tumor give us a lot of trouble. So there are many bottlenecks that is very hard for a secretary to do. I think that the most of us are very passionate and would like to arrive at the step, but for sure the major problem is the scientific director of some institutions, and I have to be frank because I am sure that there are some scientific directors that don't push the work in the network, and second, some oncologists, because it's very, very difficult to obtain the clinical data. And without the clinical data, you cannot have the tumor tissues if we are focused in the paper, and so on. This is just my brief experience.
No, but that I agree, Paola. But I guess we have to make an effort to divide to separate layers. So one thing is what Luca was saying, which is we didn't have any network infrastructure, and everything was difficult because we had to start from scratch. Ethical committee, data sharing, data storage, privacy, blah, 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 blah. Okay, and I recognize the fact that it was slowly, but many things are on the way to be resolved, many of these problems. Another thing is that if the scientific director do not perceive ACC as an opportunity and don't push clearly it is a problem. I remember when I was secretary of the ACC Genomics working group, it's difficult to keep everybody together if there is no endorsement by the scientific directors, by the institutes. And we know that. We discussed that with Gennaro too many times. And another thing is our medical oncologists, which are very busy. And that's another layer too that we have to take care of, absolutely. But still, Luca, it remains, even if these three things are real, and we're working on that. I can guarantee that, and I guess Gennaro would agree, that in the last 4 or 5 months, all the scientific directors were very heavily involved in discussion on the strategy of ACC, which whatever is going to be the final solution, the final decision, is rejuvenating the interest of the scientific director for this association. But look, whatever these three things impact, it remains, we discussed it many times with the other secretary of the working groups, the issue of the focus. Even if these three things are resolved, the focus remains a big issue. Focus means either a single project of the involvement in the data processing, paper writing, blah, blah, of many people within the working groups. Otherwise, the output remains low.
Yeah, I wanted to clarify one point to Luca. So I said that when I raised the point of the involvement, I spoke as a scientific director of a center which is heavily committed. And the premise was that talking with my folks and the representatives of working groups, I realized that there are some working groups which are not communicating at all. It's not that they are communicating and our folks do not participate, do not give a reply. It's that simply there are months and months and months in which there is no news from the secretaries. This is a different issue.
Yeah, that's another issue.
Okay. No, I thought I was clear. No, I hope I clarified now.
No. But then, okay, fine. But then, let me raise it in a different way. What type of communication do we want to have within the working groups? Because it's one thing to have vertical communication where you have the apical structure, the secretary plus the coordinators, that assign a number of tasks and then periodically investigate how things are moving on. And then there's another model that is more horizontal, where there also are initiatives that can come from the bottom. So if we want to have the first model, then I agree with you that if a secretary doesn't periodically update everyone, then it's the secretary's fault. But is this really the model that we want? I'm not sure. Also because I don't think it would be feasible, because I don't think that there is going to be anyone who works as a secretary and also is able to keep up with all the different projects. So, and also let me add another thing. It is very difficult to provide meaningful communication when you are dealing with the actual analysis. Okay, so as long as you are at the planning stage, it is relatively easy to say every month, where are we? Have we written this? Have we written that? But when you have to actually deliver, there are a number of things that are difficult to deliver until you have the data. So, you know, there have been a number at the beginning of the works of the genomics working group, because I can speak for my personal experience. As you know very well, we did a number of boot camps with different bioinformaticians. In those boot camps, in those meetings, there have been a number of very specific tasks assigned to different bioinformaticians. Then it is difficult over time to keep up with these tasks. And it is difficult both from the secretary's point of view and from the assignees' point of view, because the assignees have a competing agenda, and when you try to get a feedback, they tell you that they were not able to deliver because they obviously have other things to do. And the same applies to the secretary, because the secretary has real problems in taking care of all those aspects. So now, to finalize my point, I agree with you that the structure of at least my working group should be restructured, and that is something that I have been trying to do. I have not been able to do it as I wanted to for a very simple reason. We didn't have data to work on. So there was no sequencing from the GERSOM, there was no sequencing from the Lang project, there was nothing to work on after a certain moment in time. So what am I going to tell to the bioinformatician community? We have to work on something that doesn't exist. It wasn't possible. We finalized the sequencing of the samples only a month ago. Now we have the data. Now it's the time to reconsider the structure of the bioinformaticians within the group. Before that, we didn't have anything to work on. That's why it didn't happen. That's exactly the reason why it didn't happen. But with the bioinformaticians in your institute, I actually did have discussions, and we did, I mean, with Matteo, we spoke a couple of times on a number of things that we should deliver, and we didn't yet deliver for reasons that we have discussed with Matteo. We have tried to restart from where we had left a few months ago.
I would like to add a comment from Silvia Franceschi that she just added, and then she also sent me a mail. Basically, she's pointing out that maybe some of the, I mean, the members of the working group should be re-evaluated by the scientific directors. This is to say that the situation is evolving, and maybe we are better understanding which is the best people that can suit for this type of role. And the second, for the future, Silvia is pointing out the fact that more agnostic approaches might be more functional to our goals rather than subdividing the work in tumor types. And that's important also to be considered. Okay.
If I may say, there are some centers that have been updating their members in the working groups with some sort of regularity. I mean, we have had some input from some of the IRCs, not all, but there is in some centers there is more thought than in others, but that's not a surprise.
May I add something to the discussion? So, no, I really agree with Professor Ciliberto on the issue of maintaining the focus, which is crucial. So, for example, in the working group of hematology, we decided to focus on 2 diseases. We have only one 3-year project, but still there are some side projects that actually could start, and then the risk to spread out too much is always there. So we really should be really careful to not to risk to have too many projects that you cannot finalize. But on the other hand, in order to have to increase the participation from all centers, I think we should do all the possible efforts to let all the centers actually feel the network as an opportunity, not only providing samples from other projects or for one project that actually is led by someone else, but also giving them the opportunity to propose something. So I think all the centers should really see the network as an opportunity to pursue also some projects of interest.
May I say something? I don't want to jeopardize the discussion, but there is one point that maybe needs a very in-depth discussion, and we have limited time now after the whole day and after 3 days of a continuous meeting. But we should think about the way the working groups have to work and have to decide about projects. Because if there is, in my opinion, this is my opinion, but I might change my opinion. If there is too much bottom-up ideas coming, and then a lot of programs being started without putting order, the risk is that of creating too much entropy into the system. So I've always considered that working groups should be working with secretaries being the main decision makers about the projects to be brought forward.
Which is narrow the structure as it is now.
Yeah, but something that Luca said, let me think that there are instead other operating modes that are being considered.
And that I didn't get. Like what?
No?
What do you mean?
No, that there are also bottom-up ideas coming up.
Okay.
No, I didn't mean bottom-up ideas. I mean that once, let's say we have a project and then we say, okay, the coordinator of this part is A, the coordinator of this part is B, the coordinator of part is C. Now, you can implement the delivery of this project, which is the sum of A plus B plus C, by either saying, okay, we have one call every month, and then in 6 months, we see if we manage to deliver. And then what happens most of the times is that half of the calls are canceled, and one is not present at one call, another is not present at one call, because that's how things are. Or you can also say, no worry, A is going to deliver no matter what, B is going to deliver no matter what, and they work as sort of independent structures that then only at the end manage to, they reconvene and they say, I did this, I did that, I did that, okay? So this is what I was referring to. So the first model requires the secretary or whoever is supervising the project to be constantly present and constantly reminding that everyone should do what was agreed on. But then there's another way of engaging people, which is saying, you can, once you have delivered your part of the project, then you contact me and I know that your part of the project is final. Okay, this is what I'm trying to get at. And I don't think that we are in the condition where any secretary here has enough time to maintain that level of pressure on their working groups. I don't think anyone is here.
No, but I think
this problem becomes even more important when you actually start collecting the data and doing the analysis, because it becomes really a matter of doing sort of wet work.
Yeah, but I mean, this discussion can become very large. I agree with Gennaro. We may not have the time to do it now, but I mean, the role of a secretary is a rather difficult role, because a working group is not a research group. There's no question about it. So the structure of the research group with leadership being guaranteed through a very hierarchical path is probably not working. On the other hand, I totally agree that if the working group is based on hundreds of bottom-up initiatives, it goes nowhere. So clearly we do need to find a different model of functioning, which in my opinion is based on what is called leadership. And I think it also requires a lot of motivation from the participant to the working group. However, look, the secretary has to exert his own leadership. And I don't think that calling up the people every day is the way to go, but assigning functions and evaluating performance is one of the way to go. I don't know if are there other comments on this?
Giuseppe, so in line with what you have already discussed, actually there is my experience that I went from the very hard life of the secretary of the immunotherapy working group, in which was very difficult to reach all the unit in the working group, and now Paola is taking care of that very, very well, I have to say, trying to involve everyone to a different structure that maybe may help also the work of all of you. Indeed, in this case, for the CAR-T program, we have a completely different structure. In which we do not have only 2 representatives from each IRCS, but there are many people collaborating in each IRCS, and it makes even easy the life, I have to say. And then we have divided all the project in a VP that actually they are sub-project, and for each sub-project there is one responsible. So one of these is Enzo connected with us right now, and this makes the life of the secretary much easier. Because it's very hard, but we do 1 monthly meeting in which we have the contact with each VP leader, and then they are in touch every month with their unit. So this like a capillary way to be in contact with everyone needs to be apical. Otherwise, it's impossible to stay in contact with all the people when you have 1,000 of milestones to reach. So I hope that this can also help the discussion.
Yes, I don't want to, question and answer on my side, but actually what happened is that with your working group as well as with the radiomic working group, we did an exception with respect to a rigid organization with 2 representatives per institute. And it seems to me, from what I understand, that also in the radiomics working group, the presence of many other people allows for a better organization of the work. And I must say that I received the request from many secretaries, for some of you, to be able to expand the participation to the working groups, expanding not to the need of representation of the different institutes as a political need, but expanding in terms of taking on board the people that is needed to reach the goal. So I guess this is something that, Gennaro, we should rediscuss, because as a matter of fact, is coming up from many of you. And that's an important point, because straightforward said, we don't want a political cross-section within the working group of the Institute, but we also want all the people that is needed for the project. This requires flexibility. I agree, Concetta. And again, I have been asked by many of breaking these rules of 2 each institute. But I guess this can be easily tackled. I guess, Gennaro, we can discuss it also in the executive committee, because I have not the power to decide that. This request of flexibility comes from many different working groups. Thanks, Concetta. So listen, I think that the other issues that I posed, the data sharing and data generation, we will move it to the next table of coordination among secretaries, which I guess we take now a couple of weeks rest and then we reconvene the meeting. So if everybody agrees, it's now 15 to 8, and I guess we have to close. I'm very sorry to close this discussion, because we have to take the step of frequently discussing this issue. Thanks, Gennaro, for having raised those. Okay, so if there are no...
Can I have 1 minute?
Sorry?
Can I have 1 minute?
Yes, sure. Come on, Enzo.
Actually, that was exactly on your previous points. So the data sharing and all these things. And the fact that in many cases, the Institute would not commit to dedicating personnel to the projects. I think one big bottleneck is bioinformatics and bioinformaticians. That's a very big, and I just want to try to, I was reasoning on a possible strategic approach to this, which is to find some way for ACC to support and to somehow direct educational programs towards clinical bioinformatics, oncological bioinformatics. There could be some ways in which we could jointly promote the preparation of people, the formation of peoples in that direction, so we don't have to struggle to find bioinformaticians or to obtain the time of some bioinformaticians dedicated to ACC. This is not going to happen in 6 months, but maybe this could be, I just wanted to propose this for a strategic discussion by the board as a possible action.
Look, Enzo, I mean, you're opening another Pandora here, because, I mean, you know how many difficulties we had in the last 1 year to recruit bioinformaticians for the ACC genomics. So it is really an emergent, a very important issue, that of how do we train and recruit our bioinformaticians. Again, I guess we have to move these discussion topics to the coordination tables. So if now anybody has an urgent need of putting another stone on the table, I guess that I can say thanks to everybody, say hello to everybody. I mean, we are closing not only this session, Gennaro, we are closing the meeting. So, I mean, that's a important moment. Finally, was 3 days really very tense.
Thank you, Giuseppe, to all your efforts. You really have been central to everything. So
no, central no, but tired, yes. Now I know that it's politically incorrect. I wish you a great glass of wine, and hello to everybody. Thanks a lot, and see you very soon.
Thank you.
Ciao.
Ciao.
Tutti.
Bye bye.
Ciao a tutti.
Bye bye to everybody.
Ciao a tutti.